So I request the students to hand over the bouquet and welcome the sir. And also I request the co-chairman Dr. Arun Kumar Sarvasmani, Professor of Entomology, University of Agriculture Science, Raichur, to occupy the dais. And one more co-chairperson, Dr. Rashekara K, Associate Professor of Entomology and the Farm Superintendent, KSNUHS Shivamogga, Karnataka, to occupy the chair. And for this technical session, 5, the rapporteur, Tarini KB, Assistant Professor of Entomology, KSNUHS Shivamogga, Karnataka, and Dr. Anjan Kumar Naik, so Assistant Professor of Entomology, KSNUHS Shivamogga, to record the rapporteur report. With this, once again, I welcome all the delegates for this particular technical session program. So then I will hand over the mic to our chairman, sir, Dr. Gidjesh.
Anyway, good morning. First of all, I request all the delegates, please come front because the number is very, very small. So those who are outside the hall, also please occupy your seats because this is the conference. It is a forum for discussion as well as exposure for various aspects of the pest management. So that your main purpose of this program is to active participation. Please, those who are outside, please come inside the hall so that we will start the program, and also all please come front. So this is the technical session, third, with respect to the biological management of fall armyworm. As you know, the biological management is one of the components of integrated management of any aspects, maybe nutrients or pest or disease, whatever. Now the concept is integrated approach for all. So biological management, it is only main, the principle behind is only to have to understand the biological relationship in the nature and also the ecological situation of a particular area, how the organisms exist each other. It is only the application of biological organisms or their products or toxics in many, either in the form of predators, parasitoids, as well as their products. So in managing the pest, it is an eco-friendly approach. It is need of the day. Therefore, so I request all the participants to actively participate. There are one keynote address, 3 lead speakers, and 15 oral presentations are there. Because already we lagged behind by 45 minutes. So I am very particular about the time. So I will not extend that time. For keynote address as well as the lead papers, it is 15 minutes. Out of that, 12 minutes is for presentation, 3 minutes for the discussion. And also for oral presentation, please all of you keep in your mind, it is only 8 minutes for presentation, 2 minutes for discussion. Only 2 questions will be allowed for each speaker, each speech. So only maximum of 2 questions can be asked. If you have any clarifications outside the this hall also, you can discuss, you can have the number and other things later on, you can interact with each other for some more clarifications or the knowledge. With this, let us start with the keynote address by the keynote speaker by Adeni D. Fritas Bino is from on soybean, Embrapa soybean, Londriana, Brazil. So he is going to give his the keynote address, recorded keynote address. So there is no scope for discussion here. So anyhow, you can have any observations out of this his presentation. You can note down. Later on, you can contact over the mail or the over phone. So with this, please allow. that's ಈಗಾಗಲೇ. Afterwards, I will give the instruction at the time of.
Hi everyone, it's a great pleasure to be here with you. My name is Wayton. I am a postdoc at the Brazilian Agricultural Research Corporation, which we call Embrapa. During 4 years of my PhD, I worked with the management of Spodoptera in maize, mainly using Trichogramma cruciosum and Telenomus remus. So today I'm going to share you some of those results with you and its management using the egg parasitoid Telenomus remus. So in Brazil, we have several species of the genus Spodoptera. Here I'm highlighting these four species. However, by far the most common is Spodoptera frugiperda, which occurs throughout the whole country. It's also the most important species for us because it attacks every major crop, and of course, maize is the main host, but it also occurs in soybean, wheat, cotton, and several other crops. In Brazil, we have what we call the green bridge, which is the constant availability of food for pest insects due to the successive cultivation of host plants. In our case, for example, we have two main crop seasons, and in the first season, we plant soybean on almost 47 million hectares, and in the second season, we plant maize on nearly 17 million hectares. So on top of that, we still have other crops such as wheat, cotton, oat, and others, all of which help to maintain Spodoptera population. So, and traditionally, chemical control has been our first line of defense against Spodoptera. But in recent years, however, the adoption of BT crops have emerged and it's very useful, but we still need sustainable alternatives, and the use of parasitoid wasps is one of these alternatives. In Brazil, we already have Trichogramma preciosum registered for the control not only of Spodoptera but of several Lepidopteran pests. Our recommendation for Trichogramma in the management of Spodoptera frugipera in maize is 100,000 parasitoids per hectare, released at least 30 points within each hectare to homogenize their dispersal. And however, uh, the main limitation of trichogramma is its reduced parasitism potential in eggs of Spodoptera due to the layers and scales laid by the moths, but we have Telenomus remus. So because of that, we decided to invest heavily in research with Telenomus remus for the management of Spodoptera frugiperda. And this parasitoid is able to parasitize many more eggs, and the egg layers and scales are not really a problem for it. So after many years of studies, in December of last year, uh, Telenomus remus was officially registered by the Brazilian government for use by companies. So today, the recommendation for Telenomus remus to manage Spodoptera in Brazil is 40,000 parasitoids released per hectare. This recommendation comes from also from a study we have just published in September of 2025, this one, and which I evaluated different release densities of Pteronomus remus in the field to determine an optimal or at least a near optimal density for Spodoptera management. In the same study, I also evaluated the possibility of releasing already emergent feed adults to create a sort of shelf life. And although the paper came out this year, the field work was carried out nearly yearly last year. Yeah, but overall, anyway, this parasitoid has great potential, as we know, and studies with Brazilian populations have shown that a single female can parasitize it, up to 220 eggs, as you can see in this table below. And How to release them? According to the dispersal capacity of Telenus Remus, we carried out, as I said before, at least 30 points per hectare in order to homogenize the dispersal of the parasitoids across the field. And we also need to pay close attention to wind direction. In studies conducted by colleagues in our laboratory, we observed that gnomes tended to move almost entirely in the direction of the wind, which can compromise the control if the release points are poorly distributed. So because of that, our recommendation based on this study is to use at least 30 release points per hectare. In practice, if we need to release 40,000 parasitoids per hectare, these 40,000 individuals must be divided among those 30 points, at least 30 points. So the release, in practice, we do uh, inside biodegradable capsules, as you can see, this kind of capsules with a food source. And this was one of the aspects I also evaluated in that paper I mentioned before. I tested the possibility of releasing Clearnomus remus as adults already with access to food, and we also showed that uh, it's possible to create a shelf life of about 4 and 6 days of storage inside the biodegradable capsules. In other words, if the parasitoid are stored inside a capsule with food, they can emerge and remain inside for up to 6 days without losing their parasitism capacity. So this is very important because egg parasitoids are usually released close to adult emergence, but at that stage the parasitoid are still poop inside the host eggs and cannot protect themselves from the predators such as ants or from high temperatures, which may kill them before they emerge. So being able to release them as adults and even store them for up to 6 days if necessary, it's a big advantage for us. So here is a short video. You can see how we broke the capsules and the parasitoids, the adults are able to fly into the crop. So in the capsule at the top, you can see the food source we use, which is a small piece of cotton soaked in honey, just this. And here we carried out this release using drones. You can see the capsules being released by the drones. It's not easy to see, but it's possible. Just to exemplify, and the companies that sell egg parasitoid usually provided the product together with the drone application. So when a grower buys the product, the company is already responsible for releasing in the field. Another example for the drone, here you can see the adults being released. Here is just the adult, not the capsules. Is how the companies release them. You also can put the food source inside this dispenser, and you can see in the video. It's short video, but it's to exemplify. And each side of this drone can release parasitoids for uh, 10 hectares. And so final considerations, even though Autonomous is already registered, we still have a major challenge, which is mass hearing. This is not a Brazilian issue, of course, it's a global problem. We still need to develop efficient methods to produce this parasitoid on a truly large scale. But in general, we have made important progress, as I showed to you. In Brazil, regarding the use of telenomes, based on many studies, we were able to define the ideal release rate for our conditions, the number of release points, and the best timing for release, which in our case is when the first Spodoptera moths are detected in the area. We also understand the dispersal radius of these parasitoids and know that release must be well distributed across the field. We learned that it can and should be released as adults, already fed and mated, and that capsule-based releases can provide a shelf life of 6 days, at least 6 days. We also studied the impact of releasing parasitoids during the hottest parts of the day and saw that when temperatures exceed 35 degrees, the adult emergence is negatively affected. So that's it. I try to give you a brief overview of our recent events in managing this product of Wikipedia with this parasitoid. And I will leave here the link out to my PhD thesis, in which I conducted 4 experiments, including 2 field seasons, evaluating the combined release of Pteronomus and Trichogramma in laboratory and field conditions. And this work is under review in the Journal of Biological Control and should be published soon. But in my thesis is already written in article format and in English as well. So anyone who is interested can access or contact me by email. I'd be happy to share not only studies from my thesis, but also several works we have in our laboratory.
So now it's end of first lead speaker presentation, sir. Now I request our lead speaker one, Dr. R. Ashokan, sir. So let me introduce some brief. So Dr. Ashokan, sir, so was graduated from Agriculture University, BSc Agriculture from Annamalai Agriculture University, and MSc Agriculture Entomology from University of Agriculture Sciences, Bangalore, and PhD from University of Agriculture Sciences, Bangalore. So his employment record, he started his career as a scientist from Indian Institute of Horticulture Research in 1991 to 1997. Then he promoted as a scientist, senior scientist again in IAHR Bangalore from 97 to 2004. Now he is a principal scientist from 2019. He got a patent also, so that is single step RNAi vector for IHP RNA expression in plants. That is the final patent granted in the March 2018. He got some professional honors. So he got the best Fulbright Nehru Academic Research Excellence Award 2020 to 2021, and he got several national awards. So I will list out. Dr. B.V. David Award in 2018, Professor T.N. Anantakrishnan Award 2017, and Sukumar Basu Memorial Award in 2016, and he participated in several international and as well as national participants. He guided more than 13 students, MSc, and 6 PhD students. So this was his brief biodata. So I request Dr. Ashokan sir to deliver the key lead talk on the current status of strains of fall armyworm in India, sir. So I welcome you to the dais.
So, very good morning, dear participants and members on the dais and off the dais. So, my sincere thanks for having invited me just to make a presentation, because I was not scheduled to make a presentation here. So, it is a kind of a very quick presentations I made. So, I'm not going to go into the details of how the strains have evolved in different parts of the world and how it is relevant to India. So, from practical significance, so the strainal variations are very akin to your genetic grouping in whitefly. So, even though we molecularly we can type that there are several strains are available, whether it is a C strain, R strain, or hybrid strain, but from practical perspective of management, from a biocontrol point of view, from insecticide resistance point of view, it's not going to make much of a difference. Nevertheless, we did some investigative studies way back in 2018. So when we first reported along with other institutions like NBAR and UAS Bangalore, the prevalence of FAW on maize in India. So that was the beginning of our story. Before then, before this FAW, we also reported tuta absoluta in 2014 along with Sridhar from IHR. So quite a few other invasive pests also we reported following that reporting. One is on Franklin occidentalis, that is a western flower thrips, and presence of prevalence of TSWV, that is tomato spotted wilt virus, and quite a few. So our team, when we reported FAW, that time there was confusion whether what was there that point of time in India. So whether it was a C strain or R strain or a hybrid strain. So based on the MTCO1 grouping, so I also forgot to mention. So it is not only about the strain about FAW in this talk, also a little bit included on the genetic pest management. It's also on the part of partially it is towards genetic pest management, part of a newly emerging topic called genetic biocontrol, which is coined by Ethan Beer and also Umar Akbari in UC San Diego. So we'll see how this is genetic pest management also will be relevant in terms of giving a new insight into developing a novel pest management strategy. Okay. So then we followed during that point of time using MTC1, that was a standard technique to decipher the species level. And also in case of US and also the South America, since the population has been evolving over centuries, so they grouped into two categories. That is a C strain, which is mainly infecting corn and other broadleaved hosts. Then we have R strain, which is mainly on rice and narrow-leaved grasses. Then very infrequently, in case of even in US, so we also encounter hybrids. But this was like a runaway selections in case of evolution. So there are two species, they are trying to evolve into a different speciation, the part of a speciation. So when we did with a lot of anxiety using MTCO1, so we couldn't get a real segregation of C strain occurring in India. But whereas we got a mixed result of R strain being present on corn. So that was confusing for many of us. The similar situation also was encountered in Africa when it was reported in 2016. So in the history of FAW invasiveness, even though this migration is quite a common thing, moving from the southern part of US to the northern part of US during winter seasons, and also the reinfestation happens in the southern border. So similar thing was not evident in case of Africa as well as in India. So this was initially found out in a small island belonging to Nigeria, Sao Paulo Tome. Then it was by the next year, by 2017, we could see it is occurring on different crops, mainly on sorghum, mainly on maize or corn, and a little bit on other Gramineaceae family among 45 different countries, and they were moving from northern part of Africa to the southern part of Africa. So Garjan, who was incidentally discovered this FAW, also used this marker, but he couldn't confirm, he couldn't assure that the same category of C strain or R strain happening in Africa. Then there were a lot of international efforts to find out whether there were why there was a infestation or invasiveness into Africa. So is it anthropogenic, that is man-made? Because we didn't hear anything for the past 200 years, like it is going out of its native range of northern South America. For unknown reason, it came in. So the question was asked whether the C strain got introduced or the R strain got introduced. Whether male of a C strain or female of a C strain or male of a R strain or C is C strain. So it was not evident, and very few individuals got introduced. And the mode of introduction, all it is only under speculation. So some they said it came through the aircraft, and some they said that it is the import of produce or the export of produce from Africa to the other part of the world. So, so it was the interesting and intriguing story to unravel why the same thing what the category of C and R strain is not so distinguishable in case of newly invaded places. Then we got to work with CIRAD to in population genomics to find out what is the route of entry from Africa also to India. And also we worked with Rodney Nagoshi from US, and he exclusively working on FAW. So then we found out the haplotypes which are found in India and Africa, they are very quite similar. And currently the strainal category is more of a hybrid rather than true R strain or a C strain. Okay. So how does it matter for a economic entomology point of view? It doesn't make any sense at all, because there are, even though there are very few differences were reported, like very susceptibility to insecticide or efficiency of biocontrol agents, or there are other parameters like your management strategies, but there are very few strategies are different, but practically it doesn't make any difference at all. So it is only from the academic perspective that we are looking at. Even from the pheromonal perspective of the male of each of these strain, how they are perceived, and the crosses, whether it is a unidirectional or the reciprocal crosses, whether they are infertile. A lot of studies are being done by Astrid Groot from Netherlands, but nevertheless they are more kind of an academic work and nothing related to practical significance. So in case of India, in addition to the corn, we also reported from sorghum, rice, ginger, pearl, this finger millet, sugarcane, sweet corn, baby corn, chrysanthemum, marigold, arecanut, micuna, gladiols, and cotton. So Kalesh and so I was more closely associated with Kalesh and Saranabasappa. So there were, we also published a paper on rice strain, which is occurring which was occurring on rice in Gangavati, but at the instance of the Ministry of Agriculture, we had to withdraw that paper, that FAW infesting rice. So that point of time, 2018, we also collected quite a few samples, extensive samples from different places, and then used MTCO1, but we found out more of a R strain being present in most of the populations which are infesting corn or maize, rather than the expected role of a what you call the MTC1 based C strain. Then we teamed up with Dr. Nagoshi. So we did a TPA marker. There is a marker, triose phosphate isomerase. It's a nuclear marker, which is also associated with hostal strainal variations in case of maize. But it did indicate the TPA marker that it's a C strain. But nevertheless, whether it's a C strain or strain, if it makes only a sense in terms of management practices, it is useful. Otherwise, we also had many subgroupings within the TPA markers in the two intranic regions of TPA. And also we compared the haplotype status in India as well as in Africa, that in fact the populations, hybrid populations of Africa actually got into India, and incidentally it came near to what you call the southern border rather than the northern part of India. So we did the haplotype analysis, and also we are part of the population genetic analysis in terms of whole genome sequencing. That paper is appearing, and also we did the endosymbiotic variations among different populations, global populations, with the CIRAD. So there are several color maps we could see that point of time. So we can see what is there in corn strain, and rice strain is more of a dark and with more very dark spots. So the characteristic 4 spots, and there are so many polyphysians that were evident. So With the strainal variations, it is more of a but a hybrid. Now it is more than 80% it's a hybrid. And there are certainly there are variations among conspecific population. Variations are there in terms of different factors or criteria that we use. That is mainly for constraint, no significance for any practical purposes. Okay. So this is the same case with the newly invaded places up to now. We have seen up to the Pacific Islands and also Papua New Guinea and then Australia. So the same thing is happening everywhere. It is most of a constraint, and also it is going to get reintroduced into mainland US, and the strainal changes and patterns is going to be disrupted. Maybe in few years of time, it is going to be inoculated or reinfested into the mainland. Okay. So I'm moving to the next part. Whether so we have seen many different approaches in pest management, like the FAW pest management. So let it be biocontrol agents or insecticide studies on different biocontrol agents like bacteria, like BT. Then we have fungi. We have so many other approaches. So then another approach that could be possibly be used for area-wide pest management and for long-term suppression of FAW is genetic biocontrol. Okay. So genetic biocontrol is nothing but you use a genome editing technique. That is a newly upcoming technique in insect pest management. So either edit the crops for insect resistance. So you could do at different places, like either initial olfaction to the host localization or after alighting. So we can take different targets in editing crops to impart resistance. So that is also a very lucrative area which is being contemplated, and many people are working towards that. That is because we have guidelines for field level introduction at the SDN1 and the SDN2 level of plant genome editing. And also equally there is a possibility to use edit the insects for area-wide pest management. So this is very important when the crop is very important, very commercially viable, and it is cultivated in a larger area. So this is true maybe for red palm weevil, maybe true for Bactrocera dorsalis, oriental fruit fly, or FAW, or even for Helicoverpa armigera. But for unknown reasons, Helicoverpa armigera lost out the race. Like otherwise, it used to be more of a very serious pest in many pulse crops, and in case of FAW, it comes very rarely on the cobs. So with the FAW pest management, if you are able to take care of the crop at 25 to around 40 days at the initial vegetative phase, then you are done. So you are so there is no need to worry much for the later phases. So that is a very crucial period. And also we have to take care before they enter the hole. So whatever we talk, the FAW, there are several agents are effective, but when it gets into the hole, nothing can penetrate excepting for systemic pest insecticides. Okay. So genetic biocontrol is nothing but use genome editing to impart sterility or distort sex ratio, okay, towards males. So unlike SIT is being followed for many pests. Like in case of cotton, Pectinophora is very successfully managed using SIT, and now people are also trying to use a PGSIT for area-wide pest management in case of even mosquitoes. Okay, so this requires validation of your different targets using genome editing, especially using SDN1 level using a RNP complex. Then when you construct a PGSIT, then we have to move to the SDN3 level, making a transgenics of Cas99 as well as the what you call the SGRNA line. So we did some studies by taking different targets, like you have taken SXL gene, which is imparting the pathway of spermatogenesis. We did edit the SXL gene in case of FAW. And one thing is that we have to recall, or we have to take into care of other isoforms, and we have to make a conserved guide RNA, otherwise it is not going to be population specific. So we did the work on Spodoptera frugipeda des, that is delta desaturates in the pheromone synthetic pathway. So I will not go into the details because I have 15 minutes time only. and it requires the cloning of the gene, making a guide RNA, making a in vitro cleaving, then making RNP complex, then inject at the right stage, then go for a molecular and phenotypic validation. So I'm showing how where we have interrupted. So the red cross mark shows where we interrupted in the pathway. So also we need to study how this the protein structure got changed like after we edit. So this is to make whether the charge variations or how this become dysfunctional. So recently we also did the multiplex editing to change wing pattern as well as eye color. So this is a SFTO. And in addition to the popular Cas9, we also employed the Cas12a for editing. So this is the advantage with the Cas12a is minimal off targeting as compared to the Cas9. So we also undertook a prime editor. So this is much more specific than the Cas9 or Cas12a because we introduced a specific edit into the population. So we demonstrated for the first time outside the medical field. So RNP based genome editing is a requirement of the day, because if you employ RNA based or DNA based editing, so it takes more time for the transcription translation. So by the time our G0 stage would have passed. So we have to take into consideration, but there are many difficulties, technical difficulties in achieving this prime editor. This is also more specific even for the crop editing, because we cannot afford to have a off target as well as a non-target effect. So we can we can introduce a stop codon, or we can do whatever you want to do, depending on where you intervene. Like there are several exons, so we need to understand the protein first. Then we have to take up the editing. So this is nothing but Castrol against the Cas12a and Cas9. So we do the RNA template with a mutation that what is to be introduced as a RNA molecule. Then the Cas9 nickase is fused with the reverse transcriptase. The RNA molecule that we supply is reverse transcribed as a cDNA, and then it gets incorporated into the target DNA strand. That's then since our opposite DNA strand has not complementarity sequences, so we also introduce one more nick in the non-target strand, so that the non-target strand gets removed and the cDNA, newly synthesized cDNA gets incorporated, so that there is a specific insertion or deletion of any kind we can do. So we need to understand how the folding takes place and what is the oval base where we can change. So compared to the Cas9, so we have several variations also. We have perfect and imperfect edit, and mostly we have a more perfect edit compared to the imperfect edit in case of Cas9. So when we do the editing, so if we are doing editing of a crop, so through GEAC it can be released, so it can go for pest management. And if you do the editing of an insect, so it has to be driven like a SIT, like a sterile male technique, and all the nipplings parameters, 6 requirements have to be met scrupulously. And also we have to take care of the Hardy-Weinberg expectations, where we also have to know how the population is going to develop a resistance, because this is also going to develop a resistance when it's not that it is very immune to any resistant development. There are several papers which have come up in case of Cas9, that bacteria indeed developed different mechanism of resistance towards infecting phages. So the same thing is going to reflect in a eukaryotic cell also. So we have to, there are more than 15 different concepts in the gene drive. Okay, so the most useful and very easy concept would be CRISPR-Cas9 based gene drive, and there are depending on the migration and depending on the intrinsic rate of development, so we have to decide on the type of gene drive that we have to take care. So more than the CRISPR-Cas9, we can engineer under dominance. Some of the medfly populations have also been designed to have a media gene drive. Okay, so the gene drive purpose is to penetrate the mutation very effectively And also, if there is a larger mutations, based on the hardware expectations, it is going to replace or it is going to come back to the normalcy. But if you are introducing a very subtle mutations, but the effect is very profound. For example, if you are introducing subtle in the oval base, and if it changes the protein folding, if it changes the hydrophobic amino acids into hydrophilic amino acid by misfolding, then your job is done. So we need to understand most of the simulation as well as the protein modeling is very, very important in designing. It's not only in entomology. So we have to integrate with many other subjects and then take care as a holistic point of view we need to treat. We cannot be an entomologist at that point of time. So we also have to be a population ecologist. We also take care of the simulation modeling, statistical modeling, and also beforehand we have to be more technical. And the first and the final requirement has come from the field entomology. So unless it answers the field entomology, it has no meaning. So whatever we do, very fantastically using molecular techniques. So that remains in the paper. Okay. So we have to undergo, take this mutation into the field using gene drive, but the guiding principle is a Knipling's 6 expectations. Okay. So this is the brief. So I didn't want to take a more technical because the audience is not.
So, thank you, sir, for a very good presentation, particularly the new aspects, the genetical management of the pest, particularly through the genetics, and also its potentiality in the biological control of the pest through genetic modification or gene editing. It is a very good and the new aspect of managing the things. I only 2 minutes, 2 or 3 questions may be asked, particularly those who are breeders as well as the entomologist can intervene. How about his presentation and also the questions which you have.
Firstly,
please.
I express my sincere thanks to Dr. Sharan Basappa and having invited me for this talk, because I was not scheduled to attend this, so I came just 2 days back. And also I work closely with Kaleshwara Swami and Sharan Basappa for various projects, because N.K. Krishnakumar is our binding force. So because he was a student of N.K. Krishnakumar, and also my mentor also happened to be Krishnakumar. So we continue to work with these 2 of them. So I did not talk about the crop genome editing because time is less, and also we need to have more technical presentations on the crop genome editing for insect resistance. Let us not bother about other things. That also we are doing on for viruses as well as for insects.
Any clarifications? Yes, please. Please, Mike, whatever.
Good morning, sir. So other than the color morph that we explained for differentiate the corn strain and the rice strain, so are any other methods where we can differentiate the R strain and the hybrid, sir? So because as you said, most of the populations now distributed is hybrids. So is there any chance that because when we go to field, we also need to have a chance where we can identify between the strains. So is there any chance?
So the reliable marker as of now is a TPI, triose phosphate isomerase, that is a nuclear gene which can differentiate the C and R strain. But the thing is that we don't have much populations on the rice now. So it is mainly on sorghum, mainly on maize and little bit on sorghum. But we have done more, we have identified FAW from more than 10 different hosts. These are all very, very incidental hosts. I don't think it is like Arika also we have found here, right here in Shivamogga. But we did not see that development happening up to the adult stage. Okay. So rice strain, there are a few incidents of FAW on rice, but unlike in the US, we don't get that much population on grasses and rice, but it's more towards.
Sir, one final question.
Yeah.
Like yes or no, is the rice strain present in Karnataka, sir?
Rice strain is present.
Okay, sir. Thank you.
Sir, as you told that between the R strain and con strain, there is no difference in insecticide resistant or something like that as in whitefly, right? No, sir?
What I am telling is, so we could say I'm also an molecular entomologist looking through the molecular lens.
Yes,
That's
sir.
all. I also did many genetic grouping with whitefly.
Yes,
So
sir.
we have more than close to 40 different genetic grouping. Okay. So based on MTC1, so many markers we can use, and then
Yes.
we can have strainal variations or genetic grouping variations. But in the field, when you take a sample, so how to differentiate those? And if you have a population continuously from a cotton or from a particular crop, okay, we can show the variations. But we cannot assure that that same population will go to cannot go to the other crop.
Okay, right.
Okay. Similarly, in case of rice and R strain or a hybrid strain, there are subtle variations towards R and rice strain, con strain susceptibility, relative susceptibility to your pathogens, but it doesn't because when you go to a field, so how do you discriminate? So how do you going to differently treat those populations?
Yeah. So they can interchangeably affect both rice and corn. Then how come they have named it like that, sir? R strain and con strain when they have named
Correct.
it?
You are correct. See, in case of Americas, I am talking about the North and South America. This pest is a native pest.
Okay.
Okay, so this has been evolving along the crop for more than 2 to 3 centuries.
Okay.
So the grouping has been very distinct with even with MTC1. But when it is in a newly invaded place, there is a term called population bottleneck.
Okay.
Usually they tend to like I will make it a very simplified point from insecticide resistance point of view. We have BT resistance, so we have amide resistance. Okay, but if you look at the mating choices, these populations which are resistant to certain insecticide or BT, they will try to mate among themselves. Okay, they are called cohort. Okay, so very similarly, the con strain, the pheromonal composition, there is a subtle variation between R and right strain in case of the native range. So they try to be within that group only. Male and female, they try to mate within that group only. But also we have a hybrid population occurring even in Americas. But the frequency is lesser. But why it is not happening in a new invaded places? We did not have much individuals arrived in Africa.
Right. Okay.
So there is a phrase called population bottleneck. So they were compelled to mate because the species cannot give up. So and it is also a watertight. It is not a biological species according to a biological species concept. There is no reproductive isolation is not there. Okay, even though we call it as a one-way unidirectional mating, reverse rice strain females cannot mate with male cannot mate with a female of a con strain, and it is not effective. But it's because of the population bottleneck, all this C and R strain, the classical C and R strain has broken up. They have to survive. So now we will be seeing a different scenario, like in case up to Australia it has gone. Some of the Pacific Islands recently reported. So days are there for a re-infestation into the mainland US. Because we look from the practical perspective, let us see. The subtle variations are not useful. I can see dissect molecularly and show that there are variations. But from practical perspective, it doesn't make any sense.
So the subtle differences which you told about the pheromone, does it actually matter when you design a pheromone or make a pheromone? Like
If you develop a pheromonal trap, these subtle variations are evident.
okay,
From the work of Astrid Groot, we can see the calling time, scota phase differences are there, very classical between R and C strain.
oh right,
But the same thing is not operative either in the African populations or Indian populations. So even they overlap. Okay, so and see another thing, but we with area of pest management in case of pheromones, we never bother about nipplings. See, we don't estimate the resident population. We don't estimate the incoming, outgoing. Simply we put that pheromone. Whoever is caught, then we'll like police. We'll put all the cases onto the individuals. We don't really see how much of dip, whether they are spent to male, spent to female. Nothing outside that. Take put a trap, count some insects, and also incidentally if you see, it's the same effectable trap also attracts many other spotted species. Okay, so there is a cross-tracking happening. So my thing is practically there doesn't have any purpose.
Thank you, sir. Thank you. No more questions.
Thank you very much.
Please, thank
Thanks
you.
for the nice opportunity.
Thank you very much, sir, for your presentation on the current status of strains of fall armyworm in India. So, for this session, I welcome our dignitaries, Dr. B. M. Prasanna sir, Samit, for this technical session, sir. Warm welcome. And also our university Directorate of Education and in charge Vice Chancellor Dr. Himla Nayak sir is also present in this session. I welcome sir, both of you. Now I request our B.M. Prasanna sir and Himla Nayak sir to come on the dais to hand over the token of respect from the organization to the lead speaker. And also I request the chairman and co-chairman to join the hands. Thank you, sir. Thank you, and all. Now I request our this technical session 3, the lead speaker 2, Dr. Arun Kumar Sarvaswani, Professor of Entomology, University of Agriculture Sciences, Rajchur, who is delivering the talk on exploration of biocontrol agents for the management of fall armyworm. So before his presentation, I would like to introduce the brief biodata of Dr. Arun Kumar Osmani sir, who born in 1/7/1971, presently is heading the post of Special Officer of Seeds. So he graduated BS Agriculture from US Dharwad and Master of Agriculture from US Dharwad and PhD from Agriculture Entomology from US Dharwad. So he is having more than 27 years of experience in the farm universities of Karnataka. So he started his career as assistant professor at MARS Raichur, then associate professor at MAS Raichur, then presently professor of MARS Raichur and also the charge of Special Officer Seeds US Raichur. So he has published more than 80 scientific journal articles in the peer-reviewed international and the national journals in high impact factor. He wrote more than 15 books on the chapters, and he delivered the radio and as well as TV talks more than 25. So he has more than 25 externally funded research projects he has handled as a principal investigator with a worth of 8 crores. So he is the man who established the biocontrol laboratory for mass production of biocontrol agents with a budget outlay of 1.5 crore. And he has, as he took the charge of Special Officer Seed, the establishment of seed testing laboratory with a high seed and processing plant with a budget of outlay rupees of 5 crores. So he got the honorary with the international awards 2, national awards 4 and university awards 5. So with this brief introduction, I welcome Dr. Arun Kumar Osmani sir to deliver the talk.
Good morning, everyone. Thank you, Dr. Naveen, for the brief introduction of my profile. Respected chair, co-chair, rapporteurs, and all the delegates. I am here to present the topic on exploration of biocontrol agents for the management of fall armyworm, and University of Agriculture Sciences Raichur and especially the biocontrol laboratory has devoted its work on fall armyworm, and under my chairmanship almost 5 students have worked on fall armyworm in various aspects. I just would like to go through all those things, sir. I think no need to explain about fall armyworm and all, because since yesterday we are talking about the fall and how it is, how the important is this particular pest. I just to directly move to the topic, right? For the management of the fall army or the invasive insect pest, we always depends upon the topic called as IPM. So it is not possible, as yesterday Dr. Prasanna was mentioning, that it is impossible to eradicate the insects, but we can minimize its economic damage by utilizing all the compatible mode of components or the compatible components, so that we can reduce that pest populations. And I just wanted to go through the integrated pest management, IPM. IPM is such a complicated word. No, we always use the term like integrated pest management, but I just used the font of Edwin font. It is very complicated to convince the farmers what kind of components the farmers should use, how to convince the farmers. Very tough job. So the integrated pest management involves many of the aspects, and the major components we include the physical, mechanical, cultural, biological. And I will be focusing more towards the biology, how the biological control agents or the BCA can be employed for the suppression of insect pests. And as you all aware that whenever we talk about the practical utility is concerned, farmers are always dependent upon the insecticides. That is the one of the best component of integrated pest management. And importance and benefits of pesticides. Without crop protection, probably we are unable to achieve the agriculture productions. So therefore, the insecticides plays a vital role in integrated pest management. The only problem with the pesticide is the injudicious use of pesticides or the insecticides. Injudicious. Yeah, I would like to take the one example. India, the amount of pesticide usage is very, very less. The quantum of pesticide used by Indian farmers is very less when compared to any of the other foreign countries. But the only problem is we are using in an injudicious manner, and that has led to the problems like 3 R's. So that is, we call it as resistance, resurgence, and residues. To balance this, the problems or the problems posed by the insecticides, the best alternative which we always the nature has given is the 3 P's, that is we call it as predators, parasitoids, and pathogens. So we can balance the nature, or we can very easily manage the problems which are associated with insect pests can be very easily be counteracted with countered with the 3 P's like predators, parasitoids, and pathogens. On later note, just I wanted what is the status of our biological control? Why we are unable to attain the success in biological control is because it is very tough to catch. This is on lighter note. I just wanted to bring the notice to all the delegates that very tough job is the biological control. We can because we are unable to identify which is the major cause which is leading to the death, the mortality of that particular insect pest. So I am sure everybody is aware of this particular definition given by DBAC as early as in the year 1964 itself. Use of NEPs, predators, parasitoids, and pathogens has to be employed so that we can suppress the pest populations below the economic damage level. So which includes all the biological control includes all these 3 P's and the major issues with the biological control is mainly deals with the 3 methods of biological control. We talk about the 3 methods. One is conservation biocontrol, then augmentative biocontrol, and the classical biological control. And yesterday Dr. Susil has very clearly mentioned, Dr. S.N. Susil, the director of NBR, clearly mentioned that nowadays we have to think of use of the conservation biological control, augmentative biological control, even for that matter. The India has got lot of success with the NBR with respect to classical biological control is concerned. So the bioprotection or the sustainable approach to pest management, which includes to overcome the problems which are associated with pesticides, we can have the other alternatives like we have to intelligently select the tools so as to suppress that particular insect pest. And this is what I was referring with respect to the points that we have to keep in mind whenever you talk about the biological control is conservation biological control, augmentative biological control, and classical biological control. So in case of conservation biological control, the least disturbance should be given to the ecosystems. So we should not go on repetitive use of pesticides, or we should not disturb the ecosystems. So that kind of things we have to do in case of conservation biological control. And in case of augmentation biological control, we are encouraging the biocontrol agents in a such a way that there are two methods which we normally follow in case of augmentative biological control. One is inoculative biological control, and another one is the inundative. So nowadays we are more focusing towards the inundative as well as inoculative biological control. For inundative releases, we have to have we have to establish the biological control laboratories across India so that we can cater the needs of the farmers. So in that context, we are working. And in case of a classical biological control, we have very good examples with respect to like papaya mealybug, cassava mealybug. So that we will be going to identify the natural enemies which are associated with that native pest. And there is a huge demand for the biopesticides or the biological control. There is huge opportunities there in forthcoming years or the in coming decades or so. There is a lot of increase with respect to the demand with respect to biopesticide is concerned. And you can see that the bioinsecticides, which includes the BT, then the diversity is very huge in case of the biocontrol agents. We have bacteria, we have viruses, we have EPNs, entomopathogenic nematodes, then so on and so forth we have. And there is a huge demand is there, and probably this is the area where we have to look for our, especially the young students. Young students, they have to young students have to focus toward this particular area, because they can start as an entrepreneur in case of these biological control agents also. And this is what overall the demand of the biopesticide in global level as well as at national level. And there is huge demand is there, and probably this is the one niche where we have to work out to get to be the entrepreneur in case of biological control. And the same thing, the growing demand for sustainable agriculture and the limitation of chemical pesticide is now is demanding for the use of the biological control. Integrated pest management strategies mainly depends upon the use of biological control. So therefore, there is a lot of demand for biological control agents. And I just would like to go through one or two case studies where our students, my students have worked on use of biocontrol agents on fall armyworm. And you just see here, we have collected more than 4,477 eggs of FA, and we could able to get the percent parasitization of around 40 to the percent parasitization ranges from 2 to 85% parasitization across the 5 districts of northeastern Karnataka. I just wanted to, and the proportion of adult emergence or the proportion of percent parasitization was, it is mainly represented by two egg parasitoid. One is trichogramma and another one is telenomus. I think the keynote address given by the keynote address, they mentioned that the use of telenomus. In the similar fashions, the Indian we are mainly focusing on the exclusive egg parasitoid like trichogramma as well as telenomus. And many of natural enemies are associated with fall armyworm, and this is the area which I am talking about is northeastern Karnataka, that is Raichur, University of Agriculture Sciences Raichur, which falls under intensive agriculture, where 99% of farmers, they are solely dependent on pesticides. Under such ecosystem also we could get lot of natural enemies which are associated with fall armyworm. So this is one of the case study of one of my student work. And we could able to get around more than totally 18 natural enemies were recorded across the 5 major districts of northeastern Karnataka. And among that, the 2 egg parasitoid like Trichogramma pretiosum and the telenomus were found to be very, very effective in suppressing the pest populations. And we also given the ad hoc recommendations, because we are associated with ASRP and biological control. And with our network project as well as our students work, we could able to recommend the farmers that use of Trichogramma pretiosum, that is exclusive egg parasitoid, at the rate of 1 lakh per hectare per 3 releases, consecutive 3 releases has given has successfully suppressed the fall armyworm. And we always recommend that the trichogrammatidis or the egg parasitoids should be released when the crop is around at 20 to 30 days after sowing, because that is the age, that is the time where you find the fall armyworm lays eggs like anything. So that is the stage where we have to look for the whenever we talk about the egg parasitoids. And one more work, with this particularly the previous work, we started taking up the mass multiplication of trichogrammatids, and we are now taking, we have established large scale production of trichogrammatids, and we are supplying to the farmers. And the another study which was carried out was with respect to use of metarhizium relay. This is the very, we could get lot of natural epizootics with respect to the metarhizium relay is concerned on fall armyworm. And we could see that across the 6 districts where we have taken up the survey work, we have found in one or the other farms we are able to get the cadavers or the natural epizootics was there on maize. And sir, surprisingly we could see at the time of cob filling stage. So many of the cadavers were used to be found on the cobs, where at that time we collected many of these entomopathogenic that is fungi, that is metarhizium relay. And we have done all the morphological, molecular characterizations, everything has been done, and we have done the bioassay of the metarhizium relay, and we could get out, we could get the best of the best, the strain which was very effective was like USR BC, that is MR3, that is metarhizium relay 3. All these the best of the best strain which was found during the bioassay, like even with respect to the toxicity as well as the LT50 values, because time is very important here. So looking into the lethal time, we have selected the strain and we started mass multiplication of this particular isolate also, and we started almost all in the form of commercializations. We have started the work on this particular entomopathogens. So MR18 and MR19 were the 2 strains which we have started large scale production at our university. And though we talk about more about biological control agents, the major bottleneck is the availability of these biocontrol agents. Very, we talk every day, we talk about biological control, but availability is the major bottleneck. So keeping in that view, the University of Agriculture Sciences Raichur has started establishing the laboratory, and also started the multiplication of the potential biocontrol agents, especially on fall armyworm. And this is the how we are taking up the mass production of fall armyworm. The metarhizium relay, that is the biocontrol agents, biocontrol agent. These are some of the pictorial diagrams, the pic forms of the laboratories. And we have taken lot of extension activities, the basic way to move from lab to land. So we started doing the large scale demonstrations in farmers fields, not only the for fall armyworm, even other pests also we have taken up large scale demonstrations. Then field demonstrations, these are some of the glimpses of the large scale demonstrations. During COVID period also we have we took the activities of proliferating the technologies of biocontrol agents. So these are some of the glimpses of the large scale demonstrations, and that has led us to take up the large scale productions. I just go through some of the success stories. This is how the farmer used to come to our laboratory to purchase or to have the interaction with the scientists of the universities about the biocontrol agents. So this is there is one saying, sir, many times a farmer won't come to the universities, but this is the reverse case where the farmers are regularly coming to our laboratory, and they are purchasing and they are discussing their problems, and we are supplying the biocontrol agents to the farmers of not only the North Karnataka, even in across the Karnataka, we are supplying the many of the biocontrol agents, especially the trichogramma and metarhizium relay. And sir, last week, last 15 days back, we are getting lot of intents from Haveri, where the maximum area is under corn, especially under maize, and many of the farmers, they are taking up this our biocontrol agent, that is metrosome relay, to suppress the fall armyworm in Haveri areas also. So these are some of the glimpses where we have done a lot of activities to to bring the awareness among the farming community that even the biocontrol agents can also serve the farmers. So these are some of the activities we have done, and why there is lot of research and development is happening with respect to biocontrol agents. There is lot of opportunities. These opportunities can be made and can be taken to further to take up the mass production of biocontrol agents. Organic farming, the government of India is spending lot of money for to promote the organic farming, and the main key factor for organic farming is the use of biocontrol agents. So there also we can very well fit into take up the large scale productions, and these are some of the schemes through which also we can take up the mass production and we can supply to the farmers. And the major challenges and considerations for biological control is the scalability is a major issue, effectiveness, because unlike in case of insecticides, the farmer within a minute will look into it whether the knockdown effect will be there, but it is not so in case of biocontrol agents. Then market awareness, then lastly the regulatory frameworks, as yesterday Dr. Susil sir was mentioning. So BT was coined 1901, but still we are getting the issue with regulatory aspects. So that is the one point which making us, especially the universities where they are taking up the mass scale, large scale production of biocontrol agents. The major issues lies with the regulatory frameworks. There is a need to change the mindset of the regulators so that the biocontrol agents can be pumped into the market. So I sincerely thanks the organizing secretary and team of the National International Conference on FAR 2025, and the officers of the universities and my team, that is biocontrol team. Thank you, and thank you for the opportunity. Thank you very much.
Thank you, Arun Kumar Osmani, for your excellent presentation. As we know, the north, particularly the Karnataka, from the place where he came, by looking to the cropping pattern and also the availability of the water, the cropping system itself indicates the lot of pest problems. Farmers in that area are bounded and also compelled to use a lot of pesticides. It is the area where they are using huge amount of pesticides. Under the given that situation, there is a lot of scope for the biocontrol agents because of the awareness created among the health as well as environment concern. And also he explained about the contribution of the UAS Raichur, because in our state the UAS Raichur made a dent with respect to the biological control. And also he showed through his slides the contribution made to the farming community by demonstration of the so many biocontrol agents. He explained about the potentiality, how we can explore the possibility of using the biocontrol agents in managing the pest and diseases which are eco-friendly. Thank you for your nice presentation, sir.
Thank you, sir.
Thank you very much. Only two questions from the delegates. If you have any clarification, please. One or two questions if you have. No, that's what, because he is presented as a success stories. No, nothing is there. It is only practical aspects is present. Thank you, sir. Thank you very much.
Thank you very much.
Thank you very much, sir, for your wonderful talk on exploration of biocontrol agents for the management of fall armyworm. Now I request Dr. Hemala Nayak sir and Dr. B.M. Prasanna sir to come on the dais, and also I request the chairman and the co-chairman to join hands to give the token of respect from the organization. I also request our organizing secretary, Dr. Shanabasappa, to join on the dais. Thank you very much, sir. Now we'll break for tea for 5 minutes because all the keynote speakers and the lead talk is completed. So after the tea break, we'll be continuing with the regular oral presentations. I request all the presenters, please load your presentation so that we can complete in time. Okay, so there are total 16 presenters in this session. I request one by one to upload your presentations if you are not uploaded. So we'll reassemble here. Now it is 11:10. We'll reassemble by 11:20 in the same hall. Thank you. I request the three more presenters. One is Sonali, please upload your presentation. And one more, Annamalai Muttuswamy, please upload your present. Yeah, please. Fine, sir. Then one more, Shiva Prasad, please upload your presentations. So there are total, Sonali, please upload your presentation. So there are total 16 presentations, out of which two were not presenting and one is recorded video will be putting. So the remaining all presenters are here and they have uploaded their presentation. So I request the chairman of this session to continue this oral presentation.
So it is, please delegates, please listen to me. It is 10 minutes is allotted for each speaker, out of which the 7 minutes one first bell will be given. So it is a caution for you. By another you can extend by 1 more minute, that is 8 minutes you have to complete your presentation. Only 2 minutes for discussion. So you have to go brief. Don't give much importance to the introduction and other things. You stick on to the important points and restrict and complete within stipulated time. Please, because we are evaluating, so time is also got the marks. So we have to stick on to the stipulated time. Okay.
Sir, the press presenter is Cyril L. Tura. So he is Davao Oriental State University from Philippines. So we'll be uploading his presentation. It is recorded talk.
Good day to each and everyone. My name is Sarve Elchura. I will be presenting under Team 3, Biological Control for Armyworm with the title, Integration of Biopesticides Field in Laboratory Efficacy of a Synergistic Bacillus thuringiensis neem oil combination for fall armyworm, control, and maize. So let's proceed. Introduction. Fall armyworm is an invasive and highly destructive pest first detected on maize in the Philippines in 2019. By 2021, it had already shifted its host range to rice, posing a significant threat to national food security. Farmers commonly rely on synthetic insecticide, but this approach carries problems with resistance development, high cost, and environmental hazards. Our study therefore aimed to evaluate a sustainable IPM strategy using combination of Bacillus thuringiensis isoi and neem oil to reduce dependency on chemicals and to prevent resistance. So the study objectives of the study is to determine the effect of neem-beating combinations in relation to disturbances on the biology of fall armyworm under laboratory conditions under phase 1 and phase 2, to determine the effect of neem-beating combinations to maize growth stages and yield under field conditions. Number 3, to evaluate the cost-effectiveness of neem-Bt combinations for controlling fall armyworm and maize as compared to conventional insecticide treatments. And last, to provide recommendations for the use of neem-Bt combinations as a sustainable pest management strategy for fall armyworm and maize. In the materials and methods, the study was subjected into phase 1 and phase 2 using third instar flower larvae and applied treatments using 10-second leaf dip assay. The treatments included Bt alone, neem oil alone, in their combinations in a synthetic control. Emamectin benzoate opti-5, we measured both lethal and sublethal effects, including mortality, pupil deformities, adult longevity, and egg production. Data were statistically analyzed using ANOVA and toxicity test at 5% significance using R program analysis. For field trial, we use open-pollinated maize variety using randomized complete block design. The top-performing neem-Bt treatments from laboratory phase 2 was applied weekly between 7 and 66 days after sowing. We collected data on maize damage rating, number of larvae, larval infestation, and the presence of beneficial predators. Applications were made late in the afternoon to maximize efficacy. So the results, there is a trend between the efficacy in the laboratory under phase 1. So as you can see, the action confirmation combined NIM-BT treatments confirmed complementary modes of action where BT, mid-gut disruption, NIM oil, feeding hormonal interference. Therefore, in mortality, the combined NIM-BT or NIM-BT in NIM alone in NIM-BT also confirms that there is a significant role wherein these combinations demonstrated a crucial delayed synergistic mortality, strong sublethal effects in offering robust foundation for IPM. So in the data, the top doses validated in phase 1 were in the applications, the higher the concentrations on NIM in BT, the higher the mortality, compared also to control, which is the emamectin benzoate, which is superior in terms of mortality rate in some of the parameters in this table, where you can see the emamectin benzoate is superior, NIM oil is the second, BT is the third, when water spray confirms no mortality rate over time. So the sublethal effects recorded in the insects, Podoptera frugiperda, we have the malformed pupae, we have the deformed adults and wings, we have its few neonates and few laid eggs. At the same time, the larvae is having difficulty in proceeding in the fourth instar. So as to summarize, NIM-BT treatment significantly impaired fowl fitness and overall reproductive potential. It summarizes also that NIM-BT showed delayed and cumulative toxic effects that disrupted larval and pupal development, indicating an additive or near additive interaction under laboratory conditions. As to summarize also, the individual effects of emamectin, NIM-BT combination, and water control were in water, normal development, emamectin benzoate, total inhibition of pupation and reproduction, complete sterility and adult suppression, while NIM-BT combination have strong sublethal inhibition near sterility and delayed sublethal effects and reproduction suppression comparable to chemical. So as a summary also, the sublethal effects and deformities observed between emamectin benzoate and NIM-BT application, wherein in NIM-BT combination, the larvae is struggling on shedding E6-UBA, proceeding to fourth instar and star feeding, while on the emamectin benzoate, the curling of the larvae due to the effect of the emamectin benzoate or the control. In the field results, NIM-BT significantly reduced maize leaf damage, almost as effective as emamectin benzoate. Damage ratings dropped from over 6 in the control to less than 1 with NIM-BT. In the graphical representation, we see that NIM-BT performs closely well with emamectin benzoate in reducing damages. In the field results also, beneficial predators such as paper wasps and spiders remain abundant, indicating that our bipesticide combination is compatible with natural enemies. In terms of cost analysis, NIM-BT application saved around 460 pesos or in equivalent to $7.94 compared to synthetic insecticide, totaling over 2,762.40 pesos or equivalent to $47.63 or 42% less over 6 applications. So in conclusion, in summary, the neem oil in BT combinations offers sustainable, effective, and cost-efficient strategy for fowl management in maize. It performs comparably to synthetic insecticides, but it's safer for the environment and beneficial organisms. Technically, a proper dosing is crucial to avoid phytotoxicity and ensure consistent efficacy. At a policy level, we recommend accelerating the registration and integration of natural biocontrol products into the national IPM programs. This will help ensure long-term environmental sound pest management in the Philippines. Into the acknowledgement, the authors sincerely gratefully acknowledge the support of Dr. Elvin R. Bugwa, Campus Administrator of Governmental State University, with the students, Javen Tadeja, Jerome Kabatuan, and Warden Gonzalez, and also to my mentors, which is Dr. Ravindra Joshi and Dr. Larry Aceres. Thank you for your kind attention.
So, thank you, Cyril, for the presentation. Now, I request our next presenter, Om Prakash Navik.
Hello, I'm audible. Am I audible? Louder. Okay, sir. Good morning to all, respected chair and co-chair. I'm Om Prakash. I'm presenting my work on the ekparasitoid of fall armyworm in maize ecosystem. As you know, the fall armyworm is invasive pest in India, and due to its highly invasive nature, and then it is a migratory also. And due to nature of the polyphagous pest, it feed on the more than 300 host plant in worldwide. And as India is a tropical country, and we know that in the absence of diapause, it passes through several generations in a year. That's how we become a destructive pest in India with a high biotic potential. So any insect pest it land in a new geographical region. So the chemical control remain the first strategy to an emergency basis to control the pest. But what happened, the crop like a maize, it having a dual use in India like a feed and fodder. So in that case what happened, then biocontrol is one safer alternative that we can use in long run. So coming like in biocontrol, indigenous exploration of indigenous natural enemy is very important compared to the bringing the natural enemy from the native country like under the classical biological control. So in this case, the exploration of a native parasitoid is very much important. And coming to among the natural enemy, the egg parasitoid is very important because the nature of egg parasitoid itself says that it kills the pest at their egg stage before they cause the actual damage to the crop. So with this idea, we have formulated an objective on the survey and collection of native egg parasitoid of fall armyworm, and then also to assess their natural parasitism occurrence in the maize as it is a preferred host in India or in the global scenario. And also we went to one more step of one more objective. We have formulated that what actually happened in the season, how they are varied in different seasons, their occurrence and parasitism of this egg parasitoid. So under material method, what we did, we have collected a sample. We have sampled 12 Indian state and union territory, and around 370 field we have surveyed across the 12 states and union territory, and over 3,000 egg masses of fall armyworm we have collected. And then what we have done, we have brought those egg masses to the laboratory, and whether they are parasitized or unparasitized, and then we have incubated in the laboratory condition to check them whether they are parasitized or not. Once the parasitoid is emerged, we have assessed their parasitism rate, and then we have identified parasitoid based on the morphological and molecular identification we have performed. And then we have, after doing this all process, we went to study the seasonal parasitism, where we have taken 3 seasons as in India, Kharif and Rabi and summer season, and we estimated that what effect or what is a seasonal occurrence happen or seasonal parasitism, and any effect of the season is there or not. And that this whole idea will help in the formulating biocontrol programs, whether we have to go which parasitoid is good and what season we have to prefer them. So with this, what we got a result from the overall survey and collection, that around 20% eggs, precisely is 19.65% of total eggs we collected is parasitized by the egg parasitoids. And then what we found in the parasitized eggs is from the 20% parasitized eggs, we identified a 2 parasitoid like a Trichogramma chilonis and the Telnomus remus. And these two, the overall we have studied the composition of these 2 egg parasitoid after identification morphological and molecularly. 52% of eggs, whatever we collected across the India, were parasitized by Trichogramma chilonis alone, and then 70% around is parasitized by Telnomus remus. But interestingly, what we found that around 20% egg masses had a parasitism of both Trichogramma clonis and the Telnomus remus together. And then further we went to assess the state-wise parasitism rate of both the parasitoids. Then we found that except Tripura, whatever other states we have got a parasitism range in a different range from the 4% to around 23%. As our result says that the parasitism rate was highest in Karnataka, followed by the Maharashtra. It is because of we have taken a frequent survey in the Karnataka and Maharashtra compared to other states. And similarly, we found that in case of Telnomus remus, only we got a parasitism and occurrence of Telnomus from the 5 states. And same way we found that Karnataka, due to more number of sampling we done in Karnataka, we found that around 35% awake masses were parasitized by Telnomus remus, and followed by the Andhra Pradesh around 33%. And then we went after knowing the occurrence of this top parasitoid in the maize, we also assessed any effect of the crop age or the crop phenology has any effect on the occurrence of this egg parasitoid. And to our surprise, we came to know that the higher occurrence of this egg parasitoid, we found that the crop phenology has significantly affected the parasitism and occurrence of these both parasitoid, either both or alone Telnomus remus or Trichogramma clonis. So higher occurrence of parasitism and occurrence and parasitism recorded in the early vegetative crop growth stages is precisely, I can say, from the seedling stage, like from 15 to 30 days. We categorize this early crop growth stage up to 30 days, and then mid crop growth stage from the 30 to 45 days, and then the reproductive stage from 45 days onward to harvesting. So our result says that the early crop growth stage we found that the higher number of parasitism and occurrence by these both parasitoid, followed by the mid crop growth stage, and then the parasitism and occurrence of this parasitoid decline to the reproductive stage. So there was this crop phenology had a significant effect on the occurrence of these both parasitoid. And coming to the seasonal parasitism of egg parasitoid, we studied in Kharif and Rabi season, and we found that the occurrence of both parasitoid was significantly different in all the 3 seasons. And both parasitoid occurrence was higher in the Kharif and Rabi compared to the summer season. But what we found in here, interestingly, is that the Trichogramma clonis was higher in the Kharif and Rabi season, but it declined in the summer season. And the same time, the Telnomus remus and the combination of both species is increased in summer. So there is a reason, maybe because of the ample availability of egg masses in the Kharif and Rabi season, and the more acreage of area sown in the Kharif and Rabi season as compared to the summer. So coming to the conclusion that we have surveyed around half of India, 12 states, geographical different states, and then 2 species we collected, identified and assessed the natural parasitism, and we studied the effect of phenology on the occurrence of this both parasitoid, and then go into depth to study the seasonal parasitism, so that we can understand what stage natural parasitism is there, and how much we can add through the inundative release in the field, and what kind of parasitoid can be added in what stage. So our conclusion is that Trichogramma clonis and Telnomus remus can be used as alone or in combination to control the fall armyworm in the maize. Thank you.
Topic is open for discussion. Any 2 questions from this one? Audience, only 2 questions. Yes.
Congratulations, Dr. Om Prakash, for your beautiful presentation, which mentioned that half of the India is already surveyed, and you found 2 parasitoids. It's good. But what about egg larval parasitoid? You have not mentioned egg larval parasitoid, because egg larval parasitoids are playing a key role in management of fall armyworm.
Sir, we have surveyed egg larval parasitoid also. We have reared, we got 2 species,
Coccidigium
black and Clonus formosanus.
Okay.
So on that, Dr. Prabhulinga is working exclusively on egg larval parasitoid.
Okay.
He will be delivering lecture on this.
Okay. Okay, thank you.
So, but their occurrence is there almost what we say that to my personal knowledge, is around 20% parasitum we have recorded in the Karnataka, and that already we have published in journal.
So only Chilonus is present only. What about others, Trichogramma species?
Sir, to the order we have said, we have come across only one species as a natural parasitism.
Okay.
But as people are
Pretiosum
using,
is also working.
Pretiosum also is recommended, and Chilonis is also recommended for augmentative release in the field.
Okay.
But as naturally what I got a one species right from the field as a natural occurrence.
Thank you and best wishes.
Thank you, sir.
Okay, there is no questions, sir. So you have mentioned what are the reasons for variation across the states with respect to the parasites?
Sir, the one this all we have to analyze, sir. We have recorded the data, but since it is a long-term study I am doing since 2018, so there may be the climatic effect, sir. Second thing is that one reason what I found that Trichogramma chilonis is more occur than the Telenomus is that because we are releasing a lot of Trichogramma chilonis in the field, inundative release compared to the Telenomus. This is the one reason because this, and the second is the climatic effect, and there is the area coverage.
No, you have to be very specific. Don't explain things. When you ask whether it is whether it is a cropping pattern or the weather conditions. So
Sir, cropping pattern also affects. Second is crop phenology also affect, and then what you said, the geographical area because sowing acreage.
okay. No more questions, please. Thank you. Thank you very much, Navik.
Okay, thank you so much.
Next speaker. Next, I call upon Rupa S. Patil.
Good morning to all of you, respected chair, co-chair, dignitaries of the dais, and other senior teachers. Myself, Dr. Rupa S. Patil. I'm working as senior scientist and head entomology at ICAR Krishi Vigyan Kendra, Uttara Kannada, which comes under UAS Dharwad. I'm working in the KVK system for the past 18 years. Majority of my, it's not like a research type as all are presenting. It's just my observations during the survey work. And the topic it is on occurrence of invasive fall armyworms, Podoptera frugipada, and natural epizootics of Metrrhizim raleigh from finger millet fields of Uttara Kannada district, Karnataka. Next. Yeah. As you all know, this invasive alien species, they are the significant threat to agriculture. This invasive polyphagus, this Podoptera frugipada, it's native to Americas. It's causing a severe pest on maize, and it is also causing a major damage to more than 350. As I just observed here that it's more than 350 host plants, including cereals, oilseeds, and the horticulture crops. And coming to our district, Uttara Kannada, it comes under Western Ghats, and it covers both hill and the coastal ecosystem, and one of the biggest districts in Karnataka. And we receive very high rainfall, up to 5,000 mm rainfall, and the predominant cropping system is arecanut based cropping system. Spices are the major ones, and among the field crops, rice is a rice based cropping system. We are not is there, and other major field crops are in Kharif are the sugarcane, and BT cotton is there, and nowadays the BT cotton area is replaced by maize. And whereas this finger millet, that's ragi, though it is a dryland crop, in our area it is grown in small pockets in guntas. It's limited for the home consumption.
Lot of cross talkings are going on, please.
And regarding to the objective, there is no specific objective with respect to FAW. But during our KVK system, we usually do regular survey and surveillance for monitoring of insect pests and the diseases, along with the department officials, especially the Department of Agriculture and Horticulture. This was one of the our monitoring studies that we done during 2021. Unfortunately, I couldn't publish, but I thank the organizer for giving me this opportunity. And regarding material methods, we used to do regular field surveys to know the incidence of insect pests and diseases in farmers' fields, mainly on paddy and sugarcane and other major crops of our districts. But during our survey, we noticed very severe incidence of FAW on finger millet in the farmers' fields in Yellapur Taluk, which is a actually that's a forest area is more in that area, but farmers they do cultivate this paddy, sugarcane, and bt cotton. And when we visited that time, the crop was at reproductive stage, and the surrounding, it was surrounded by the rice and the sugarcane. And those fields were actually, we went to visit, we visited those paddy and sugarcane fields, but accidentally we saw the severe incidence of FAW on finger millet. And so there only we took the observation on person damage, and also we noticed natural epizootics of this EPF, that's and it was identified as metrizim reli, and even the percent infection was also calculated, and we made observations in 2 months, September and October. Actually, this was noticed in September. And regarding to the results, that the damage extent was to the tune of 97.56% with respect to FAW on finger millet, and very little yield. I can say it is nothing. That's farmer, he got 28 kg, just 28 kgs in half acre of from the finger millet field. And regarding FAW observations, 2.56 larvae per shoot were observed, and the damage symptoms, it was similar to that of maize, like early instars larvae, they scrape the chlorophyll and leading to the elongated white patches, and later instars they fed on the shoots and leaves, and also they caused very severe damage to the earheads, and fecal pellets were noticed on whorls, infested shoots and the fingers. Here you can observe we have noticed all the instars of this FAW. And regarding this natural epizootics, 2 samplings were made in September and October. 54.29% of infection was noticed due to this metrizim reli, and these are the some picture photographs showing. And there we have also noticed this sporulation was also noticed, and usually we notice the white cadavers mainly, but in our case there because of the congenial conditions, this sporulation was also noticed. And coming to the conclusion part, though this finger millet, no, it is a dryland crop and it is a very hardy crop, and it doesn't require any specific plant protection measures as such, and even no major pests were also reported from this finger millet. But looking to this situation, and this this FAW has caused a very severe damage and severe reduction in the yield also, and the affected panicles, they become unfit for the consumption, and this necessitates for the early monitoring of the pest. We can say it is a key in for the timely management measures, and further studies are required to determine the damage potential on FAW on finger millet, because in our district it may be a very limited area, patchy area, but in southern Karnataka and other places it is grown in lakhs together area. So certainly studies are required to know the damage potential, and integrated strategies required for the effective management with promising EPF. Here I can say metrizim reli is one of the main component. So acknowledgement, ICR Attari, Zone 11, and our UAS Dharwad. And so thank you so much. Thank you for the opportunity.
Thank you, madam. A good presentation. So as we are always talking about this Odoptera only on maize and other things, here is an interesting that how it is affecting a particular finger millet, that in the non-traditional areas like Uttara Kannada.
Yes, sir. Yes, sir.
And I ask one question, so I hope that you have listened to the Ashokan talk.
Yes,
He
sir.
expressed about the C type and R type.
Yes,
how
sir.
you classify this one, whether it's C or R or into C, because as his explanation, it is a narrow-lived crop. So
Yes, sir.
if you, of course, if you are not an expert, you can, it is an input for other entomologists to study on that with respect to genome and other things. So thank you very much.
Yes,
Is
sir.
there any clarification or two questions from delegates? There are no questions. Thank you very much, madam. And it is an interesting. Please, entomologists, look to this one. Ragi, Nuttara Karnataka affected by Sodoptera.
Yes, sir.
Please.
Thank you, sir. Thank you for the
Why
point.
not in dry areas, Maidan areas?
Sir, actually, I forgot to tell while searching for the information in the same year, same months, 20 September and October in Gujarat, Dang district also, they have reported, sir, this one on finger millet. Apart from that, I couldn't see because. Thank you, sir. Thank you.
Okay.
Respected chair and co-chair of the session, rapporteurs, and my dear delegates of the conference. So good morning and welcome all of you for my presentation. So myself, Dr. Prabhulinga, scientist in ICR NBR Bengaluru. So here I am to present my work on bioecology of egg larval parasite Kilnus black bunny of maize fall armyworm. So since yesterday we all listening the management aspects of fall armyworm, mainly the biological methods. So in biological methods also, so majorly macrobials and microbials we have seeing. So in particularly in macrobials, the egg parasites have been covered. Like sir was also asking, so egg parasites only egg larval parasites not. So this is about the egg larval parasites. Just like Trichogramma parasites and Telenomus parasites, the egg larval parasites also play a crucial role in the management of fall armyworm. And not only the fall armyworm, in a different crop ecosystem, like in cotton also, bollworms can also be managed effectively by using this egg larval parasite, this particularly Kilnus black bunny. So we all know that the genus Kilnus is having around more than 1,000 species across the globe, and which are known as egg larval parasites and the majority of the polyphagous leptopteran pest. And in particularly in fall armyworm, around 11 species of the Kilnus have been recorded globally. So out of which India is recorded so far 2 and yet to report some. So and field parasitism also have been recorded by this egg larval parasite Kilnus black bunny and Kilnus formosanus. And by considering the occurrence and the parasitism of the Kilnus black bunny to the fall armyworm, the present study was conducted to see what is the species composition of this egg larval parasites in maize, and what is its biology on natural target host and laboratory host also, and to evaluate the field efficacy. So to collect the Kilnus species from the maize fall armyworm affected fields, so we surveyed in Karnataka and the maize growing regions, and we collected the population, and the Kilnus black bunny, the biology we studied both on the natural target host and the laboratory host Carceres cephalonica, right from the egg to adult period, and also we worked out the host preference studies and also the parasitism efficiency under both laboratory and field condition. So in total, we conducted around 23 surveys in 5 districts of Karnataka. So the collected population subjected for identity through conventional taxonomy and also through molecular characterization. So with the study, we could able to see the two major this one egg larval parasites, which are Kilnus formosanus and Kilnus black bunny. And the percent parasitization of these parasites recorded in the natural ecosystem was around 12 to 24%. So out of these two species, the majority of the dominant species we found was Kilnus formosanus compared to the Kilnus black bunny. So around 82% of the populations were Formosanus and only 18% were Kilnos blackberry. Due to the not suitable for laboratory rearing, so we could not able to rear the Kilnos formosanus. So we continued with Kilnos blackberry. So when we studied the biology of these particular parasitoids on both target host Spodoptera frugiperda and Kilnos Carcera cephalonica, there is a significant difference observed with respect to different life stages of the parasitoid, right, from egg to the adult period. And compared to in natural target host and laboratory host, the time taken by the parasite to complete its developmental period was less shorter in case of the natural target host Spodoptera frugiperda. However, with respect to the fecundity was quite high in case of frugiperda, natural target host, than compared to the cephalonica. And coming to the host preference study, so after collection, so the whatever the populations of Kilnos blackberry, that we reared continuously and the natural host, that is Carcera cephalonica, under laboratory condition. So due to continuous this one rearing, the conditioning of the parasitoids was more stronger to the natural laboratory host compared to the target host. When the laboratory host we tested for host preference studies under choice and no choice condition, the parasitoids were more inclined towards the laboratory host immediately compared to the natural target host frugiperda. Within few seconds, the Kilnos blackberry reached the eggs of Carcera cephalonica, however, and started parasitizing. However, to Spodoptera frugiperda, though it is a natural target host, but it took more than 20 minutes to reach to the eggs of natural target host, because it is due to the continuous rearing on the laboratory host. It gets conditioned to the olfactory cues which is emitting from the laboratory host. So in order to break, it's observed in both choice condition and no choice condition, but the similar results we found when we tested even against the other host likes Pectinophora gossipella, Aereus vitella, and that also the similar kind of this one observations we got. So due to under no choice conditions, it got inclined to laboratory host. So when we started continuously rearing it on the natural target host, that is Spodoptera frugiperda, though initially in the first generation we got around only 5% of parasitagium. So however, in the whatever the adults we got in the first generations, though so we subjected to frugiperda eggs in the next generations. So in the second generation, so we could able to see around 19% of parasitism. So there is a drastic increase in the parasitism. It means that it started accepting the eggs of that natural target, and in the third generations we could able to see even increased percent parasitism, like 28%. So what I mean to say is that so due to conditioning of this parasitoids to the natural target host, we can increase the percent parasitism efficiency. So also we have tested this Kilnos blackberry under field conditions through case studies. So initially the laboratory cultures of Kilnos blackberry gave minimum parasitism. So when we started conditioning to the natural target host, that population was giving a good percent of parasitization. So with this, I would like to tell the house is that just like egg parasites like Teleomas and Trichogramma, egg larval parasites also play a key role in sustainable management of fall armyworm through biological means, and we can increase the efficiency of these parasites through olfactory conditioning. So thank you.
Thank you, Prabhulinga.
And I would like to acknowledge, thank the Director NBR and ICR New Delhi for the opportunity. Thank you.
Now it is any questions regarding his study? Please.
So congratulations, Dr. Prabhu Raja,
Thank you, sir.
for your wonderful work. So there is one, not question but query.
Yes.
For rearing Chilonas blackburni,
Yes.
I think Spodoptera eggs are recommended.
Yeah.
So why you have tried
Uh,
Coursera cephalonica?
no,
Because it is very difficult to rear on Coursera cephalonica. What I have experienced.
Okay. Sir, especially in our NBIR, so
Culture is available.
no, that Coursera cephalonica is quite easier to maintain. Not only for this
Chilonas is not preferring.
Yeah, it is preferring. It prefers. It is preferring, but continuously, since we are rearing it on Coursera cephalonica, then when we suddenly we are checking it on the natural target was Spodoptera frugipada, so then it was not preferring.
Okay.
The preference, it got conditioned to the volatile cues of Coursera cephalonica under laboratory, because continuous exposure, generation by generation.
Okay, thank you. Next thing, what about Chilonas? You got Chilonas blackburni,
yes.
formosanus. Again, one species.
Yes.
It is available. I mean, it is present in
Yes,
nature.
yes.
Do you got that one?
As our colleague has got collected and he has given it for identification. So the identity is yet to be ascertained, sir.
Okay, okay. Because 3 species are there.
Okay.
Thank you.
Thank you, sir. Okay, problem. Thank you. Next speaker, be ready after the presentation to some of the sponsors. Next is Tamoga Saha. Just a minute. After that, one presentation is there. After that, please be seated only in front of them.
So, good afternoon. So I request Sushil sir to come over the dais to receive a small memento. So Sushil sir is from Godrej Agrovet. He is our proud sponsor. I request Sushil sir to come over the dais. I request the chairman of the session to join. Thank you, Dr. Sushil ji, coming all the way from Delhi and joining this conference. Thank you very much.
Next speaker, please.
Respected chair, co-chair, distinguished delegates, very good morning to one and all. I, Dr. Tamogna Shah, Senior Scientist, ICR-IRI, Regional Station, Pusa. At the outset, I'd like to thank the organizer for giving the opportunity to speak a topic on the diversity of the gut bacteria in the larval midgut of the Spodoptera frugipada from the eastern part of the India. This is a part of my DST funded project. So coming to the introduction, so as we know, the insect microbiome play a very crucial role in the shaping the interaction between the insect pest, host plant and the natural enemies, and also they play a key role in the adaptation of the insect to the environment and their growth and development. These gut microbial symbionts are known to influence the insect nutrition, also play a very crucial role in the defense and also the metabolism of the insects. Previous studies also identified that the microbial symbionts can break down the complex molecule into simple like insecticides, And therefore, the studying of the gut microbe is not only the important from the standpoint of understanding the mutualistic relationship, but also playing a crucial role in the foundation or future project aimed at the developing the microbial biocontrol agents. Relatively very few studies have been assessed in this Phototrope Fujiparidae associated the gut and the gut microbes. However, the very limited information is available on the microbial flora present in the gut of the Phototrope Fujiparidae. Therefore, in this study, we use the 16S RNA sequence profiling to characterize the diversity of the bacteria associated in the gut of the larva of the Phototrope Fujiparidae population from the eastern part of the India. So we have dissected the gut from the 5th and 6th install larva and they extract the genomic DNA from the gut with the help of the genomic DNA extraction kits, and we followed the standard protocol, and further we have sent the sequence, and after sequence we have submitted from the NCBI gene bank. And this is the 3 states we have collected the sample from Bihar, West Bengal and the Jharkhand. And this is the list of the collection site we have collected and the host plant, life stages and date of collection, and after getting the sequence we have submitted the NCBI gene bank. This is the accession number. So coming to the result, here we found out majorly 2 taxonomic category from the phyla to species level we have categorized in the gut of the Phototrope Fujiparidae. We found that 2 phylum, that is Firmicutes and Proteobacteria, mostly present, and under the class Bacilli and Bacteriaceae, and we found total 9 genus in our study, and total 32 species. Coming to the next slide, that is the genus level composition of the bacteria across the different sample. We found among the different genus, the Enterococcus, they are most abundant and dominant, present across the different sample, and which is followed by the Klebsiella and Enterobacter. And regarding the species level is concerned, we found that the most dominating species which are present in the gut, that is Enterococcus munditi. It is present most of the gut sample which I have collected, and it is followed by the Klebsiella pneumoniae. And further, we have tried to compare that whether our sample, that bacterial isolates, whether closely aligned with the previously submitted the sequences, and we found that our samples have been matched with the previously reported sample through this phylogenetic tree we tried to show. And further, we have morphologically characterized the dominant bacterial isolates of Phototrope Fujiparidae in the maize crop. In most of the, out of 6 bacterial isolates, we found the 4 bacteria, that is Enterococcus munditi, Enterobacter chloi, Enterococcus Enterococcus gallinarium, and Enterobacter ludwig, that shows the gram-positive reaction, and the cell size shape is the coxy, and the color is creamy to white color. And whereas Klebsiella pneumoniae and Enterococcus caeciliiflava, it shows the gram-negative reaction, and the shell shape is rod-shaped, and their color is the white to yellowish green. And further, we tried to understand what are the functions, whether this bacteria, whichever we found that is beneficial or it is pathogenic. So most of the bacteria we interestingly found that beneficial plays important role in the nitrogenous and carbohydrate metabolism, and nutritional balance and detoxification, also defense mechanism, except one bacteria which we found, that is Pantua, that is a pathogenic in nature. And further, we tried to compare our study with the previously reported study in the world, whether any new bacteria we have recorded from our study or not. So interestingly, we found 2 new bacteria from our study, that is Kluvera and Euconella. This is the first report from our study, and we have published. So coming to the summary, the 2 bacterial phyla mostly we have found the abundant, that is Proteobacter and Firmicutes. And among the genus level, we found the Enterococcus, that is present most across the all different samples, followed by the Klebsiella and larva, Klebsiella and Enterobacter. And as far as the species level is concerned, among the species we got the Enterococcus munditi is the most dominating species, followed by the Enterobacter species. And few literature when we studied, we found the Klebsiella Enterococcus exhibited the notable effectiveness in in combination with insecticide to mitigate the damage caused by the pest, and also previously report says that the Enterococcus reduce the mechanism of the insecticidal resistance. So when we tried to compare with our results which we have studied, and we found that most of the different insecticide when we studied the resistant reaction, you can see the resistance ratio, it shows between the 1 to 9-fold. It shows the very minimum level of resistance we have observed. So it whatever they reported that matched with our study, that it shows the low level resistance because of might be present of the bacteria. This. So coming to the conclusion, collectively we can say the finding illustrate the potential insect microbe inbounds as a strategic avenue for the biological pest management, and in the context of farm management, our findings shed the light on the comprehensive interlay between the insect exposure and the gut microbes of the Spodoptera frugiparda. And I will acknowledge my colleagues, and also I would like to acknowledge the ACRB DST funded project for providing this work, and we finally published the paper in that paper. Thank you very much.
Thank you. Congratulations for Tamogana Saha for his presentation. Please, any questions or clarifications from the delegates? Okay, please.
Thank you for a good presentation. Is it bacteria or what is it, beneficial to that insect, or giving any harmful effect?
Now, whatever I recorded, that mostly are beneficial in nature, that I have shown the function also in the table, that in defense mechanism and also digestion, and it helps. And except one bacteria which I have recorded, that is Pantua, that is only pathogenic in nature, others are all beneficial.
Thank you.
So whether your work with respect to the management of the pest is on beneficial bacteria or the harmful bacteria?
Yes. Beneficial only, sir, that is having the symbiotic, synchronous effect with the insecticide, and the beneficial is coming. It shows maybe the one of the.
What is your main objective? It is to identify the beneficial or the harmful to the insect?
No, I tried to identify the bacteria. In that study, I got that the beneficial and the pathogenic. So beneficial is further useful for that biological control program, sir.
Thank you very much. So next speaker is Sonali Diol. Sonali Diol. Not. Next is Manjunatha C.
Respected chair, co-chair, dignitaries of the dais, and all the delegates of the International Conference on UFA. It's my privilege to present the few findings on unveiling the insecticidal potential of indigenous Bacillus thuringiensis, Bacillus talworthy strain NBR BT25. So after the first report from the India, ICR NBR started working on the biocontrol. In that, we were very successful isolating one of the very important indigenous Bacillus thuringiensis strain. To date, whatever we in the India and globally have the maximum commercialized products from the Bacillus thuringiensis are from the Serover Kushtaki. We were able to identify, isolate and identify and characterize the strain which belongs to the Serover talworthy, and we, with the objective to identify efficient strain and to decipher the molecular mechanism behind the biocontrol potential of the strains, we have approached whole genome sequencing. So from the isolation, we have confirmed the Bacillus thuringiensis strain from simple microscopy. Further, we have done a scanning electron microscopy to identify the different spores which are present in the indigenously isolated novel strain. We would able to identify many crystal proteins which are in regular, sporadical and regular size. Once it is been confirmed as a talworthy, and it is been phylogenetically confirmed by using the multi-locus analysis. To date, whatever we have the data on Bacillus thuringiensis as an insecticidal only, and when the insecticidal property we look at it, it is been believed only as a gut microbe. And when it is toxic, when it is digestion is taken into the host gut. However, there are plenty of reports, and we also studied one of the many assays to prove the contact basis of the Bacillus thuringiensis strain. It has the strain which we isolated been evaluated for the many tests, including protease, lipase, lecithinase, and chitinase tests. These are the few tests which are confirming the entomopathogenic capacity of a strain, where the tests like chitinases, where we got the inhibition zones, showed that it can act by degrading the chitin which is present in the endoskeleton of an insect. Similarly, the protease test which confirmed it can act upon the protein degrading by the upon the insect, and lipase and acetynase, which are one of the important parts of an important ingredients of an insect system. So by degrading these things also, so Bacillus thuringiensis can degrade and create a mortality. Additionally, we explored the PGPR activity of an Bacillus thuringiensis, where the farmer willing to have a multi-role strain rather than having only one. In that, we have tested the IAA production test, ammonia reduction test, phosphate mobilization and siderophore test, where we could able to find a very significant results in the for the NBR BD25 strain for IAA production, ammonia and phosphate solvenation. Siderophore test is a very efficient, important character of any antagonistic organism, where the strain has proved its potential to act as an antagonistic organism. Apart from this insecticidal property, it has the capability to act as an antagonistic against the pathogen, and there are many reports to date, and we have found it in our indigenous strain also. Once the PGPR and antagonistic biochemical characterization done, we have done an in vitro bioassay with the international standard, and we found and other better isolates.
Sir, there is a disturbance for the speaker. Please maintain silence.
So we found our strain NBR BT25 is working extremely very nice, and with a LC50 value of 42.0 microgram per microliter was sufficient to kill the 50% of the populations at second insta. We next, after the once it has been tested, we have formulated the product into two different formulation. One is water soluble liquid and capsule based formulation. In this, we have further taken up the, in view of the registration of the Bacillus thuringiensis strain NBR BD25 in the under the CIBRC registration for the under 90 section, we have taken the multi-location trial under 3 agroclimatic zones, and we found very impressive. And we are fortunate to get an funding from the ICR PDP funding, corpus funding for the registration of product, which is under process this year. After knowing the potential of a strain under the in vitro bioassay and the field evaluation, we were into the genomic insights, deciphering the genomic insights by making the hybrid genome assembly, both by nanopore and Illumina sequencing. We have found that we have assembled the genome into as low as 6 scaffold, and this is the genome map of the strain. And we have done many downstream analysis and multiple bioinformatic analysis, which however I will be presenting few which are very relevant to the house. The average nucleotide identity studies of an NBR BT strain been identified, studied and kept as a record, so that the intellectual property right of an strain with the institute will be kept active, alive. Otherwise, the strain which will be taken by any other firms or the any partner, public people, that can be later be challenged. So once the average nucleotide identity been already recorded, and if a next strain be somebody taken, we can test, protect our intellectual strains. Additionally, we have found many phase test studies and antimicrobial this one to say that strain has been evolved with the evolved strain. And this is the another study where we another analysis where we could found the 5 CRISPR-Cas regions, which will again serve as an marker, then molecular marker to protect the intellectual property. Additionally, we have the biocontrol potential of the strain was very nice because of the 33 insecticidal genes which has been bioinformatically analyzed and molecularly validated. As I told the antagonistic property, we have also confirmed the antagonistic property by anti-smashing analysis. With this, I would like to say that we have isolated the one of the indigenous Bacillus thuringiensis stalworthy strain, which is not there in the commercial market, and we have deciphered the molecular mechanism with 33 insecticidal genes, and 2 genes are novel, now new to the science, and we are now into the cloning and expression of those novel genes, and we are with this I would like to acknowledge the director ICNMBR and division, Dr. R. Rangeshwan, whose work I have continued, and the ASRB collaborators who have evaluated our strain under the multi locations, and funding energy from the ICR and All India Network Project on fall armyworm, and our team from the insect bacteriology lab. Thank you.
Thank you for a good presentation and a good effort for the new strain with respect to the native BT. So now I request the audience to have clarification. Any 2 clarifications? Anything? So, okay, if there are no clarifications, thank you very much for Manjana.
Thank you, sir.
Thank you. On the,
Again, it is a great pleasure for us to welcome Dr. K.C. Ravi sir and Sharath K.N. and Dhananjaya Swami. All are from Bayer Crop Science. I wholeheartedly welcome to K.C. Ravi sir to come over the dais. Similarly, Dr. Sharath K.N. and also Dhananjaya Swami. Dr. K.C. Ravi sir is a head Asia field testing, Bayer Crop Science. He is having a vast experience of 37 years in corporate agriculture. And another, the delegate, Dhananjaya Swami, Dharnendra Swami from Bayer Crop Science, and Dr. Sharath K.N., an young entomologist. In fact, he is my PhD student. Now he joined Bayer Crop Science. He is a very good photographer. So just imagine, he took many photographs of BPH, eggs of BPH, with a very, very clear cut, with a high resolution. Give him a big round of applause. We are so proud of you. Thank you. I request our Dean Agri and also BM Prasanna sir to come over the dais and present the memento to all these dignitaries. Please, sir.
Dr. K.C. Ravi happens to be my classmate and Dr. Sharath happens to be my student. It's a proud privilege and proud moment to facilitate them.
One photo. Yeah, please, sir, please.
Good afternoon, everybody. Thanks for this honor. On behalf of Bayer, we would like to thank the organizing committee for inviting us for this symposium and presenting a memento to us. This is really special to us. So thanks a lot, and thanks a lot, Saranu, for inviting us here. Thank you so much. Okay.
So
another thing is, I had to make a presentation to the students this afternoon, but I'll not be able to do that because there is an event that got scheduled for this evening. I need to be there. But I've, kind of promised Sharanu that I will come again to this university and, address the students separately. So please excuse me for that, but I would look forward to meet you people once again. Thank you.
So please pardon me, I would take 30 seconds of yours. So I just want to put few words because I'm much privileged and I feel so honored to be here, and of course in this another 20 seconds I want to talk about my guru. So who has transferred his emotional connect with this fall armyworm to all the students like me. I'm in the audience also there are so many students of him who is sitting there, and he's been an inspirational element for all of us. And although I worked on pheromones and which was not his area of expertise, the kind of support he has given to us in all the way is something I cherish for my lifetime. And the most hardworking and a workaholic person I've ever seen is Dr. Sharanu. So thank you so much, sir, for all your support. Yes.
it is Annamalai Muthuswamy. Please.
It's okay, no problem. It's there, it's there. So, very good afternoon to all of you. So, I would like to thank the organizers, especially Dr. Sarana Bashappa sir, for giving me the opportunity. So, I am going to talk about the how we can exploit the endophytes, so within the plant system. Can you go to next slide, please? Next slide. Okay. Right. Yes. Okay. So, as all of us aware, any organism on the earth, it's not individual. So you can say I'm individual. No. Every organism have interaction with the other organism, either positive or negative way, for survival, growth and reproduction. Okay. So fortunately, we don't have any underground enemies, but plants do have underground enemies. Okay. So, but all organisms are intelligent, how to survive, how to grow, how to reproduce. But among the organisms, plants are most intelligent because they are sitting outside, sensing the environment time to time, and they are preparing themselves either physiologically or biochemically or genetically to cope up with any stress conditions. Okay? So, plant knows how to restrict the enemies with the help of their friends. Okay? And also, like how human beings are behave with their friends and enemies. Similarly, plants prepare themselves along with to cope up with the enemies as well as the friends. Okay? So, in order to invite the friends, plants will secrete some chemicals. In order to prevent the enemies, plants also secrete some different type of chemicals. Okay? So, that's what we are going to discuss. Okay. So, this endophytes is sitting inside simply. So, it's a 50-50. Sometimes they are friends. Whenever some situation is going on, it may turn into enemies. Okay? So, this endophytes is a microorganisms. It may be bacteria or fungus. Okay? So, this host plants giving is a shelter. Okay? So, this maybe provides beneficial effect in the host plant through the hormones, secondary metabolites, and also some of the endophytes mimicking the plant mechanism and secreting similar metabolites. Okay, and in that metabolites very effective in terms of biotic antibiotic tolerance. Okay, and also if you see the literature, the endophytic fungi play a very important role in biological control of the pest, almost all pests. So we can have the literature. Okay, so this fungi kills insects and it secretes insectal mycotoxin or infection penetrating the circular exoskeleton insect. Okay, so this strong insectal activity from the endophytic fungi interfere with the DNA repair and its replication. Okay, so this experimental we did one of the very important geographic indication GI tagged brinjal variety, that is the Mattugulla, which is cultivated almost last more than 500 years in Mattu village nearby Katpadi, Udupi district. Okay, so this is very unique brinjal, and so we are working almost last 15 years this variety with a different breeding aspect and all. So we would like to analyze what are the endophytes sitting in the Mattugulla variety and whether it is beneficial, whether they are giving harmful effect. Okay, so what is the objective? So we screen the endophytic fungi and determine and what are the major dominating fungi, endophytic fungi, then take the extract of those fungus and go for insectal activity against Spodoptera frigidera cells. So our work is completely in vitro, in the laboratory condition. Okay, so it's completely different from the speakers, though they have their very hard work in the field condition. So we are just mimicking what is the effective. And finally, what is the mechanism of cytotoxicity in the insect cells? So this is our overview of the methodology. So the 2 weeks old seedlings we cultured in the option MS media along with different salinity conditions. Okay, then we fresh and healthy roots of the Mattugulla we cultured, and roots cultured in the PDA media and with the chloramphenicol and all the external conditions. Then primary culture of the endophytes. Then we went for subculture and incubated for 2 to 3 days morphological culture. Then we also analyzed the colonization frequency, then density percentage and contribution by the dominant endophyte. Then identify the spores, observe under the microscope, identify the spores, identify the genus level. Then we went sequencing and identify the species level also. Then we collect the spores. Then we did the cytotoxicity against the ovarian cells of the Spodoptera frigidera. Okay. Okay. So first what we did, we just identify the IC50 of the different fungus. So we almost screened around 15 group of cultures. Out of 15 groups, we got around 18 which have very effective IC50 value against the insect cells. Okay, and also we analyze the mitochondrial membrane potential of the insect cells using the flow cytometry and compare the control. The mitochondrial membrane potential of the insect cell is decreasing dramatically. Okay, and also we analyze the mitochondrial structure of the Spodoptera frigidera cells after treating with the different concentration of the extract. For example, Cortiotrichum, we treated with the 50, 100 and 200 microgram of per ml of this extract, and you can see the the ring-like structure. For example, this is the control. Here you can see the round ring-like structure is the intact mitochondria. You can see the disrupted round-like structure. It indicates the
constructed with the total killing power of 0.4220. Then the third generation, here comes the egg parasitoid, where in the third generation there pop-ups the particular egg parasitoid Trichogramma chylonis telonimus remus. It was recorded in the third generation of Spodoptera eggs, and it proceeds with the late instar and early instar larva, where we can see the NPV infections in the late instar larva. So the budget table has been constructed with the total K value killing power of 0.6499. So these are the parasitoids I have observed and recorded. Parasitoid Chilonas black bunny, Campylortus chloridae, Coccidium milium, Trichogramma and Telonimus remus. So these are the fungal infection and bacterial infections. So the conclusion is the effective parasitoid in the study, during my study, is Campylortus chloridae, and all other parasitoids corresponding with the egg parasitoid specifically shown the parasitism rate in the third generation of the particular pest. So when we shift to all this biological control, we can have a sustainable environment. Thank you, sir.
Thank you, Ashwini. It's time for the clarification or the questions. Anybody?
Thank you, sir.
Okay, thank you for good presentation. Thank you very much. Speaker is Bhumika K.
Good afternoon, everyone. I am Bhumika from College of Agriculture, Shivamogga. So I am going to present my study entitled Interactions of Novel Insecticide with Entomopathogenic Nematode, Heterorabies indica and Folarimum Spodoptera fugipeda. So as we all know, Folarimum is a major pest in maize that can also colonize 353 different plant species. So the common management practices is use of insecticides. So other alternative management is the biocontrol agents, which includes entomopathogenic nematodes, which have the ability to infect, kill and reproduce inside Spodoptera fugipeda. The only limitation is that these EPNs are sensitive to environmental stress. So they can be incorporated into insecticide formulations or many other biopesticides, so that they can be combinedly applied in the field condition to achieve the better control of this pest. So with this background, we conducted the study of objective to know the compatibility of these Heterorabies indica with the different insecticides tested on Spodoptera fugipeda and also the interaction between them. So for this study, we maintained the two cultures. First is entomopathogenic nematode. So for this, mother culture was obtained from NBIR and it was mass multiplied on Corsera cephanica, and they were infective juveniles were extracted by setting the Y-trap method. So these are the infective juveniles we have extracted, and the other is the culture of Spodoptera fugipeda, which were maintained under laboratory condition to get the F1 generation. So the tested chemicals insecticides were chlorantraniliprole and spinetoram, because these insecticides are commonly used against Folarimum in the field conditions. So for conducting this study, we have used the we have prepared the stock solution of recommended dose under CIVRC recommendation. A stock solution was prepared for 1 liter, and from this 1 liter we have prepared 1/4 and 1/8 recommended dose concentrations by serial dilutions. So for conducting the study, for nematode concentrations were prepared in the range of 1000 infective juveniles per ml, and the third and fourth instar larvae were tested of same size and same weight were segregated before the bioassay. The bioassay we conducted were artificial diet overlay method. So for this conduction of bioassay, we have included 10 treatments. That treatment, first 4 treatments is we have used only the insecticides, that is chlorantraniliprole and spinetoram, their 1/4 and 1/8 concentration. And for the 5th treatment, we have used only the EPNs, that is Heterorabies indica. For the 3rd instar, we have used 30 infective juveniles, and for 4th instar, we have used 170 infective juveniles. And for the remaining combination of treatments, that is 6th, 7th and 8th, 9th, we have combined these 30 infective juveniles plus 1/4 and 1/8 concentration of both the insecticides. Thus, totally we have released 10 larvae per replication, and treatment include 4 replications. So the mortality data was recorded for 3 time intervals, 24, 48 and 72 hours. For the first 4 treatments, we have marked the larval mortality like they are failed to write back themselves. For the 5th, 6th, 7th, 8th and 9th treatments, we have found that the mortality, because the larva has changed to a brick red colored cadaver and without any odor emission. This confirms the mortality by EPN plus insecticides in the 3rd and 4th. And these mortality data were subjected to variant analysis, and the analysis for the interaction between these EPN and insecticide were classified into 3, and this 3 is based on the comparison of expected mortality and observed mortality percentage. They were calculated using the appropriate formula. So interaction is 3 types, that is additive when the chi-square value was recorded less than 3.84, and the interaction was antagonistic when the chi-square value is more than 3.84 and the observed mortality was less than expected mortality, and it was synergistic when the chi-square value is more than 3.84 and the observed mortality is more than expected mortality. So coming to the experimental results, in our study, in case of 3rd instar larva, we found that in the mortality recorded in the combinations, that is insecticide plus EPN, was higher compared to the when the insecticide or EPN applied alone. So highest mortality was found in the 1/4th recommended dose of spinatorium plus Heterorabies indica after 24 hours. So here the interaction we found was additive in all the combination. So while it goes on, after 48 treatment, we have found that these interaction was changed into synergistic in the case of spinatorium 1/8 recommended dose plus Heterorabies indica, and another combination 1/8 recommended dose of chlorantraniliprole along with infective juveniles. And also the more mortality was found in these combinations. Other 2 combinations, it was followed by these first 2 combinations, but the interaction was additive, it was not changed to synergistic. Also, here also we found that the increased mortality was in the combination compared to the application of insecticide and EPN alone. So after 72 hours, we found that the 3 combinations, that is spinatorium, both the concentration 1/4 and 1/8, and in chlorantraniliprole the concentration 1/8 plus Heterorabies indica. So they have changed into synergistic, but the other combination, that is chlorantraniliprole and Heterorabies indica, remained additive. So these additive and synergistic may be as the time exposure increases, the 2 different mode of action, that is insecticide mode of action along with EPN mode of action, has increased and has achieved more mortality than compared to the insecticide alone and the EPN alone. So these are the graphs showing the time period level, that is as the time period increases, the mortality also increases, it is significant. And also we have found that only synergistic and additive interaction was found in the 3rd instar without any record of antagonistic interaction. So in case of 4th larval instar, the same treatments, in that we have found that additive interaction was found and the increased mortality was found in this combination of the 2 concentration compared to the insecticide alone and EPN alone. So here also the highest mortality was found in the 1/8 recommended dose of chlorantraniliprole along with Heterorabies indica. And after 48 hours, here we have found that all the combinations have turned into synergistic interaction, and in also 72 hours we have found that these all the combinations give a synergistic interaction compared to the insecticide alone and EPN alone. So these are the gaps showing the 4th instar larval mortality. Here also as the time period increases, the mortality date increases, and here also there was no antagonistic interaction we have found. So coming to the conclusion, so from the study we have concluded that the increased mortality was found in the combination when they are applying together EPN with the insecticide compared to when they are applying individually, and also this finding confirms that only additive and synergistic interaction were found in 3rd and 4th larval instar without any antagonistic interaction, and these can be used as a least toxic and also an alternative biocontrol method to manage these fall armyworm pest, as this is a new study to India. So future studies are needed to under field condition to test this synergistic interaction. So I like to acknowledge my mentor and chairperson and advisory committee members, KSNUAHS Shivamogga, and ICR and NBI for providing the culture, and organizing committee INCOFA. Thank you.
Thank you, Bhumika. I have one question regarding the synergistic effect.
Okay.
What is the, for example, what is the, whether it is more due to your heterodyptities or due to your chemical?
No, sir. Actually, we have used both.
Correct. That is correct.
IGS we have used is 50% mortality, which is going to cause only 50% mortality. IGS we have used, and only the least concentration of the chemicals. So there is no more EPN has increased.
But I think only biocontrol agent have got 90% mortality. When it is in combination, it is less.
Yes, in combination.
What is contribution of biopesticide is more, or contribution of chemical is more in your combined treatment?
Sir, contribution of EPN is more.
Effective?
Yes, sir.
That's smart. When that is, if you work out the what is the economics and also other environmental concern, so your EPM is better than the chemical. Please, we have to look into that. Any other questions? Please.
Sir, may I have a moment? Sir, coming to economics, we are using less the recommended dose, so it will be economical.
Bhumika, what I feel is if you would have counted number of nematode emerged from cadavers,
yes,
so you could have interpreted what is the contribution of insecticide and what is the contribution of EPN. Have you counted nematodes from emerging from?
Yes, sir. Actually, the concentration we have concentrated 1000 insecticides per ml, and then only we have used. So. After.
Yeah.
No, sir.
From cadaver, you have not counted nematode emerging from the cadaver.
No, sir.
Okay.
Thank you.
So next speaker is Pratiksha B. Khedkar.
Shall I start, sir? Okay. So good morning to all dignitaries, co-chairman, chairman, scientist, research fellow, and all the students. Myself, Khedkar Pratiksha. I am delighted to share my, today I am present my work on the topic of assessment of indigenous parasitoids and the entomopathogens for the sustainable control of Spodoptera frugiperda in the baby corn ecosystem of the Maharashtra. So as we know, the fall armyworm Spodoptera frugiperda is a destructive pest and it is spread worldwide in many years. So there are the different strategies for the control of this different type of management strategy we have used. So that is the integrated pest management. There are the different types of management which are included in this integrated pest management. So in my research, I have to focus on the biological control, different biological control aspect, that is the predator, parasitoids and the pathogen which can be utilized for the effective control of this fall armyworm. So these indigenous parasitoids and entomopathogen which also provide the long-term suppressions. So my objective, that is to assess the parasitism potential of the indigenous parasitoids and the entomopathogens against the Spodoptera frugiperda in the baby corn ecosystem. So the material and methods, in this I have shown the baby corn, that is G5417 at the experimental field at our college. Under the unspread condition, there is no any fertilizer or the insecticide spray on that particular field. Then 48 quadrants were monitored weekly for the Spodoptera frugiperda larva and also collected the pupa. And in larva, we have also collected the early install larva and the late install larva. So collected larva were rare individually for the emergence and the mortality assessment. Also the parasitoid emergence and the entomopathogen induced mortality were assessed simultaneously to check the parasitism percentage. So the parasitism percentage were calculated by integrating the parasitoid emergence plus the entomopathogenous infection. So these are the results which are displayed on this table. That is parasitism percentage of parasitoids and entomopathogens of the Spodoptera frugiperda. So in these different orders of the parasitoids and the entomopathogens are given. So the most effective parasitoid which is come from the Braconidae family, which is about the parasitism percentage, that is 38.75%. After that, there is also another family, that is Ichneumonidae, which is about the 20%. Then Eurytomidae, which is about the 7%. All these belongs to the order Hymenoptera. Then next one is the Diptera, which is the Tachinidae, that is tachinid fly, which is about the 3.75%. Then entomopathogenic fungus, Beauveria bassiana, and the Metarhizium anisophila, both are contribute about the 8%. Then after that, the bacteria, which is the entomopathogenic bacteria which have used, that is contribute about the 3.12%. Then virus, entomopathogenic virus, that is Spodoptera frugiperda multinucleopolyhydrosis viruses, which contribute about the 8.75%. Then last one is the myrmidid nematode, which is the nematode species that contribute about the 1.26%. This is general distribution about the 100% population that I have covered. So this is the general scenario, that is the percent parasitism of the different agents like the parasitoids and the different types of the entomopathogens. From the all 100%, there is a total parasitism percentage which is observed about the 90.63%, and only the 9.37% adults were emergence from that collected larva. So these are the photographs that are captured during my research work. The first one is the Chilonas blackburni, and next one is the Chilonas formacinus. Both are the egg larval parasitoid, constitute about the Braconidae family. After that, the Cotacea rufricus, then Coccygidium melanum, Alioda species, and Micropiltis demoliator. These are also the larval parasitoids, belongs to the family Braconidae. Then Campolitis chloridae, it is also larval parasitoids, belongs to the family Ichneumonidae. Then Eurytoma species, it is also larval parasitoids, belongs to the Eurytomidae family. Then Exorista xanthopsis, it is belongs to the family Tachinidae, it is also known as the tachinid fly. These are the parasitoids. Then different types of entomopathogens that I found during my work. First one is the Metarhizium anisophila and Beauveria bassiana. Both are the entomopathogenic fungus. Then second is the nucleopolyhydrosis viruses, that is the virus, entomopathogenic virus, which are observed during the field. Then BT, which is known as the entomopathogenic bacteria, that is also found. Then entomopathogenic nematode, in this I have found the myrmidid nematode. The conclusion, the overall parasitoid percentage which is achieved the high natural suppression rate, that is about the 90.63% when we sow the baby corn in an unspread condition. Then from all the different families we have studied, the Braconidae was the most dominant parasitoid family with additional contribution from the other families like Ichneumonidae, Eurytomidae, and the different types of tachinid fly. So also there are the entomopathogen, we also contribute for the effective management of the fall armyworm populations, that is virus, bacteria, fungus, and entomopathogenic nematode. So finding demonstrated that there is a strong natural regulatory capacity in the environment which have the ability to control the fall armyworm population naturally, and also integration of these different types of biocontrol agent into the IPM program. The collaborating of both the things, we contribute the effective management of this particular pest, and also it maintain the ecological balance and for the biodiversity conservation, and also reduce the reliance on the different types of pesticide. So the biological control is act as an effective management strategy for the management of these fall armyworm Spodoptera frugiperda. Thank you so much.
Pratiksha, for any questions or clarifications, please.
Good afternoon, ma'am.
Good afternoon.
Ma'am, as your study is based on entomopathogens and parasitoids, my question is that how do entomopathogens compatible for parasitoids?
Okay. So the most of the larval populations of the fall armyworm, which generally controlled by the different types of parasitoid. But in favorable condition, when the favorable condition is available, like the high humidity and also the rainy season, there is also side by increase the entomopathogenic fungus, which side by control the other remaining populations of the larval. So this is the synergetic effect of these both entomopathogens plus the parasitoid. The both are controlled effectively that particular population. But in my observation, there is a more populations of the parasitoid than the entomopathogens.
Ma'am, my second question is that I'm also working on baby corn. Then what hurdles do you feel in the whole research?
Means there is means you have to rear properly that all the larval populations, because you have to detect the effective stage. If you can delay your observation, you miss that particular larval parasitoid stage, then you can't take photograph or can't detect the particular parasitoid. So there is a need for the daily feed change and daily observation that particular parasitoids or any larva for the emergence of the any biocontrol agents.
Thank you, ma'am.
Thank you.
Thank you.
Thank you so much, sir.
Okay. So the next speaker is Ratnakala. Ratnakala. Okay.
A very warm good afternoon to everyone. I am Ratnakala, PhD scholar from the Department of Entomology, US Harvard. I am here to discuss my research findings on the topic, the biocontrol efficacy of the entomopathogenic the indigenous entomopathogenic nematode and its symbiotic bacteria against this Proptera frugipada. As we all know that the Proptera frugipada has entered India during 2018. Ever since its inception, various management practices have been developed and deployed with the inclusion of the biocontrol agents. Among the biocontrol agents, the entomopathogenic nematodes have been proven to be the potent candidates. The entomopathogenic nematodes in the two genera, that is Stenonema and Heterorhabditis, are symbiotically associated with the Xenorhabdus and the Photorhabdus respectively. And this nematobacterial complex acts as a subtle assassin which leaves the insect dead within 24 hours. This could be used in the classical, augmentative and the conservation biological control programs. With this, we have formulated certain objectives, that is isolation and identification of the indigenous strains of the entomopathogenic nematode along with its symbiotic bacteria, and also the metabolomic profiling of the identified symbiotic bacteria. For the isolation of the indigenous strains of the entomopathogenic nematodes, we have collected the soil samples from 77 locations across the 3 agroclimatic zones of the northern Karnataka. Out of these 77 soil samples collected, the only one sample, that is the soil collected from the rhizosphere of the mahogany tree from the Kadwa Bidar of the Karnataka, was found to be positive for the presence of the EPN, which was later identified as the Stenonema abbasi USD BIDS. And the isolation of the entomopathogenic nematode was carried out following the standard protocol, that is the insect baiting technique using the Galeria melonella, which was given by the White during 1979. And the morphological and the molecular identification of the same EPN was done at the NBAR, the ICR NBAR. And the isolation of the symbiotic bacteria from the EPN was carried out by using the standard protocol, that is hemolymph drop method, which was given by the Park and his co-workers during 1999. And the metabolomic profiling of this identified bacteria was done using the HRLC-MS, which was equipped with the Agilent 6545 Q-TOF mass spectrometer at the SAFE facility IIT Bombay. The results included the female which is characterized, the female of the nematode, which was characterized by the presence of the vulva, which is the reproductive opening, and also the presence of the female tail. And the male characters such as the male testis reflection, the spicule and the gubernaculum, their shape, size also matters when we consider for the morphological identification. And the infective joiners are distinguished or characterized based on the presence of the hyaline tail and also the pouch or the receptacle which holds or houses the symbiotic bacteria. And this is the list or the table of the morphometric measures of the particular strain, that is Stenenema abbasi USDB IDS, which includes the certain characters like all the characters such as the length, diameter, and so on. And the molecular identification was done using the ITS primer, and the sequence was submitted to the NCBI, and the phylogenetic tree was constructed using this Xenorhabdus the elegans as an outgroup. And here we could see that our strain has formed the close clustering with the other known strains of the Stenenema abbasi. And the bioassay was conducted against the third instar of this Proptera frugipada, but at varied concentration ranging from the 50 to 500. And here we could see that the 500 IGs per larva, it has induced complete mortality against the third instar on the first day itself. And on the fifth day, all the concentration could cause the complete mortality against this third instar of this Proptera frugipada. And this is the plate showing the cadavers which are manifesting the symptoms of the USD BIDS infection. And the invasion efficiency is nothing but the rate at which the nematodes enter, the IGs enter inside the host, that was significantly higher at the higher concentration, that is 500 IGs per larva. And similarly, the reproductive rate is the rate at which the nematodes or the IGs will multiply within the host cadaver, which was also significantly higher at the higher concentration. And this is the plate showing the emergence of the IJs after 3 and 5th days of the dissection. And the morphological identification of the bacteria was done based on the presence of the maroon colonies on the blue media, that is a primary phenotype, and after 24 hours which converts into the secondary phenotype with the blue color colonies on the media, and it is a gram-negative bacteria. The molecular identification was done using the 16S rRNA, and the sequence was submitted to the NCBI, and the phylogenetic tree was constructed by taking the Proteus mirabilis as an outgroup. Here also we could see that our strain of the Xenorhabdus indica, which was identified, has formed the close clustering with the other known strains of the Xenorhabdus indica. And the bioassay was conducted against the third instar of this Proptera frugipada by taking the two formulation, that is self-resuppinated and the cell suspension, and at the varied concentrations of 10^4 to 10^10, which was done using the leaf-D bioassay method. Here we can clearly see that at the higher concentration of 10^8, there was a complete mortality or the 100% mortality in case of the 5th day, that is in the self-resuppinated, and approximately 90% of the mortality can be seen in case of the cell suspension. On the contrary, at the lower concentration of 10^4, 5 and 6, which apparently did not yield the considerable mortality. And this is the plate showing the Proptera frugipada cadaver, and also cadaver manifesting the malformations or the deformation when infected with the Xenorhabdus indica. And as the lower concentration did not yield the considerable yield, the mortality, we have further analyzed its effect on the larval development and also the feeding behavior. To our surprise, the larva had the least or the minimum feeding or no feeding on the leaves treated with the higher concentrations of the Xenorhabdus indica USD BIDS, as we can see in the graph, and which was very evident with the reduced larval weight at the higher concentration, which was again significant. And the similar effects we could also see in case of the pupa, as the pupa the larvae which were fed on the treated leaves of the concentration of 10^6 CFU per ml had the pupal weight of 0.11 and 0.08 in case of the CFS and the CS, as against the control, that is 0.33 and 0.32 respectively. And 80% of the udders that have emerged from the pupa had the deformed or the malformed udders. The HRLC-MS analysis had showed that we could identify 26 bioactive metabolites. Out of that, 7 had the insecticidal activity, and these were the novel like the chrysin, ogonin, anchorin, the tacrin, and the embelin. With this, I would like to conclude that this is the first report of the indigenous strain of the Stenenema abbasi and its symbiotic bacteria from the North Karnataka soil, and the discovery of the novel compounds such as the chrysin, embelin, or the galangine, which could unveil the new promising candidates for the new generational insecticidal compounds. Thank you. I would like to acknowledge the DST for providing the INSPIRE Fellowship and USD and also the NBIR. Thank you.
Congratulations, Ratnakala, for your effort as well as good presentation, and also your contribution by identifying a new approach for reporting the new things. Thank you very much.
Thank you, sir.
Please, any questions?
Did you identify, did you validated those metabolites which are identified?
Yeah, actually I have validated using the molecular docking because there was a time constraint I couldn't.
Okay, you have taken only molecular docking or other methods?
No, only molecular docking. If you know any other methods, you can suggest me. I'll try the method.
qPCR, different methods are there, no? I thought
The qPCR, yeah, that can be done. Yeah.
Okay, if there are.
Nice presentation, actually, ma'am. Actually, I have one simple question. As we, as the earlier presentation, the myrmidid, it is also nematode. Myrmidid,
yes.
it is also nematode, no, worm. It is, its infestation is very high in the fall armyworm and all insects. But as per your presentation, you collected several samples, 77 I think, and you get one. So what's the reason that EPN is very virulent? So what's the reason we get a very that least nematode EPN, especially EPN from the soil? What's the reason behind it?
Actually, when I was collecting the soil samples, here I have collected 77. Out of 77, I think I have got 16 nematodes which are not EPN, which are saprophytic, which can be, which feed on the cadavers, which are dead already. That could be because of any matter, okay? In that case, what happens, there will be competition between the EPN or maybe the saprophytic, which are saprophitics, which are very aggressive, could sideways this EPN. There could be chances, and the location where you are collecting, that also matters. If you are collecting to this, from the southern parts of the India, you may get the good numbers of the EPN.
Okay, thank you, Ratnakala, once again. Now the last speaker of this session is Shiv Prasad.
So good afternoon, one and all present here. So today I am Shiv Prasad, PhD scholar from UAS GKVK Bangalore. So I'll be presenting part of my work. So that is collection, isolation and morphomolecular characterization of entomopathogenic fungus infesting Phalaromium Spodoptera frugipada. So we all know that agriculture is one of the dependent sector in India. More than 50% of the population depend on it. And with increasing population, we also have to increase the food production. But with this comes the strains of biotic and abiotic factors. So majorly biotic factors of insect pests and diseases. So when this comes, the first and the foremost thing the farmer thinks of is the use of the chemical pesticides. So we all know that there is very undue use or very unmaintained use of these insecticides, due to which there are problems of resurgence, resistance and secondary pest outbreak. So now presently there are more than 3,137 active molecules which have seen resistance in various pests, and with all these insecticides there are also grave danger of the environmental pollution and also on the effect of human beings. So with all these background, so we have to use some sustainable ways of managing this insect. So one such is the use of entomopathogenic fungus for the management of these pests. So we all know that insects and fungus, they have co-evolved around 400 million years ago. So when they have co-evolved, there are certain interactions that have developed between these two. So one such interaction is the development of pathogenicity in several hypocereal fungi, especially Ascomycota and Basidiomycota. So the first report was by Agostino Bassi from 1773 to 1856, where he reported Beauveria bassiana, okay, or white muscadine fungus. So later, so when we talk about other biopesticides like bacteria and nematodes, so the mode of action depends mainly on the feeding of the insect. So when we talk about fungus, the mere contact of a spore is very much enough to start the infection process. So out of the 31 insect orders known, so more than 20 are known to be infected by the fungus, and also the all development stages starting from egg to adult are known to be susceptible for this fungus. So And also about fall armyworm, this pest needs no introduction. So it was first reported in 2018 by Sharanabasappa et al. From Shivamogga and Davangere districts. So we all know that it can reduce up to 57% yield, so if left unchecked, and according to APRD database, it has developed resistance to more than 47 active ingredients. So talking about the biopesticides or the fungus infecting fall armyworm, so majorly it is Metarhizium relay followed by Beauveria bassiana and Metarhizium anisoplea. So with this, I would like to focus on three objectives, that is collection and isolation, morphomolecular characterization, and also evaluation of various solid substrates for mass multiplication. So for material methods, I have collected the infected fall armyworm larvae from various districts of Karnataka. So the dead cadaver were brought to the laboratory, and they were kept for sporulation with the white with the germination paper for creating moisture. And once the larvae has sporulated, it was again isolated on the suborhodextrose agar supplemented with the yeast extract. And later the cultural and morphological characters were studied. So and later the DNA extraction was done using modified C-tab method, and the PCR amplification using ITS 5 and 4 primers, and they were submitted to GenBank, and action numbers were obtained. And later so the after studying the cultural characters, we also evaluated the substrates for mass multiplication. So these are all the isolates, 11 isolates belonging to 3 genera, that is Metarhizium, Beauveria and Cordyceps. I think this is the first report of the Cordyceps infecting fall armyworm from Karnataka, I am not sure yet. So these are the 11 isolates collected from various districts of Karnataka. And coming to the results, so various morphological characters such as the mycelial growth, colony color, texture, surface and topography, margin, pigmentation and conidial shape and length was considered for the morphological characterization, and the keys developed by Hubner 2012 and 5 were used for the identification of these fungi. So these are the morphological characters of all the 11 isolates. So this is the pairwise distance dendrogram that is constructed based on the morphological characters of these fungi using R software. So here you can see that the Beauveria and other isolates have grouped based on their morphological characters into different clusters. So later, so here you can see these are the macroscopic and microscopic characters of the 11 isolates of Metarhizium and other isolates. So this grown on PDA and SDAY. Okay, so these are the isolates.
Yeah, just a second. Let me check my presentation. I try to upload the presentation, but I don't see where it's located. I think that maybe it's this one. Yeah, I think that. Yeah. Yeah, can you see the presentation?
Yes, sir.
Yeah, okay.
Okay, let us start.
Good afternoon to distinguished delegates, esteemed speakers, rapporteurs, and participants of this conference. On behalf of the organizing committee, I extend a very warm welcome to all of you to Technical Session 4. We are now ready to begin the Technical Session 4, focused on the theme Insecticide Resistance in Fall Armyworm. In this session, we have 2 keynote address and 5 oral presentations. Parallelly, poster presentation will be ongoing beside the corridor of the MP Hall. To guide this session, we are privileged to have with us Dr. B.M. Dishant Kumar sir as the session chair, who has got prestigious awards like Nagama Dattatreya Rao Desai Award in the year 2018 by USB, Best Researcher Award in the year 2016 by KSNUAHS Shivamogga. He has around 110 publications, out of which in 46 papers, he has worked as a lead author and published in reputed journals. He has totally released 12 rice varieties, and out of which some are bold varieties, some are fine rice red varieties. Also, 1 cardamom variety. He has also 5 varieties registered with the PPA and FRA in his portfolio. He has registered and documented 98 rice land races of KSNUAHS Shivamogga at NBPGR New Delhi, and several externally funded projects. It's our pride, sir, to have with us, and he is presently serving as Director of Research in our university. I also welcome our co-chairman of the session, Dr. Srinivas Parami, Global Director, Field Development and Technical Partnerships, Provavi Inc. USA, who has a strong research background in the field of entomology, crop protection, and stakeholder relation, working in cross-functional, multidisciplinary, and cultural diverse environments, and has got several prestigious awards in his field. And I would also like to welcome Dr. Narendra Kulkarni, Principal Scientist, ICR IGFRI, Southern Regional Research Station, Dharwad, Karnataka. For this session, rapporteurs for this session will be Dr. Shivaputra Bommanalli, Assistant Professor Agroforestry, KSNUAHS Shivamogga, Dr. Gajendra Kirdapure, Assistant Professor Seed Technology, KSNUAHS Shivamogga. Now, I welcome you all for the keynote address. on global resistance of the fall armyworm to insecticide, a study of island-type conditions by David Mota Sunsets, Professor of Entomology, Michigan State University, Lansing, USA, and co-director of the Arthropod Pesticide Resistance Database, and having 30 years of rich experience on pest management and insecticide resistance, and guided more than 10 students and having many projects. Now we shall move to keynote address by Professor David, sir.
Sir, you can start.
Yes, yes. Hello.
Hello, sir.
Sir, you are.
David, sir.
Professor, you can start the presentation.
Yeah. Okay. Can you hear me?
Yes, sir.
Yes, sir. You are.
Okay. Okay. Thank you. Thank you so much. Thank you to the organizer of this symposium. I want to talk about the resistance of fall armyworm to insecticide, story island time conditions. This is a kind invitation for the organizer, Dr. Sharapavasta. Thank you so much. So, in the Americas, fall armyworm is a pest that has been for thousands of years. And I understand that in other parts of the continents is completely different situation. But in the Americans, North America and South America has been for a long time. PhD students and people from other people working in fall armyworm. So I don't pretend to have a comprehensive or cover all the aspects of resistance of fall armyworm, but I'm going to talk about the specific conditions in Puerto Rico. Sorry, Puerto Rico. You can see here. This is one picture of the crop devastated by larvae of fall armyworm, even after intense use of pesticides. In this case, it is the core treated with pyrethroids, and this treatment resulted the same than the control. So fall armyworm has distributed from the Americas to Africa, India, China, Australia, and different places in Europe. So it's a big problem. One of the problems we face, not only fall armyworm, is the ability to develop pesticide resistance. In this graph, you can see is in general the number of species that has developed a resistance to pesticides. There are 637 species with about 20,000 cases to 367 compounds. So this situation happens since 1940 to now. So in our database, you can check www.pesticideresistant.org, the cases of resistance all over the world and different compounds. So the situation is one of the points that is really important. Let me see, switch to the, okay. Let me see, so it's better now? I think that there is some issue with the presentation, but can you see chat program? Mm-hmm. Yeah, okay. So I don't know if you can see this. This is the view presentation. So there are many cases of resistance, and this is one of them. So there is lab-selected resistance. There is fieldable resistance, and we define resistance in the field when the pest developed more than tenfold resistance. And then there is another situation that is practical resistance, when there is a pesticide field failure. So this is a concept coined by Dr. Bruce Tabashny from University of Arizona. The fall armyworm now is the species that is in the top 20 resistant species in the world. And this happened because the explosion invading other niche. So now you can see in this slide that the pest is ranked number 18 in resistant development in the world. And then one of the things that happened with this pest is it has developed resistant there is reports of 272 resistant cases, 47 active ingredients to 8 motor actions. In the next slide, you can see the progression of resistance in 1965 to lindane DDT at the time the compounds used. And then through the time, you see the progression of resistance cyclodines in 1975 on organochlorinase. Then in the 90s, resistant to pyrethroids and OPs. And then the resistance continue to all over the decades and every 5 years to different type of compounds. And now recently more resistance to BT, OPs, insect growth regulators, spinosyn, carbamase, damase, and other new compounds. So in the case of Puerto Rico is an island, and then here the core is produced not for core production, but for seed augmentation and resource. So this place is very important for nurseries, one of the most strategic places for resource and augmentation in the world. But then the pest, this pest in Puerto Rico has certain conditions, like it's a tropical island with favorable weather for crop and pest development. There is 3-4 seasons of crop production. So the fall armyworm population are congregated there. We know that fall armyworm is a potent flyer, but then if they are in island time conditions, even there is a potent flyer, many of the populations remain in the same locations. One of the things that happens is also the number of generations per year. More than 10 generations per year made is present the pest all the time. And in this place was the first case of resistance to BT crops, including Cry1F. In 2003 was introduced in Puerto Rico, and resistance was developed in 2006. So this is the place that I'm doing research on fall armyworm. Because on the sake of the time, I'm not going to describe in detail the resistance, but here is the after 10 years of finding resistance in Puerto Rico, my PhD student Rebecca Gutierrez find that the fall armyworm is still the gene was fixed in the population. You can see the log dose response for a susceptible colony here, and then resistant to Cry1F at 10,000 nanograms per square centimeter. The larvae didn't die in the fall armyworm. And then resistance was found to different compounds, dimais, OPs, insect growth regulators, so many different compounds. My PhD student Omar Pozos worked more in other type of chemical groups, synthetic pesticides, and then recently Shiva Shatrughn from India found resistance. He's going to talk about resistance in emamectin benzoate and found resistance. So this is one of the places, perhaps one of the places that is the most resistant species to pesticide in the world. And I put this example because I believe that this is the situation that is going to happen 3 to 5 years in India and perhaps other places with resistance to pesticides. In the case of diamide compounds, these compounds target the rhinodine receptors, and here is one of the examples of the way that we do the assays. We use costar cells, apply 1 milliliter of dye, and then we place the insecticide over the dye and then put the larvae over them. And here are some results of resistance of the amide compounds, more than 94 in Puerto Rico populations. This is the susceptible, and this is the resistant population. And also high levels of resistance to fluindamide, more than 2,700 fold resistance.
I wanted to provide you with a map for reference of where these two populations of fall armyworm were collected, one from Texas and the other one in Louisiana. And these populations are at a distance of about 272 miles or 438 kilometers. So it's a significant distance, but still relatively close compared to, for example, to the native and the invasive range of fall armyworm. Our efforts to identify mechanisms of resistance to BIP3A in fall armyworm have focused on comparing susceptible and resistant strains and looking at the different steps in the mode of action of the toxin. So similarly to cry proteins that are also expressed in transgenic maize, there is the ingestion of the material, processing of the insecticidal protein to an toxin active form that binds to receptors in the midgut cell surface. This binding is conducive to pore formation, from which the
Discussion or questions. Discussion or questions. Okay. So now let us begin our first presentation will be coming. Okay. I request our Dr. Dushant Kumar sir, chairman of this session, to take the session forward.
Good afternoon, and welcome to the important session, that is insecticide resistant in fall armyworm. So we have actually completed two important sessions, three important sessions. The one is on debate, so global resistance of fall armyworm. Another one is a fantastic session made by Louis Jorot, and also now we want to plan, this is oral presentation starting with Yashashwini. Okay. Yeah. Satujna Shiva. Yes. Okay. So the timings and allotted timings, and the topic is field evolved resistance of fall armyworm. So I request Satujna Shiva to present this field evolved resistant of fall armyworm to mimicans benzoate.
Hello, can you hear us, Shatrugna sir? Are we audible? Shiva sir?
You can start.
Yes sir, you can start your presentation.
Everyone, can you hear me, please?
It's audible, sir.
So, good afternoon again. My name is Siva. I'm going to present my talk on the title Field Evolved Resistant of Falarium to Memectin benzoate. Under the theme 4, insecticide resistant in falarium. So, before begin the talk, I would like to polite request, please don't take picture and don't share on any.
A warm good afternoon to respected chairpersons, rapporteurs and fellow delegates. My name is Amoga. I'm pursuing PhD in entomology in USS GKVK, and I'm here to present my part of my research work in the title Gut Microbiota as a Modulators of Insecticide Susceptibility in Spodoptera frugipoda. As we all know, the fall armyworm is a major invasive pest which recently entered India during 2018. In order to tackle this pest, many insecticides have been recommended, and due to the indiscriminate usage, this insecticide has exerted selection pressure which has led to the development of resistance. Moving on to the insect and associated gut microbes, the gut microbes play a vital role in the survival and development of the host insect. They play a crucial role in biology and behavior of the host insect. They also involved in the digestion of recalcitrant food components and helps in modulating the immune responses, and also they provide protection against pathogens and toxins. Recently, the studies have found that these gut microbes, they are also involved in the detoxification of xenobiotics like insecticide, where they use insecticide as their source of carbon. So it has been suggested that the development of resistance might be influenced by the gut microbes, which add another degree of complexity to the process of resistance. So keeping this in view, the research was conducted to study the influence of gut microbes on the susceptibility of Spodoptera frugipoda larva to insecticide, and also to isolate the gut bacteria of Spodoptera frugipoda larva. The fall armyworm egg masses and larvae, they were collected from the maize field of USS GKVK, and they were brought to the lab and reared in a laboratory condition in a plastic container, and they were fed with the maize leaves. Then the subsequent F1 generation, they were reared on the artificial diet as per the protocol given by Ballal and his co-worker in 1995. And here rearing, it comprises of two groups. The first group, that is larvae fed on the diet without adding any antibiotics, mainly to maintain the habitual gut bacteria. And second group, that is larvae fed on the artificial diet with antibiotics to have a free gut bacteria. So in this experiment, it has two groups, T1 and T2, and these two groups were maintained separately in order to avoid the mixing of population. And antibiotic used in this experiment was the streptomycin sulfate and rifampicin. Prior conducting bioassay, the antibiotic dosage was determined, and the standardized dosage was 800 microgram per ml. So the cocktail of streptomycin sulfate and rifampicin was used, and this was mixed into the freshly prepared artificial diet, and then it was fed to the frugipoda larva from first to third instar continuously. And in order to check the presence of gut bacteria, the 5 larvae were randomly selected, and the gut was dissected, and homogenate was flattened on the nutrient agar media to check the presence of gut bacteria. And at this dosage, that is 800 microgram per ml, there were gut bacteria was culturable, mainly culturable gut bacteria was not present in the nutrient agar media. So, and this dosage was finally used and further procedure for conducting the bioassay. For conducting the bioassay, mainly 3 insecticides was selected, that is emamectin benzate 5% Hg, profenofos 50% EC, and lambda-cyhalothrin 5% EC. The bioassay was conducted for F1 generation field population and also laboratory rate population, that is F13 generations, and the bioassay was conducted for third instar larva using diet overlay method, and the LC50 values were determined using 72 hub mortality data by probit analysis using SPSS software. And in order to isolate the gut bacteria of Spodoptera frugipoda larva, here the larva from the control was selected, that is 15th star larva was selected, and it was pre-starred, and then it was surface sterilized using 70% of ethanol followed by 0.1% of sodium hypochlorite, and then it was washed with double distilled water, and then the gut was dissected in a aseptic condition in a laminar airflow chamber, and gut was added in a 1 ml of pH solution, and serial dilution was made up to 10 to the power minus 7, and from these each dilution 100 microliter of aliquot was taken and plated on the nutrient agar media and incubated for 48 hours for the formation of bacterial colonies. Once the bacterial colonies were formed, the pure culture was maintained, and from this it was subjected to bacterial DNA isolation, and finally the identification was done. So moving on to the results. So this table depicts the influence of gut bacteria on the susceptibility of Spodoptera frugipoda larva to field population, that is F1 generation population, to different insecticide. And among the tested insecticide, the emamectin benzate was found recorded the lowest LC50 value, followed by lambda cyhalothrin, and then the profenofos 50% EC. And among the treatment, that is with antibiotic and without antibiotic treated larval population, in amamectin benzoate, without antibiotic treated larval population, it recorded the LC50 of 0.29 ppm, where with antibiotics treated larval population, the LC50 was recorded slightly lower, that is 1.18 ppm. Similarly, profenofos without antibiotic treated population, the LC50 value was 1311.44 ppm, where with antibiotics, the LC50 value was 501.68 ppm. And lambda cyhalothrin recorded the LC50 value of in the without antibiotics, that is 433.10 ppm, and with antibiotics, the LC50 value was 373.49 ppm. Overall, this indicates that in the presence of antibiotics, the LC50 values were recorded with lesser values. It has shown the antibiotic larval population has shown the increased susceptibility to tested insecticide. And the LC50 ratio was also calculated to evaluate the influence of gut bacteria, and highest LC50 ratio was found in the profenofos 50% EC, that is 2.274, followed by amamectin benzoate. Similarly, the influence of gut bacteria on susceptibility of Frugipada larva to laboratory rat population, that is F13 generation, was tested two different insecticides. Here amamectin benzoate without antibiotics treated larval population recorded LC50 of 0.16 ppm, and with antibiotics it has recorded a lower LC50 value, that is 0.095 ppm. And profenofos has recorded the LC50 of 883.80 ppm in without antibiotics treated larval population, and with antibiotics it was 312.12 ppm. And lambda cyhalothrin without antibiotics, the LC50 was 224.66 ppm, and with antibiotics the LC50 value was 173.40 ppm. Similarly, here also in the presence of antibiotics, the larval there was an increased susceptibility has been observed. So this graph depicts the comparison between field population and laboratory rat population, where field population recorded the highest resistance compared to the lab population, and without antibiotics has recorded highest LC50 value compared to the with antibiotics. It indicates that in the presence of antibiotics, the culturable gut bacteria were eliminated, which has showed the increased susceptibility, which indicates that gut bacteria do have some role in the development of resistance. So this is the table depicting the gut bacteria that have been isolated from 15 star larva, that is Enterococcus gallinarum, Enterococcus munti, Bacillus cereus, Serratia maracens, and Bacillus subtilis have been isolated. And this is the morphological characters of the bacterial isolates that have been isolated from the 15 star larva. Finally, that in the presence of antibiotics treated larval population, there was increased susceptibility has been observed to the tested insecticide, which indicates that gut bacteria they have resistance. So these act as a modulators for insecticide susceptibility, and these findings they provide novel insights into improving the efficacy of insecticide for the management of this pest, particularly in the population where resistance have been reported. Finally, I would like to acknowledge the chairperson, Dr. Shivanabi, and the advisory committee. Thank you.
By showing this slide, so it has been told earlier that you are not supposed to show the slide of your chairman or members. So anyway, I will ask a question. So would you like to say that antibiotic treated larvae, they are more susceptible to insecticides? So how you are going to spray or induce antibiotics in insects? Okay. How this will be translated into the field conditions?
For future, for developing the new insecticide, might be incorporation of antibiotics in combination.
Do you know the how create havoc when they enter the biological ecosystem? If antibiotic treated eggs, if I consume, it's not safe. if they're entered into the ecological system, so it is creating an havoc. How you say that antibiotics should, you are in search of reducing susceptibility of phala, but you are inducing antibiotics into the ecosystem. Humans may consume those antibiotics, cattle may consume, it may enter the water, it may enter any biological system.
Any review on this? Any review on this? What the question is made by your co-chair regarding antibiotic activities?
Now you please find out these answers. You find out these answers. Okay. Thank you.
Any questions from audience? Yes.
Actually, I should appreciate because for experimental purpose, you have, you can say, given an antibiotic as a feed to reduce the microbial, you can say, population. But my question was, without antibiotic, what was the count of microbiota? Have you concentrated on that aspect? No, sir. Microbial count I have not taken. Yeah, if possible, you include that also. Natural microbiota in natural population. Thank
you. So, if there is no question, thank you, Shivamogga. Well presented. So next move on to another speaker, Chinmay, College of Agriculture, Shivamogga. And topic is variability in susceptibility of fall armyworm population to novel insecticides. Okay.
A very good afternoon, everyone.
You can start.
I'm Chinmay. Welcome. I'm Chinmay, and I'm presenting on variability in susceptibility of fall armyworm, Spodoptera frugipada populations to novel insecticides. Good afternoon, everyone. I am Chinmay. I welcome you all. I am presenting on variability in susceptibility of fall armyworm, Spodoptera frugipada population. As we all know, this fall armyworm became one of the major invasive pests in India after its first detection in 2018. As most of the farmers rely on insecticides as a frontline approach to control this fall armyworm. Because of its high fecundity, rapid spread, and polyphagy, make it difficult to manage, as it is having overlapping generations which lead to quick reinfestation. Repeated applications exert strong selection pressure, accelerating resistance development. Therefore, a regular resistant monitoring is essential to detect early shifts and guide selection of effective insecticides is very important. By keeping all these in mind, the following objectives were taken, that is to evaluate the current susceptibility of fall armyworm populations to novel insecticides and to determine the resistant ratios and identify insecticides showing reduced performance or emerging resistance, and to compare the effectiveness of novel insecticides with commonly used chemicals to support the resistant management strategies. Methodology include like this: a laboratory selected strain which is maintained in the laboratory, that is since 2019, that is for about 80 generations without selection pressure to the insecticides, and for field the population from hotspot locations from Karnataka and Telangana to perform the bioassay studies. And here, after collecting the egg masses from different locations, these egg masses were after emergence, they were transferred to the artificial diet. Up to till pupation, they were cultured on the artificial diet. Later, in the F1 generation, they are subjected to bioassay. Here we have taken about 9 different insecticides. In that 6 are a novel insecticides which is having a different mode of action, and another 3 are very commonly used by farmers in the field condition. The 6 insecticides include tetraniliprole, broflanilide, isocycloserum, chlorphenapyr, cyclaniliprole, fluxametamide, and 3 commonly used insecticides like chlorantraniliprole, spinetoram, and emamectin benzoate. Here, bioassay was conducted with the 1-day-old third instar larva, which is reared on artificial diet, and later the freshly prepared diet were poured into multi-cavity trays, Petri plates, and later the diet overlay bioassay method was followed, and these insecticides were for dose bracketing. First, different concentrations were prepared and to get the 10 to 90% mortality. After that, after concentrations are fixed, about 7 to 8 concentrations were maintained, prepared for each insecticide, and as a spreader, the Triton X100 is used, and in each concentration 36 larvae were used with 3 replications, that is each 12 larvae per replication, and the mortality was recorded up to 120 hours, and after mortality recorded, the values were subjected to LC50, the values are subjected to probit analysis to estimate the LC50 values. The results include here, when compared to different locations and also a susceptible population, most of the susceptible population found to be susceptible with very low LC50 values. With respect to different populations like Karnataka and Telangana, the LC50 values in the most of the cases, like in case of tetraniliprole, isocycloserum, chlorantraniliprole, and spinetoram, they found in Telangana region the LC50 values were found to be high when compared to that of Karnataka region, and except the fluxametamide, it is the slight LC50 values are slight higher when compared to that of Telangana population. Apart from that, most of the chemicals found to be similar with respect to both the locations. Here also, this is the graphs showing that the LC50 values are higher with respect to chlorphenapyr, chlorantraniliprole, and which is followed by isocycloserum and tetraniliprole, and other insecticides found to be less, LC50 values found to be less. And later, with the LC50 values, the resistance ratios were calculated. That is, resistance ratio is equal to LC50 of field population divided by LC50 of susceptible population. Here, LC50 ratios with respect to both the population, when the resistance ratios found to be very least, and they are less resistant when as there is no insecticide found to be high resistant and highly resistant, and with case of like tetraniliprole, isocycloserum, chlorantraniliprole, and spinetoram, the resistant ratios were slightly higher when compared to that of Karnataka population resistant ratios, except in fluxametamide, which is slight higher when compared to that of Telangana. And most of the we can say that most of the insecticides found to be very effective with respect to on fall armyworm, but when compared with the commonly used insecticides like chlorantraniliprole, spinetoram, emamectin benzoate, they also found to be very effective. So we have to integrate these molecules by continuous, usually farmers are continuously using emamectin benzoate alone as a management strategy with respect to this fall armyworm, but integrating these insecticides on multiple sprays can be beneficial. We can avoid a resistance quick development of resistance, and selection pressure can be reduced. From this, we can expand we can go for expand resistant monitoring across different hotspots, and we can identify further molecular or biochemical mechanisms for early detected resistance, and we can study inheritance and fitness cost to understand the stability of resistance, and we can also develop a IRAC based rotation modules for sustainable farm management. In conclusion, fall armyworm population showed clear variability in susceptibility to these novel different mode of action molecules, and certain chemistries remain effective while others exhibited reduced sensitivity in specific regions, and continuous monitoring and location specific insecticide rotation is essential to slow down the resistance development, and integrating these results with IPM based decision making will be help to help the sustainable efficacy of these insecticides.
Thank you, Chinmay. It's well presented. So open for discussion. Any questions from audience?
Well presented, Chinmay. Do you have data on insecticide usage, particularly in those regions where you have conducted your study?
Yes, sir. I have collected an information that most of the farmers who are using emamectin benzoate, still they are saying that this emamectin benzoate is not effective against fall armyworm. But as in earlier in morning discussion, they have said, as Mulli Mohan sir said, since this pest will reinfest after 7 days, 7 to 15 days. So most of them are saying like that. Still, emamectin is effective in field conditions, and most of the farmers are using this. Apart from this, most commonly used next one is chlorantraniliprole and spinotorum. Sir.
Yeah. So if you see the insecticides, whatever you have chosen, so some of the insecticides are very novel, and recently they got label claim, particularly for fall armyworm on maize. But your results are showing that, say for example, isocycloserum in Telangana, it is for that molecule, insect has developed already 6-fold of resistance. It's somewhat alarming. So that's why I just wanted to know, does the farmers are using extensively those particular molecules?
No, population, as I collected the population from ICRISAT region, since the ICRISAT region is mainly focusing on a genetic material like seed material, there is a huge dumping of insecticides, sir. Actually, they will take more multiple sprays of insecticide inside the campus, since it is mainly focusing on the genetic part, that is seed part. There is a huge high number of sprays.
Do you have any data? Do you have any data regarding pesticide usage pattern, particularly on fall armyworm in those regions?
No, sir. Now I am studying on it.
Okay. Okay. Very good.
Chinmay. So I have just one small observation. So FA is developing resistance against insecticides over years, and over years the FA incidence is coming down in India. So how you justify this statement?
Sir, it may be alarming signal for us. It may reduce due to our environmental conditions, like huge rainfall is happening in our country, and maybe in further days it may boom out and resistance may develop in a quick sense.
Okay, thank you.
Still, there is no field level resistance with respect to any insecticide, like novel insecticide or commonly used insecticides.
Okay, thank you.
Okay, thank you, Chinmay. Well presentation and good answer. Next, move on to another topic. Mayur B. More. More. Yes. And More. So it's from College of Agriculture, Maharashtra, Luthar, and field evolved insect resistance in Pseudoptera frugivanda, population of the Marathwada region, Maharashtra. So you can start.
Good afternoon to everyone.
1 minute. Slide move.
Good afternoon to everyone. My name is More Mayur Baba Sahib. Today I am privileged to present my research entitled Field Evolved Insecticide Resistance in Fall Armyworm Population of. As we know that the fall armyworm is the invasive pest and labeled as agriculture important concern, and it's have a high reproductive rate, broad host range and rapid dispersal, and its nature is a polyphagus. It attack on a multiple crop. So to control of this pest, farmer overuse the insecticide. Due to this, this pest started developing the resistance, and it impact in also the environment. So sustainably to manage this pest, we need a region specific management strategy. So to address this challenge, our study evaluated the field evolved insecticide resistance in fall armyworm population across the maize field in Marathwada region of Maharashtra. So the primary objective is to evaluate the field insecticide resistance in Spodoptera frugiperda population from the Marathwada region of Maharashtra. Our study provides the latest insight about the resistance status of fall armyworm in a major maize growing region, which is Marathwada. So firstly, we collected fall armyworm larva across 8 districts from the different region. Then we rear it in laboratory and for the generation for F1 generation, and we conducted the lip dip bioassay on second instar larva, and a susceptible strain was maintained 7 generation without the insecticide exposure as a baseline. And we selected the 5 insecticide, that is chlorantraniliprole, spinetoram, flubendamide, imamectin, and lambda cyhalothrin. And mortality was corrected by the using the Abbott's formula, and we did analysis with Obstat software. So this is the this is the area that we have selected for conducting the study. And second image is showing the insecticide that we have used in conducting bioassay, and that different concentration. And this image is showing the overview of my research from field sampling to bioassay, which include the collecting of sample, then rearing it in laboratory, then preparation of insecticide concentration, then performing the lip dip bioassay, and assessment of mortality count. So our result shows the district-wise variability in the ELC50 and the resistance ratio of Spodoptera to the 5 insecticide in Marathwada region. For spinetoram, the highest resistance ratio shown by a Latur population, which is 3.97, and the lowest resistance ratio was found by Nanded population, which is 1.17. And for imamectin benzoate, the highest resistance ratio was shown by the Dharshu population, which is 2.55, and the lowest resistance ratio found by Nanded, 1.15. Same as chlorantraniliprole, the highest resistance ratio found for Dharshu, which is 3.02, and the lowest resistance ratio found for Parbhani district, which is 1.16. For lambda cyhalothrin, the highest resistance ratio found for Jalna district, which is 15.93, and the lowest resistance ratio as compared to Jalna found for Chhatrapati Sambhajinagar, which is 5.65. For a flubendamide, the highest resistance ratio was recorded for a Bid district, which is 83.47, and the lowest resistance ratio was recorded for Nanded district, which is 17.8. So this data shows the heterogeneity in all the insecticide resistance ratio. So conclusion, our study detected the significant heterogeneity in insecticide susceptibility population across the Marathwada region, which is due to the highest selection pressure and the high insecticide used by the farmers. Imamectin benzoate, chlorantraniliprole, and spinetoram demonstrated the high toxicity and low resistance ratio, which confirming their continued susceptibility in resistance management in fall armyworm program. And for lambda cyhalothrin and flubendamide, markedly elevated resistance ratio was shown, and susceptible resistance development and reduce the field efficacy. So our result underscore the importance of data-driven insecticide rotation and the avoidance of chemistry showing the early resistance signal. So our findings strongly support the integrating the routine resistance monitoring within the fall armyworm management framework. And overall, our study provide the necessary for the scientifically grounded and sustainable and region-specific stewardship to support a long-term fall armyworm management in a maize ecosystem. Thank you.
So good presentation within a time. So now open for discussion from audience. Yes. Only 4, 5.
So I feel like so whatever the title you are given, so you are mentioning it as a field evolved resistance.
Yes.
Okay. So when you choose the title for your study, you should be very careful. So the definition of IRAC for resistance, what it says is, so resistance is nothing but heritable change in the sensitivity of a pest population, which is reflected in repeated failure of the product when it is used according to the label claim recommendations. Okay.
Yes.
Label claim. If you come to, if you could come to the table, I will tell what it exactly the meaning. So you say if it is like emamectin benzoate, when it is used at the label claim recommendation.
Yes.
So if you take 0.4 gram per liter as a recommended dose, so if you calculate when you are doing bioassay, it will come to 40 ppm. You compare the 40 ppm and your results. Your results is saying 0.0 something, and the recommended dosage is 40 ppm. It is similar with all the molecules you are shown whatever the results in your study. So what I would say is, instead of you are saying field evolved resistance, you can say it a change in the sensitivity of the fall armyworm to the insecticide. It's not like field evolved resistance.
Okay. Sir.
Okay.
Okay.
If you just see, because when you are presenting here in international forum, then you are misleading the delegates and also the people. That was my concern. Maybe you can revisit your results, because we are all like, we are being working on same aspect. I'll tell the results directly. In the sense, so for emamectin benzoate, the recommended dosage is 40 ppm. For spinetoram, recommended dosage is 67.5 ppm. For chlorantraniliprole, it is 75 ppm. You compare for all three, your results are way less than those recommended dosage. You should not be called as field evolved resistance. Maybe I request the chair and co-chair of the session to look into this issue. Thank you.
Okay, thank you.
Sir.
So thank you, Dilip, for your suggestions. Mayur.
Yes, sir.
So what is your suggestion or recommendations for farmers who are growing forage maize? Because you are saying all your new insecticides, so they are developing resistance. So what is the choice then next?
Sir, I think rotational use of insecticide is necessary for farmers, sir. Rotational insecticide use. In first spray, if you use emamectin benzoate, then we should for a second spray we should use a chlorantraniliprole, sir, to avoid the resistance development, sir.
How about this cost of cultivation?
No, sir, we didn't calculate it.
Because one farmer who is going fodder maize, so why he will spend this much of money on chlorantraniliprole or spinosad? They are all very expensive.
But sir, instead spending on such an insecticide which not showing the good result, farmer can spend on one insecticide and then another insecticide. This will benefit the farmer, sir, maybe.
Okay, thank you.
Thank you, sir.
Mayur, as per the suggestion of scientist, so you know that it is an international seminar, and regarding topic title selection.
Yes.
So just modification we need.
Okay,
you know that you have to review all this based on the what type of insecticide they are used and all. for the study. So you are a PhD student or?
No, sir.
So try to modify that because no international seminar, the topic will attracts.
Okay.
That is the first titles. No. Okay. Thank you.
Thank you, sir.
Thank you.
You told susceptible population, no? You have taken susceptible population, and how you choose it as a susceptible population?
Ma'am, in laboratory we rear a 7th generation, ma'am.
In 7 generations, can you show the clear difference with respect to normal field population and also
Yes,
susceptible?
yes, ma'am. Yes, ma'am.
How much difference, for example, with respect to emamectin benzoate, how much difference you have found?
Yes, ma'am. You can clearly see for susceptible population, our analysis show the LC50 value 0.020, and for another it shows the elevated LC50 values.
Okay. And also how you classified that resistant population, like resistant ratios you got, no? How you classified as a less, medium or moderate or high resistant, based on what? Like
Ma'am, it is scale given by WHO, which shows the 1 to 3 is the low resistance, and then 3 to 10 it is the moderate resistance, and more than 10 is the high resistance.
You followed WHO.
Yes, yes, ma'am.
Like usually 5 ppm, like 5-fold, we'll call as a actually resistant, highly resistant.
5-fold, it is moderately resistance.
Actually, we can call it as a low resistant. More than 10-fold, 15-fold, we can tell it as a moderately resistant. And if it is more than that, we can tell it as a high resistant. Please correct. 1 second, recheck.
Okay, ma'am. Okay. Thank you.
Okay, thank you. Thank you. So next move on to Shambhavi, HT, College of University of Agriculture Sciences, College of Agriculture, GKVK, Bangalore. Susceptibility status of all armyworm to selected insecticides in Karnataka. Okay, you can start. Use that slide more.
Good evening, everyone. I'm Shambhavi, a PhD scholar from the University of Agricultural Sciences, Bangalore. I'll be presenting the data on susceptibility status of fall armyworm to selected insecticides in the state of Karnataka. Fall armyworm is a notorious invasive pest introduced from America and first reported in India from the Shivamogga itself in the year 2018. And it has a wide host range and can infest more than 70 family plants, preferring the grasses prevalently. And if not checked properly at proper time, it can cause yield loss up to or more than 50%. There is a report of 272 cases where it says fall armyworm has developed resistance against major groups like carbamates, OPs and others. But none of these reports are from India. It is necessary for us to maintain this status of susceptibility of this pest to particular insecticides in future days also. It is only prudent for us to incorporate the judicial usage of popularly used insecticides based on the data obtained by continuous monitoring. With this thing in mind, we have conducted a bioassay studies using fall armyworm as a test insecticide. Insect against popularly used insecticides. Firstly, populations were collected from various regions of Karnataka, and in my study they will be broadly classified into North Karnataka and South Karnataka populations. They were reared on chickpea flour based meridic diet. For bioassay studies, I have chosen 3 insecticides. These are recommended by CIBRC and are also popular among the farmers: Spinoterum, emamectin benzoate and chlorantraniliprole. For bioassay, newly moulted third instar larvae were chosen, and they were let to feed on the diet overlaid with different concentrations of 3 different insecticides. And after 4 days of treatment, the mortality was recorded. Each treatment was replicated thrice, and each replication had 10 larvae. The outcomes were analyzed using Excel, OPStat, and SPSS software version 27. The outcomes are like this. I have kept 3 graphs here for different insecticides. The green block indicates the South Karnataka population, and yellow one indicates the North Karnataka population. We don't have to worry about the light blue color, which is the laboratory population I have read up to F10, and the bigger blocks indicate the recommended dosage for each insecticide. We can see in Spinoteram that LC50 values are comparatively higher than the South Karnataka population, which is contrary to emamectin benzoate and chlorantraniliprole, where the values are comparatively bit higher in case of North Karnataka population. These values are also in line with the previous studies with slight changes. Even though the values are varying between the populations within one insecticide, they are not significantly different. Here we can see in emamectin benzoate, LC50 values of North Karnataka and South Karnataka populations are not significantly different, and that holds good for other insecticides too. This is the overall look of probit analysis. For different insecticides, the pink line indicates recommended dosage for each insecticide, and the green circles are LC50 values, the blue triangles are LC95, and the yellow squares are LC99. I don't know the exact number, but the recommended dosage is usually some number multiplied by LC50 or LC95, which we can see in the emamectin benzoate that the recommended dosage is comparatively higher than the statistical data. But in case of Spinoteram and chlorantraniliprole, we have to only concentrate on the triangles here. The LC95 values are on par with the recommended dosage, which is a fine thing, because in field condition also we are not trying to eradicate any pest. 90 to 95% mortality in field condition is just fine, and we are getting the similar results. Only it becomes a topic of concern when these values in triangle keeps moving upward. If the values keep on increasing in future days, it will become a concerning topic. We could also calculate the resistance ratios. I have used the data from previous years, like the population was collected during the year 2018, and the LC50 values were worked out. I have used the data for as a baseline value. The results are like this. If we look at the graph suddenly, first thing we notice is Spinoteram, South Karnataka population, it has the highest value of 18.32 resistance ratio. Similar to LC50 values, if the resistance ratios are more, the effectiveness of the insecticide is comparatively lesser. But the thing is not the same in other insecticides. Similar to LC50 values, in North Karnataka, both emamectin benzoate and chlorantraniliprole has the higher resistance ratio. But even though the values are looking bigger in the big screen, the less than 2 is not considered as resistance. If the resistance ratio is less than 2, it is not at all considered resistance. Maybe here 18.32 there is a low resistance, and the more than 2 less than 10 is a very little resistance development is there. Despite this, the insecticides are working well, maybe hopefully better in field conditions too. And these values are in line with other data also. And one thing I would like to mention is this is a paper from Northeast India. Here, if we look at the resistance ratio, the values are okay because these populations were collected before 2022, just after a few years after introduction of the pest. But if I take the baseline values I have referred, the values vary greatly, which means the resistance ratio that we calculate depends highly on the LC50 values of the susceptible population or the reference values that we use. We got to know all the insecticides are working just fine in the laboratory condition, but among the 3, which one is better? I have considered the relative potency, means the insecticide with highest LC50 value is the least potent one comparatively. So if chlorantraniliprole, we have to only consider SK and NK, which is the field population. If chlorantraniliprole is performing 1 time, how many times more the other insecticides are performing? Spinoteram is performing just more potent than the chlorantraniliprole. And similarly, previous data also shows here, if we see in the middle column, Kumar and Murulis, the efficiency of emamectin benzoate was 70 times more in the year 2018 and spinosad was 11 times more. But in my data, it is around 20 times more for emamectin and 1 to 2 times more for spinosad. The efficiency might have been reduced, but despite this, insecticides are working fine. To summarize my presentation, all the insecticides that are popularly even now being used by farmers are working efficiently, and among them emamectin benzoate is more efficient, and both North Karnataka and South Karnataka populations are responding in a similar way to their respective insecticides, and they are not significantly different in LC50 values or resistant ratios. Thank you.
Thank you. Very good presentation. Open for discussion. Sir. You.
Any questions from the audience? Shambhavi, you should have also avoided showing that last slide as per the guidelines fixed by these organizers, not showing the name of your chairmans and members. Okay, it is left to the organizers. But my question is, what is the logic of dividing Karnataka into North Karnataka and South Karnataka?
Sir, actually, the pesticide usage pattern is a bit different in North Karnataka. When I went for survey, North Karnataka people use more of emamectin benzoate and chlorantraniliprole together. But in South Karnataka, spinosad is little bit more used, maybe because of its recommendation on vegetables and fruits. I felt.
Is it assumption or a survey?
Sir, initially I did not plan to do that, but when I went for survey, I thought maybe there might be difference.
No, because when you are working on fall armyworm insect, your test insect is fall armyworm?
Yes, sir.
So you should have chosen maize growing areas district wise. So Davangere tops #1 maize growing district. So where farmers are using more insecticides for managing this insect. So you should have chosen district wise in Karnataka rather than dividing North and South Karnataka. It is already creating too much distance between the people.
I did not intend to do that, sir. I just wanted to collect population from all over the Karnataka.
No, you have done wonderful studies. So you should have chosen district wise maize growing districts of Karnataka. You should have chosen top 10 maize growing districts of Karnataka. So that would have been better studies. Because dividing South Karnataka, North Karnataka, there are so many reasons to divide people. But you should have chosen better title and better study area.
It's just a part of my research, sir. I have other states to cover. So Karnataka is just one among them.
Because maize is growing main in central Karnataka, neither north nor south.
Yes, sir. That is true, sir.
So this part is #1 maize growing Karnataka.
That is true.
Okay. Okay, very good presentation and thank you. Actually, in survey, you have to do logically. The district is a better rather than distributing south, west, north. Okay, thank you very much. Okay, so nice presentation. So, yes, sum up madada mugitu. So, okay. Okay, thank you very much for completing the 4th session, very good session, that is insect resistance in fall armyworm. So in that totally there are 2 keynote address made by David Mota. So clearly the Mota actually addressed the speak about evolution and of the pesticides from 1914 to 2005, and also what is the resistance mechanism, like that, the population studies and all. And this was very elaborative. And followed by another important Louis Jorith, identifying mechanisms and detecting resistant alleles to BT maize. This is one fascinating actually keynote address. He has made several studies, mechanism and detecting resistance alleles in BT maize. So about especially WIP3A resistance like that, and also at molecular science and mechanisms, he made several studies that to cry F1 like that, even whole genome sequencing and all. And this was very good study and also best lecturing, followed by Shatudna Shiva from USA, field evolved resistance, the same, FIM. There also that is especially the all analysis including the dosage and response, high level resistance in all. So he has made several achievement, and that followed by our Indians, Shashpini also microbial alleles, gut bacteria, that is an important studies made by Shashpani, and Amog, so microbiodata, that is a gut as a modulator, so it is an important study. And another speaker, Chinmay, so variability studies. So this is also the regarding armyworm, so that is population study. It is a novel approach, and Mayur. So he studies, that is a modification as per the title, insect resistance, that is field evolved instead of that some title. And it is a good also study. Several generation he made that with, and finally Shambhavi. So this is a status, what North Karnataka and South Karnataka, including all studies he made. And I would like to congratulate all the speakers and keynote address. Well, thoroughly made all steps, including introduction, material methods, and results discussion with photography and all. Congratulations to all of you and also the audience. Thank you. Thank you.
Thank you, sir, for nicely summarizing. So I congratulate all the young speakers. So you have presented your results very well. So kindly publish all your findings in very high rated journals, and whatever any suggestions made is only for improvement of your understanding and presentation. So not to mean to hurt somebody or to say something. So whatever any presentation, any questions asked, only to improve your answering ability and improve your presentation further whenever you go to any next platform or further to move up in your career. Thank you, Ananda. Thank you.
Over to.
Good evening to all. So we reached the sixth technical session, that is the last technical session, that is academy industry interface. Basaraj, is it audible, sir?
Good evening, Dr.
Yeah, is it audible?
Sharan Deshmukh and
yeah,
conference participants. Hope I am audible and visible.
Yeah, thank you.
I assume.
Yeah, just give me 1 or 2 minutes to introduce.
Set for this next 10 minutes discussion or my presentation. Can I get a prompt, Dr. Desh? Otherwise,
Basu?
It may sound like monologuing to me, but nevertheless, if everybody is up there ready, I can get into the presentation more.
Wait for 1 minute. Basu, is it audible?
Why? Yes, PPT is not loading. Vasudha?
Hello?
Yes, sir. Can you do the display of the presentation, please? Okay, sir, I'm going to wait for 1 more minute.
Is it audible? 2 minutes, not more than 1 minute I will take. ಹೌದು ಸರ್. ಇದು ಸಹ ಸೇಮ್ ಅನಿಸುತ್ತೆ ಬಸ್. ಹಾಂ ಸರ್. Yeah.
Yes, sir.
Basu, is it audible?
Am I audible?
Yeah. Is it audible?
Fantastic. Fantastic. When you prompt, I will start, Dr. Deshmukh.
So I'm very happy to welcome Dr. Basuraj Girennavar, the Managing Director of BioBrahma Private Limited, Mumbai. And also he is my best friend since from UG days. And also glad to invite for this international conference on sustainable management of fall armyworm. To introduce in brief about Basuraj Girennavar, so he completed his undergraduation from US Harvard and post-graduation from Haryana University and PhD from Texas University. And he is having a lot of experience with respect to the molecular aspects of pest management. And also he is an entrepreneur and having a wealth of knowledge, I can say, with this brief introduction. And also he is a farmer son. That is very, very important one. And he started his career starting a Creagen private company at Bangalore, nearly 500 crores worth turnover yearly. So now he started another company that is BioBrahma. With this brief introduction, I invite Dr. Basuraj Girennavar to deliver a talk on converging forces, how collaborative innovation will redefine crop protection. Basuraj, over to you, sir.
Thank you, Dr. Deshmukh. It is a privilege, indeed a great opportunity to connect with your conference and audiences which are going to be participating from academy and industry across the country and the globe. My topic of this, I know we are already late and I'm the last speaker. I just made a note. I'm going to spend next 10 to 12 minutes on how do we converge the forces so that collaborative innovations can bring better IPM or integrated pest management between industry and academia. Basically, what is a new model for us to engage so that we can better address the coming challenges in the integrated pest management, more specifically FAW. That I have seen Dr. Deshmukh putting together, identifying the pest and publishing and making this FAW firsthand during 2017, 18 and 19 from Bangalore around. Congratulations to you, Dr. Deshmukh, for being so persistent on FAW and from those initial field days around Bangalore to an international conference. Congratulations to you. I'm so proud, so happy to be part of this. Vasudha, I request you to next slide, please. I think there is an urgent need for us to collaborate in India for IPM. When you look at Indian context of agrochemical industry, our pest management, our pesticides more specifically, we don't have one or two molecules we can claim. You know, we are pioneers. Hence, we have an opportunity to reclaim or claim. Given that background, we also have a huge climate pressure, climate change, pest emergence, overuse of pesticides. Hence, there is a IPM, integrated pest management solutions, which are needed for us to practice a sustainable agriculture. And for this, we need to unite. United innovation between industry and academia is the only way forward in terms of innovating commercialization at scale, which helps the farmers to stay ahead of the pest while production of the crop and be resilient when the climate is not good or the pest emergency is too big of a threat. So for this, it's important call for industry academy to engage to craft this IPM story or opportunities. Next slide, please. I think I wanted to tell you about the current state, the gaps and opportunities in Indian industry and academia collaboration. I think we have a fantastic ICR institutions, universities, KVKs, crop-specific institutions, or pest or problem-specific institutions, along with IITs, which are also global eminent institutions. However, when you look at the partnerships, collaboration between these world-class research institutions having 50 years, 60 years to 100 years of experience, and industry partnerships and collaboration, it is not going well, folks. Look at there are no incentives. When I look at the critical barriers, which I listed 4 of them, look at the number 1 is a fragmented incentive structure. People get motivated by incentives, be it monetary, be it career, be it, advancing their knowledge, opportunities, or, career. And the other one is siloed research approach. That's hurting us more than anything else. And third is limited technology transfer mechanisms. We have plenty, let me tell you, I go around the world, I attend World Agritech Conference in San Francisco to, the World Microbiome Conference. I see a lot of people coming and signing up the projects, technology transfer, research innovations, and figured out how do we do that transfer technology, right? In India, that is missing. And also skill set is missing, mismatch is big between industry and academia. Industry talks about only money and business, while academics sticks to petrol plates and, the lab. While the land, the farmer is struggling with his unit economics or crop production, objectives. The opportunity is very simple. How do we bring initiatives which are transformational in nature for both collaboration, productization, and impact and outcomes? For that, I think we need to recraft. I will cover that piece of information in next couple of slides. Next slide, Vasudha. I think I wanted to context what government is doing. Government is, I think, putting forward these couple of initiatives called National Education Policy, which aligns students and faculties or institution in line with industry requirement, joint research initiatives, and applied innovation in agriculture and allied sciences. I think NEP is one of the good initiatives, while also putting together this imprint and Uchatar Avishkar Yojana. These are the 2 initiatives where they are funding the milestone-based collaborative funding. I think that is out there. And the industry organization like Confederation of Indian Industries Global Summit emphasized the sunrise industries or emerging technologies like agri-tech, biofuture, economy, to establish a platform between industry and academia to exchange knowledge, information, and projects and programs to intertwine their objectives. So these are the government initiatives. However, we are having this phenomenal today. I wanted to pause for 10 seconds and mention, can we stitch these 3 things for FAW management? Because maize is one of the phenomenal crop, and FAW seems to be $1 billion pest in India. That calls for, serious planning, commitment, and collaboration at institutional level and industry level. Next slide, please. I think I wanted to mention a couple of success stories. See, ICR has signed up with Bayer to manage, the pest better so that the pesticide could be reduced. Their objective is to reduce 30% of the pesticide consumption in cotton, while boosting the yield. That's very important, and also reducing cost of production and increasing income to the farmer or profitability of the farmer. I think that is one industry has done, one institution has done. Can we replicate, emulate one industry picking up one of those kind of partnerships? And look at IIT Delhi. They have launched an agri-tech initiative, which is going to be AI-driven pest detection tools, and they are into successfully commercialized with industry partners. Can we pick up, again, many pests, many crops, many opportunities in the country? And can we build a capacity building? I know, government has come up with DESI program to train a lot of dealers. Can we elevate a notch up and make the real entrepreneurs, which are going to be service oriented and technology oriented? Next slide, please. The future is it's bringing the talent and the technologies and techniques at scale, right? That needs, I think, curriculum transformation, aligning our academic programs, research, and embedding them into entrepreneurial thinking, practical and market-driven solutions encompassing IPM and productization of our IPM research or outcomes. And also experiential learning. I know as an agri student, 25 years back, we all went to Ravai program. Last 25 years, that has now evolved into experiential learning program. Good to see that. Can we push a little harder for students to pick up one crop or this thing end to end, where he has interest for masters on industry oriented knowledge? So I think experiential learning is going to give more confidence to the students or professionals, while nudging them to become partner for solutions, partnering with industry, communities, the farmers. And meanwhile, can we promote innovation hubs? While doing this, say for example, the big cotton growing areas of the Vidarbha needs an innovation hub for cotton pest. And similarly, Davangere, Belgaum needs one for maize. And Guntur needs for chili, and parts of East Godavari, West Godavari, or Telangana needs for rice. Hence, can we think about innovation hub? Those are the three things which are going to help facilitate the talent which is going to be ready for the future. Next slide, please. I wanted to quote, Dr. Deshmukh mentioned, hey, can you quote how it happens in the US or in Western countries? I may not be able to do justice, albeit I passed 9 minutes of my presentation. But when I went to US for my PhD was funded by, the Texas Citrus Mutual, like Texas Citrus Growers Industry Association, and USDA, and various other funding which was coming in from US AgriLife. So I'm one of the beneficiaries 20 years back because their industry was structured like that. Can we do that structural arrangements and cultural arrangements? Basically, the philosophy gap which exists in our industry, academia, for research. Researchers in India are stuck in TRL 1, 2, 3, while industry always looks at 7, 8, 9. And the funding and infrastructure gap between industry and academia is what we call the valley of death, or death valley. Nobody is coming between two of these TRL models. For others, TRL is technology ready level, TRL, whatever they call. That's from 1 to 9, while industry academia stuck in 1, 2, 3, industry stuck in 7, 8, 9. The valley of death needs to be addressed through structural challenges and through communications and through funding, industry, academia in a well-oiled machine. And also setting up a joint steering committees and transparent collaboration frameworks and continuous communication and trust building activities. I think these are the few things which are going to help a lot as we look at bringing the barriers down and facilitating the collaboration. Next slide, please. I'm going to touch upon the strategic roadmap. Okay, if we got to bring in more and more of the possibilities for pest management, like the FAW. FAW is a very ugly pest, having a huge economic impact. we can manage it. There is nothing like it is not like a pandemic like coronavirus. This is an insect which we can manage culturally, botanically, biologically, green molecules, blue molecules, yellow. Of course, Rambana, the red one, has to be there later. But IPM always tells manage, don't kill. Keep their population below ETL, right? For that, we need IPM alliances. For that one to happen, these major pest centers, crop centers, we need an innovation clusters. I think that is one of the key, item for interconnected strategies in terms of putting the collaboration to, translate the opportunity. Second one is IPM commercialization. See, everything has to be documented as an innovation if it is a novelty one, right? For that, we need to have a mechanism of licensing, revenue sharing, incentivizing, streamline. Let the government or one department or one university come up and do that in the interest of farmer, interest of the nation, interest of the ecology, and publish. Okay, here is what we standardized, right? Then we can copy as a nation, copy across the industries, across the institutions, so that we don't need to reinvent the wheel. And as I mentioned a while back, government is already through imprint and Uchitkar, the program, outcome-based funding. Industry should pick this up. Okay, let's give you 1 million rupees or $1 million, and then showcase the product, showcase the performance, showcase the adoption, showcase the revenue. We'll keep funding. The barrier between the university or academic researcher to industry researchers has to reduce and merge into one, because the projects have to be formulated like that. And continuous skill development. Look at 10 years back, the maize was a small crop, but still then there was no FAW issue or any pest for that matter. If I remember, this is the only crop with use of no pesticides in India. Today, this is also getting 2 sprays and sometimes 3 sprays. One to compulsory hai. I mean, one you have to spray, but 2 is also common. Given the challenging times, as we navigate the climate change to food production pressure, we need to have a continuous skill development for the students, professionals, researchers, industry folks. This can be accelerated through private-public partnership, right? So we need to formalize the pilots to validating the opportunities or the product projects and deploying IPM solutions at scale across the crops, agroclimatic zones. Next slide, Vasudha, please. See, I'm going to talk about a little bit, I already consumed a couple of minutes extra, but I'm going to leave with this slide. Hey, Basavaraj, you coming from industry and having a PhD, what is that 1 or 2 or 3 metric? If you can set the context for this wonderful conference, right? I'm going to visualize the future as India needs to emerge as a global leader in sustainable pest management through IPM technologies. I'm calling upon all the academicians and experts sitting here with partnership, the industry and the students, and obviously the farmers. Can we set something like this? 40% reduction in pesticide consumption. Let me tell you, pesticides, I hate it. It's inevitable. It's a necessary evil. But can we move towards alternatives? When India exports a lot of rice, we feel good about it, right? But when our rice also gets rejected because residual levels, we feel bad about it. And remember, we are also consuming that pesticides. Westerns are very particular about the residues, especially the red molecules and the yellow molecules, right? That could be one of the important metric. And the second one is, we have 5 lakh to 6 lakh villages in the country. Can we train one guy for those select crops in that village and identify the pest and give the techniques, tools, and make him professionally certified like primary healthcare givers in human industry to upskill as an agri-entrepreneur? And the third and the last is, when we talk about the Viksit Bharat 2047, can we set the stage by 2030 to build crop resilience and food security through integrated pest management and reduced pest consumption by the crop, by the farmer, by the consumers of the food. These are the three metrics. Next slide, please. I think call to action is something like this. Join the movement to transform India into a IPM hub or a leader in the world. Academia should get out of that, the lab part of it. Look at industry, what they want, how they are practicing, and get into the land, basically lab to land. And industry has to actively engage with academia and also put this innovation hubs right there where the market is, where the customers are, where the pests are prevalent, so that everything happens in one pane of glass at the site of action, which is crop cluster, innovation center, the crop, pest, farmer, industry, and innovators. I think that is need for us to stitch between industry and academia. While policymakers have to put together the policies which are funding in nature, which are supportive in nature, and also streamlining regulation. India is a hell a lot of difficult to register molecules. Let me tell you, especially green molecules. If I gotta go to register a biopesticide or a botanical is more challenging than chemical. So I request all the regulatory experts out there to ease that out and also reward this collaboration, reward the progress, reward the achievement, so that our big pesticide industry takes a beautiful green shape, sustainable shape. And also we have an opportunity, if we prove domestically, we have an export opportunity for this. Next slide, please. I think that was my last slide. Now, 30-second summarizing of this entire presentation in terms of convergence. Industry, academia, policymakers needs to unite with a larger agenda of Viksit Bharat to use less harmful pesticides, practice sustainable pest management, use integrated approaches, bring green technologies, green molecules, green initiatives, and enterprise all of this, put ecological sustainability and consumer preferences of no chemical or no harmful chemical residues beyond the tolerable limits. If we could prioritize those three metric and put this conference outcomes and collaborations, I think we can do a phenomenal job in terms of pulling together the opportunities and the innovations in the space of pest management, more specifically FAW and likes of FAW, and we can progress well, achieve well, and export to other countries as a knowledge hub and economic activity for Indian industry and Indian economy. With this, I thank all of you for patient listening, and I wanted to finish in 15 minutes max. Already I took 20 minutes. Without no further ado, I'm going to conclude my presentation, and congratulations to all of you for gathering and debating and discussing and taking initiatives in FAW at this international conference. And I wish you all the best. Dr. Sharan Deshmukh, over to you, sir.
Hello. Thank you. Thank you very much for elaborative address by Dr. Basaraj Girananavar. So actually, in this address, especially industry academy, so in that the current status of what the gaps and opportunity in India, so address made by Girananavar, and also new pathways in collaborative and also the initiatives regarding IPN and also global meet 2020-25 also made successful emphasis given on success stories, pesticide consumptions reductions. So that is a main theme, and also building the futures, especially IPM initiatives, curricula developments, or like that experimental learnings, especially in the universities and innovative hubs, and also mentioned about overcoming the barriers regarding both, especially in India, cultural and structural challenges made, and finally briefed what is the roadmap for to solve this the IPM regarding all clusters and IP, and also the skill developments and based on the public and private partnerships and also India as a global leaders where we can have the reduction of pesticides about 40%. So that address made by this elaborative and informative. Congratulations, Dr. Basaraj, for his valuable address. Thank you very much.
Thank you, Dr. Basaraj sir, for sparing your valuable time and delivering a lead talk. Thank you so much. On behalf of KSNUAHS Shivamogga and the knowledge partners like ICRISAT, then CIMIT, UAS Dharwad and ESI, and also from organizing committee, wholeheartedly thank you. Thank you so much.
Thank you, sir.
As we come to the end of this technical session 4 and 6, I would like to thank all our distinguished presenters for sharing their valuable research and innovative ideas. My heartfelt thanks to our session chair, Dr. Dushant Kumar sir, for your guidance and leadership throughout the session, sir. And I also thank co-chairman, Dr. Narendra Kulkarni sir, for enlightening us with your kind words of wisdom and for your suggestions throughout the session, sir. I also thank Dr. Srinivas Parami sir, also in his absence as he left in the middle itself due to some urgency. I also thank our audience both on-site and joining us virtually for their active participation and enthusiasm. I also thank rapporteurs for their patience listening and recording the proceedings of this session. I cordially, now let me invite the organizing secretary to present a memento to chairman and co-chairman of this session as a token of gratitude. I request Chairman sir, Dr. Dushant Kumar sir, to receive the memento. Now I request co-chairman sir to receive the memento. I request chairman and co-chairman sir to give certificates to rapporteurs. I request chairman and co-chairman sir to give certificates to rapporteurs. Bani. Bani, ikkada bani. I thank organizers for giving me this opportunity to serve as a moderator in this session. And I use this opportunity to thank each one of you for sparing your valuable time with us. Have a wonderful day ahead. Thank you very much.
ಅದು ಕಟ್ ಕಳಿಸು. ಮ್ಯಾಮ್ ಕಾಲ್ ಪಿಕ್ ಮಾಡಿ ಮ್ಯಾಮ್, ಎಲ್ಲಿ ನೋಡ್ತಾರೆ ಮ್ಯಾಮ್? ಅಲ್ಲಿ ಬಾಲಿಂಗಲಿ ಸರ್ ಹತ್ರ ಇಲ್ಲಿ ಡ್ರೈ ಫ್ರೂಟ್ಸ್ ಇದೆಲ್ಲ ಬೌಲ್ಸ್ ಬೆಳಗ್ಗೆ ಅರೇಂಜ್ ಮಾಡಿದ್ವಲ್ಲ ಇನಾಗ್ರಲ್ ಸೆಷನ್ಗೆ, ಆ ಥರ ಅರೇಂಜ್ ಮಾಡಬೇಕು ಮ್ಯಾಮ್. ಅದು ಬೌಲ್ಸ್ ಎಲ್ಲ ತೊಗೊಂಬರ್ರಿ ಮ್ಯಾಮ್ ಸ್ವಲ್ಪ. ಅನುಷಂಗೆ ನೀವು ಅನುಷಾ ಅಲ್ಲಿ ಹಿಂದ್ಗಡೆ ಹುಡುಗಿ ಫೋಟೋ ತಕ್ಕೊತಿದ್ದಾರೆ, ಅವ್ರಿಗೆಲ್ಲ ಸ್ವಲ್ಪ ಕರ್ಕೊಂಡು ಹೋಗಿ ಮ್ಯಾಮ್. ಹಾಂ
Good evening, one and all, respected dignitaries, distinguished scientists, delegates, scholars, and dear students. With immense joy and gratitude, I welcome you all to the valedictory function of the International Conference on Sustainable Management of Fall Armyworm, abbreviated as INCOFA 2025, a conference that has brought together brilliant minds and pioneering ideas on various aspects for the sustainable management of fall armyworm. Over the last 2 days, we have witnessed enriching sessions, insightful discussions, and a wonderful exchange of knowledge from experts across various institutes. As we gather here for the concluding ceremony, let us celebrate together the success of this remarkable scientific event. To begin this valedictory function with divine grace, I now invite Ms. Ananya for the invocation.
ಶರಣೆಂಬೆ ವಾಣಿ ಪೊರೆಯೇ ಕಲ್ಯಾಣಿ ಶರಣೆಂಬೆ ವಾಣಿ ಪೊರೆಯೇ ಕಲ್ಯಾಣಿ ವಾಗಾಭಿಮಾನಿ ವರಬ್ರಹ್ಮಾಣಿ ಸುಂದರವೇಣಿ ಸುಚರಿತ್ರ ನೀ ಶರಣೆಂಬೆ ವಾಣಿ ಪೊರೆಯೇ ಕಲ್ಯಾಣಿ ಜಗದೊಳು ನಿಮ್ಮ ಪೊಗಳುವೆ ನಮ್ಮ ಹರಿಯ ತೋರಿಸೆಂದು ಪ್ರಾರ್ಥಿಪೇವಮ್ಮ ಪಾಡುವೆ ಶ್ರುತಿಯ ಬೇಡುವೆ ಮತಿಯ ಪಾಡುವೆ ಶ್ರುತಿಯ ಬೇಡುವೆ ಮತಿಯ ಪುರಂದರ ವಿಠಲನ ಸೋದರ ಸೊಸೆಯ ಶರಣೆಂಬೆ ವಾಣಿ ಪೊರೆಯೇ ಕಲ್ಯಾಣಿ ವಾಗಾಭಿಮಾನಿ ವರಬ್ರಹ್ಮಾಣಿ ಸುಂದರವೇಣಿ ಸುಚರಿತ್ರಾಣಿ ಶರಣೆಂಬೆ ವಾಣಿ ಪೊರೆಯೇ ಕಲ್ಯಾಣಿ. ಥ್ಯಾಂಕ್ ಯು.
Thank you, Ms. Ananya. Your prayer has truly set a positive tone for today's valedictory program. I request Dr. Vijay Bhamre sir to please occupy the dais. Please. Sir. So, as the saying goes, a journey of 1,000 miles starts with a single step. And INCOFA 2025 has begun with a vision, and today we stand at the successful culmination of that journey. It's my pleasure to invite the man behind this vision, Dr. Sharan Basappa S. Deshmukh, Professor of Entomology and Organizing Secretary of the INCOFA 2025, to deliver the welcome address and also present the proceedings of the conference. Please, sir.
Good evening to one and all. I welcome you all to the valedictory program. At the very outset, I am so happy to welcome our beloved teacher and also Honorable Vice Chancellor of Gulbarga University, Kalburgi, Dr. S.S. Udikeri sir, for gracing the occasion. He is a great entomologist. He worked particularly on Helicoverpa armigera, more than 30 years on cotton, and also he worked on insecticide resistance. He is a very well-known scientist, and we are so proud of you for gracing this occasion, and you are with us, sir. Sir, on behalf of organizing committee and KSNUAHS and from all the delegates, I wholeheartedly welcome you, sir. Thank you, sir. It is a proud moment to welcome our Director of Education and in-charge Vice Chancellor, Dr. B. Hemla Naik sir, gracing the occasion and as a President of today's valedictory program. Sir, on behalf of the organizing committee and from the delegates, wholeheartedly I welcome you, sir. It is a proud moment to welcome our Director of Research, Dr. B.M. Dushant Kumar sir, for gracing the occasion. On behalf of organizing committee, I wholeheartedly welcome you, sir. I also welcome our Dean PGS, N.S. Mavarkar sir, for gracing the occasion. On behalf of the organizing committee, I wholeheartedly welcome you, sir. I'm so fortunate, or we are so fortunate. We are having one more very important person, none other than Dr. KSNUAHS sir, my teacher, and Dean Postgraduates from University of Agricultural Sciences, Raichur. He worked on pink bollworm, fall armyworm, and also mating disruption technique. On behalf of organizing committee, we wholeheartedly welcome you, sir. I also welcome our Dean Agri and Campus Head, Dr. D. Tippesh, sir. On behalf of organizing committee, I wholeheartedly welcome you, sir. Our university librarian, Dr. B.K. Shivamogga, sir, entomologist. I wholeheartedly welcome you for this valedictory program, sir. It is a proud moment for me, those who sponsored generously, and I wholeheartedly welcome all the sponsors. It is a big list. You might have seen in all the posters. One eminent the person, Dr. Mishra sir, from Crystal Crop Care, New Delhi. He is gracing the occasion. On behalf of the organizing committee, I wholeheartedly welcome you, sir. I also welcome our head of the department, B.C. Hanumanthasamy sir. On behalf of organizing committee, I welcome you, sir. I welcome Dr. V.K. Bhamre sir from College of Agriculture, Latur. He took 17 students. All the students, they worked on fall armyworm. When I contacted Dr. Saab, tension mat lo. I will be with you, and I will send all my students, and I will be also joining for your international conference. Sir, on behalf of organizing committee, I wholeheartedly thank you, sir. I also welcome our controller of examination, Dr. H.K. Viranna, sir, and also chairman of the program committee. Sir, on behalf of organizing committee, I wholeheartedly welcome you, sir. I welcome Dr. Hosmani, sir, special officer, UAS Raichur, and N.S. Kulkarni, sir, Hanchinal, sir, and from private company, Raghunathji, Pheromone Chemicals, and all the sponsors, and from our university, Dr. Nagaraja Adoppa, sir, and my colleagues, Rajshekarappa, sir, and Girish, and all my students, scholars, and Vinay is there at the end, and all other scientists, and also the students. I wholeheartedly welcome you for this the valedictory program. In 2 days marathon session, we completed all the 6 themes. Totally, we received 140 abstracts, 60 oral presentations, and 40 the poster presentations. Nearly 95% attendance we got. Out of 140, nearly 130, they presented the oral and poster presentations. The program was inaugurated by esteemed scientist, Dr. B.M. Prasanna, sir, a well-known maize breeder, and also having wealth of knowledge of entomology, particularly on fall armyworm. He addressed the gathering, and he suggested there is a need to relook with respect to the sustainable management of this fall armyworm. There is a more chance of using of biocontrol agents, mating disruption technologies, and other novel technologies. There is an urgent need. Don't dependent on only the pesticides. So he gave his wonderful remarks. And also the chief guest, Dr. S.N. Sushil sir from ICAR, NBAAR, Bangalore, he emphasized there is a need to utilize the three P's: natural enemies, predators, parasitoids, and pathogens. And the program was presided by our Honorable Vice Chancellor, Dr. R.C. Jagdish sir. And in his presidential remarks, he said, to organize a conference, we take 100 insects and we invite the scientists. When I approached, he said, how you will do this international conference with single insect and inviting many scientists? Then I briefed about this insect, convinced the Vice Chancellor and DOE and other authorities when they took a meeting in the month of July, sir. Then convinced and agreed to proceed to organize this international conference. I also thank the keynote speakers from USA and Ethiopia, then Germany, and many of the scientists joined internationally. Dr. David Mota Sanchez, one of the world-renowned scientists working on insecticide resistance exclusively on fall armyworm. One more scientist, Jurat, from again USA, he is working exclusively on BT resistance with respect to fall armyworm. And many lead speakers, they delivered a talk exclusively on fall armyworm with respect to biological control, with respect to the IPM, with respect to climate change. Sir, nearly 12 lead speakers across the country delivered their talk on fall armyworm. I wholeheartedly welcome sparing their valuable time with us and sharing their knowledge to the youngsters. So on behalf of organizing committee, I wholeheartedly welcome all the keynote speakers and also the lead speakers. And there are awards. We are hosting the awards for the best and best oral and poster presentations. And we are also bringing some recommendations in the form of proceedings, and we will submit these proceedings to the higher authorities or Entomological Society of India. With this brief introduction about the proceedings of the 2 days international conference, and also giving me a chance to welcome all the delegates on the dais and off the dais. I wholeheartedly once again all the delegates on the dais and off the dais. Thank you very much, sir.
Thank you very much, sir, for your warm welcome and also for summarizing the key highlights of the conference. On behalf of all of us, I welcome you to the valedictory function of the INCOFA 2025. Now, with great honor, I request our chief guest, Dr. S.S. Udikeri sir, Vice Chancellor of the Gulbarga University, Kalburgi, to address the gathering. Please, sir.
Good evening to everyone here in this beautiful hall, and with the first thanks to my young friend Deshmukh. He puzzled me a few days back. Sir, you were invited for the valedictory function,
And the second puzzle was from my wife, that should be on Saturday, why it is on Friday? Then he said, sir, it would be on Friday only. Saturday, it is going to be a post-conference trip for them. Then I clubbed my Mysore trip and here. It's my pleasure, great pleasure to be here in this beautiful, good, glamorous environment. Respected President of today's valedictory function, the in-charge Vice Chancellor and Director Education of this great university, Dr. Hemla Naik, my senior and good friend, and the Director of Research. Yesterday itself we could talk in the evening through one of my friend, and Dean PGS of this university, Dean Agri of this university, Dean PGS of new Dean PGS of Raichur Agriculture University, and the MD of Craft Care, and my friend Vijay Bamre, my good friend, ESI friend as well as cotton friend. And I feel very glad you have come with a big brigade here, and that should be the spirit of participation. ADR sir and my friend Hanumant Swami and colleagues from Raichur and other universities. Actually, it is true. These days, organizing conference itself is very much challenging. To pull the abstract and bring the people and make them to talk and to be here and keep them attracting and perambulating here only around the conference hall for 2 days or 3 days, it is not that easy. It's not that easy. But it's only one pest, very interesting pest, fall armyworm. It has made, and they have been dare enough. Saran Basappa has been become dare enough, and the university has sustained his pressure and made it very sustainable as well as successful conference. A big rounds of applause to Saran Basappa as well as to the university officials. Right. Especially in India, only this university can do that, and only this person and this university. Because now you have participated here, you have presented and you have heard, no why. It has been reported here only by him. Okay, that story started almost in one decade after they started, and every people, especially scientists, would like to run behind a successful event, successful issues or current issues. Anytime anyone would be like to work on Helicoverpa armiger, it's a evergreen insect, right? The problem will not be solved like that. ನಾನು ಒಂದು ಸಣ್ಣದೊಂದು ಜೋಕ್ ಹೇಳ್ತೀನಿ. ಕೆಲವೊಂದು ಯಾಕೆ ಅಂತ ಒಬ್ಬರು ಕೇಳಿದ್ರಂತೆ ಮಗನಿಗೆ MBBS ಆದಮೇಲೆ ಏನು ಮಾಡ್ತೀಯ ಅಂತೇಳಿ. I want to become a skin doctor, dermatologist. Oh, very crazy. Someone want to become surgeon, someone want to become MD, someone want to become eye specialist. Why skin specialist? Maga uttara kottante. He said, one, nobody disturbs me at midnight. ರಾತ್ರಿ ಬಂದು ಯಾವ ಡಾಕ್ಟರ್ ಬಾಗಿಲ ಬಡಿಯೋದಿಲ್ಲ, ಭಾಳ ಕೆರ್ಸತೈತಿ ಬರ್ರಿ ಸರ್ ಅಂತ ಹೇಳಿ. ಸೆಕೆಂಡ್ ಈಸ್ ಸ್ಕಿನ್ ಇದರಿಂದ ಯಾರು ಸಾಯೋದಿಲ್ಲ, ನಾ ಏನು ತಪ್ಪು ಮಾಡಿದ್ರು ಅಂತ ಹೇಳಿ. ಸೊ ದಟ್ ಈಸ್ ಕಾಮನ್ ಟೆಂಡೆನ್ಸಿ ಇನ್ ಅವರ್ ಸೈನ್ಸ್ ಆಲ್ಸೋ. I want to link it there. There is a common tendency, everybody want to become in the where a successful events or hot cakes are there, work a few days or few months and become a scientist there. Going behind a new insects, it may be accidental or now he may rethink back. ಅದಕ್ಕಿಂತ ಮುಂಚೆನೇ ಬಂದಿರಬಹುದು couple of years back also ಅಂತ ಹೇಳಿ. And why not in some coastal areas? Why not around some airports? That time Shivamogga airport was not active, right? Where is the reason? Why fall armyworm altogether moved when it has moved to India, and how we got it settled, and how my friend looked at it, and then there is a different story, and all those things have to be look upon. And whenever there is something new, you have to be after that. And that should be the spirit of being a science, especially for the entomology and breeders also, for looking for the new crops and new methods or something like that. But especially for entomology and pathology, if something is new, that would be very, very good. Okay, and sometimes if it is altogether not new, it brings a disappointment. Oh, that disappointment was there with meredbug for me. I thought it's altogether new, but entire India takes me as a meredbug scientist, but that's not at all a new pest. But it came into the limelight now, that's all. But the fall armyworm was new into this atmosphere, which is a global pest. So that was important, and it is carrying the history of resistance also, carrying the history of BT resistance, carrying the history of insecticide resistance, and economically prominent pest. There are many pests which have moved across the boundaries and invasive into different land boundaries and established themselves. And some are not that important. Spiraling whitefly is not that important to us now. Likewise, there are many insects, but the insects like fall armyworm are very much dangerous. So this has been found here, and that was not a lesson for him also, and to everyone also. Everyone has to take, whenever there is something new, become first to publish it. So publish or go off the line, not perish, off the line. Now perishing is different. Perishing also takes time. Immediately off. If you won't publish, it will be off. If Sharan Basappa couldn't have fought it properly, he would not have been here. Someone have been taking the all glamour of this one. He could fight, and that is also great history in our entomological this one and publication fraternity also. The credit ultimately was assigned to him because he has reported us. Okay, so why I am giving this information to? There are many scholars, MSc, PhD, and just who have finished. Whenever it is something new, you just start working seriously on that, whether it is that serious or not that serious. In 2022, one small farmer brought me some Galladia flowers which were rottening, and we worked, and it was a new pest. It's not that famous also. On that spray ಮಾಡಿಬಿಟ್ರೆ control ಆಗ್ತದೆ, but we could name, and that's altogether new species. That is Dacinura baswani. Baswani is a farmer's name who brought it to me, and my students at Dharwad, they know. We published it very recently in entomological news, published from the America, and sometimes it may take other end. And myself and one Vasant Kumar from ZSI have the credit of it as a joint authority. So that is important. That is very, very important. Whenever new pest is there, and later on many people started working in Karnataka, and the pest started giving opportunity to everyone. It went on touching each crop. Rupa is not there. She also find on some minor millet, but couldn't publish. Her student wrote it, but couldn't publish. So like it went to after maize, it was on sorghum, then on other crops also, and it attracted me also. I also could produce one thesis on cotton, but that was a Corona period. I particularly in Corona period making a room outside the hostel. That girl worked, Chaitra of my US Dharwad. She just finished MSc in 2022 or 23 like that. Could produce a small thesis and could publish only one. And that was quite interesting also. In entire wherever it has moved, and in its history, that's a cotton pest also. But why not in India? Why not in India? So those who are very, especially cotton scientists, as well as the fall armyworm scientist, I don't call them as a maize scientist, fall armyworm scientist, could take it very positively, that message, and readdress whether it feeds or not like that. And if that results are reproducible, better work on that again, and there could be some principle in Indian cottons which don't attract fall armyworm. Forcibly it could be feeding, that's all. Okay, and you can include those things. So you have heard many things since yesterday I could see many people, many scholars. I could meet R. Ashokan and some of my friends from ICRISAT and others also. Must have brought, Sagar was also there. He has produced one on gynandromorph, right? Gynandromorph. Quite interesting observation. Quite interesting observation. So this insects always gives very good opportunity. And our biocontrol laboratory of Raichur went to international fame for providing the mixture of or cocktail of bioagents, fungal bioagents, right? And people were in queue to purchase that. And that brought laurels to my friend Hosmani. So insect gives opportunity to everyone. Insects gives opportunity to everyone. Which is big, which is new, which is small, nothing is important. You have to go after the insect, like pest or any crop or something like that. Because as cotton scientists, we were very like a downtrodden around 98, 99, something like. Once BT cotton entered, you are all gone. You are all gone. Especially we were 3 in the Ebali farm, renowned international farm. We were 3, and I was new. Ivrige ibburge ಏನು ಕೆಲಸ ಇರುತ್ತದೆ ನೀವೊಬ್ಬ ಬಂದು ಕೂತಿ ಅಂತ. So I earned more the name than the other 2, because I touched new pest, merit bug, flower bud maggot, and then the pink bollworm came back, and my science, as you said, resistance to BT, resistance to insecticides. And once I was examiner, and his student was also there, a girl name I don't remember. We discussed about 3-4 hours. He has already by that time started work on the resistance also. And with that understanding, lot of understanding from him, I asked my friend, my colleague at Dharwad, Dr. Guru Prasad. He could produce 3 or 4 theses on that. Quite interesting, this one we have taken. One student was here, Krishnaveni, whether she has gone back or I don't know. Krishnaveni is here. Please be seated. Krishnaveni worked on adult toxicity, it seems, I remember. Adult toxicity as Krishnaveni? Adult toxicity and ovicidal action. Adult toxicity only. Okay. So adult toxicity, killing the moths at adult stage would end the problem of, but it never happens. That's the fantasy of the insects. You don't have the good adulticide. Adult response is very poor. We recommend pyrethroids for pink bollworm because it will render about 50 to 60% adult-side action. That's all. But any insect would be killed to or attract at adult stage, male or female, that would be fine. That would be, and ovicidal action also started. One international student was there, Miss Reyes. She has also produced 1 or 2 papers. So it was interesting to everyone. So keep on working. Many people are working biological control. My friend has worked on mating disruption, and he visited some Australian island, Solomon Island, as an FAO visiting fellow. Okay, so we bringing a sustainable solution for a pest like fall armyworm. It's not that easy. It's not that easy. Biological control, especially the most successful biological control agent would be the egg killer, but it lays the eggs in mass. It lays the eggs in mass. It's not in isolated. That is one of the hindrance. And now it's almost touching every crop. If it is a multi-crop, it's still dangerous. So still, there might be a lot of enlightenment since yesterday, a lot of challenges, and all together, one successful strategy, if we achieve here in through Karnataka, that would be a great achievement through this university, and give back our contribution of this will catch back. So there will be a big contribution from this conference, not only to the India, to the entire globe, because the Indian applications or Indian outcome, Indian research outcome would be applicable to the many countries, Indian subcontinent, others, and towards the east, it would be Chinese easily take our messages. That would be fine. And this conference has run for 2 days successfully, and tomorrow there are no scientific events. And immediately after this, I believe there will be award ceremonies, and many people will be eagerly waiting for you. Without standing between you and your awards, or you and your night plans or late plans, including my own plan running back to the home after a month, I just have 3 suggestions to this university and my young friend Sharan Basappa. I don't know right now it is there or not. You have a maze network, Sharan Basappa. You have a maze network. But earlier we used to have network project on termite. We had network project on root grub, which has run very successfully in Karnataka. Dr. Raj Gopal and all have heard name. So there should be immediately one network project, whether ICAR funds or state fund it or not. But this university should fund it immediately and then start getting the funds from others. So otherwise someone, these days everybody, this is a hot cake, everybody want to test that. No, overnight, don't know who will grab it, okay? Because in one of the conferences at Dharwad, I saw how it went on. Someone else was talking much that as if he has traveling in a flight with a fall armyworm, like that. And Sharan Basappa and me were laughing, okay? So let us not give, I don't want to take the name, okay? But things will happen like that in the science. So it's matter to this university to establish a network project immediately, either small or larger scale, on fall armyworm in India. This is my first request. And second is Center of Excellence or Advanced Center for fall armyworm, and if it appears a little bit small, invasive pest you can. Fall armyworm and other invasive, because there are many invasive pests, all are in Karnataka. Whether they go to other state or not, no, but Karnataka they will definitely visit. Papaya mealybug has visited, cotton mealybug has visited, coconut mite has visited, coconut whitefly has visited, okay, everything will come over. Fall armyworm, if it the dimension become too small, fall armyworm and other recent invasive pest. These two. And last is, we need an exclusive journal for the fall armyworm, because many insects and many crops have like that. Or you make it like this only again, fall armyworm and invasive pest. Okay, exclusive journal. Until journal, immediately the newsletter has to be started. Starting newsletter doesn't take much of the time. You start the newsletter, and with one or two copies, you register immediately for the journal, and you start writing your own this one. Don't wait for NAS and other things. Okay, even elsewhere publication, immediately they won't get the impact factor. After 3 or 4 years they will get. Okay, don't wait for that. But these 3 things should immediately happen, and this is my humble submission. And whenever this happens, I am the first person to be very much happy. And I thank everyone here for inviting me. And after 3 months of leaving my parent university and agriculture science, I am enjoying this evening, and it's as good as homecoming for me. And thanks for this opportunity, and thank for everyone. I greet everyone who has taken lot of pain and participated, who have heard to the many big names, who have made name and fame in the fall armyworm or other science applicable to the fall armyworm, and those who are successful in reaching the goal of getting some awards, and all others also. Basically, enthusiastic participation and good hearing is required, so that the temper of science and temper of being in the conference is very important. With this note, I again thank everyone for giving me the opportunity. Good evening again. Thank you.
Thank you very much, sir, for your insightful address regarding various insect pests and also for sharing your experiences on varied entomologists, and also we will try to inculcate your valuable suggestions. We are extremely sorry for the inconvenience that happened. Now, next, we would like to hear from the participants themselves. I request a few delegates to please share their feedback and experiences in the conference. It will really help us improve and enrich the future scientific gatherings. Any of the delegates, please. Participants. It's voluntary, please. I request Mamata from Telangana for share her experiences. Please, Mamata.
Good evening to one and all. Then I attended this Armyworm conference, sustainable Armyworm. I learned so many things from oral presentations and postal presentation. When I'm doing some pheromonal, that plant volatile compounds and related that information also I gained from one oral presentation. Not only from that one, and these 2 days I'm feeling wonderful and giving more experiences. And 2 days I'm morning to evening I'm full happy with this one. Thank you for giving this opportunity.
Any other delegates? Please come. Now it will be Pratiksha who will be addressing, who's a wonderful dancer. We really experienced her wonderful dance yesterday, sir, in cultural program. She's a wonderful dancer.
Thank you so much for this appreciation. Good evening to all the dignitaries, all the research scientists, research fellows, and all the students. So myself, Pratiksha Khedkar. Currently, I am pursuing PhD in Department of Entomology at VNMKV Purban University from Maharashtra. So I also thanks to Mr. Sharam Basappa Deshmukh sir and their team for organizing this wonderful conference. As we see, there are the different conference which have combined aspect of the different pest complex and like other subjects. But this conference is specifically based on the fall armyworm. As we know, from the 2018 up to the till, there are the different types of research have conducted in a worldwide. So there is a need to exchange this research and ideas in among all the different types of birds. So this was a great experience to come here, and we all the students in our literature and our all the research paper always mention that Sharam Basappa Deshmukh sir 2018 in introduction first sentence. So I also eagerly waiting to meet the sir, because we always think that this sir was reported that pest, but it was we are lucky to meet the Sharam Basappa sir, and we really appreciate. And thank you so much, sir, for bringing the best platform for all the students, because in this aspect we have means there are the different students who have worked on the fall armyworm. So we also got to know that which student work on which aspect. That will also helpful for the future collaborations. So I would like to thank you so much, sir, and all this organizing team and the College of Agriculture, Shivamogga, Karnataka, for bringing this opportunity to all the students. And also I would like to request her to please in upcoming days please make the same conference as like this. And sure, now there are the 70 students from our college, so next time the ratio will be increases. Thank you so much, sir.
Thank you very much, Pratiksha. We'll now have a brief remarks from our esteemed key officer, the Director of Research, Dr. B.M. Dushyanta Kumar, sir. Please address the gathering, sir.
Good evening to all. So, the final validatory section on International Conference Sustainable Management of Fall Armyworm. So, on the dais with Udkeri sir yesterday called and also invited today function regarding the timings and all. And so, respect to honor our Vice Chancellor Udkeri sir, and also so present in charge Vice Chancellor of KSNUAHS Dr. Yamla Naik, and also Director of Education, all the officers of our university, and Srinivas, Dean PGS, all the eminent scientists in the front, principal scientists and professors and all, and the participant, especially the participant and all delegates and the students and the staff of KSNUAHS. So last 2 days there are 6 sections, and regarding the major achievement made by our one of the eminent scientists, Dr. Shanabasappa. So 140 out of 140, 130 participated. It is the use numbers to conduct a seminar in 2 days. So what I have seen, what I have observed, many conference here, it is very difficult to accommodate even 50 or 60 in 2 days. So it is a huge number. Congratulations. So this is actually international conference. It is a high-level conference, as actually in the introductory remarks made by Udkeri sir, know that how the entomology, especially the university, how the hallmark of this department here. So as you know that I will start with I express my gratitude, especially the hard work made by this team and to complete 2 days, and followed by I acknowledge all the contributors to take up this event more successful, especially the teamwork of Department of Entomology and individuals, not only individuals, the staff of the department and staff of the college and the PG and UG students. And whatever the topics selected by all sessions, what is very the theme wise, and also the knowledge in which where breakthroughs are there in all aspects, all these sessions, especially what I chaired 2 sessions here. One is insect resistance to fall armyworm, and another one is industry academy interference. So in both what I observed, the international scientists made several addresses regarding the resistance mechanisms in fall armyworm. That too, David Mota and also Jesus. So both are actually including not only practicals, even IPM models, even they have identified the alleles and also made genome sequencing. So that is what the breakthrough, what even in India also, major scientists, so they have that, but I have chaired. So they have the knowledge regarding how we can go for all mechanisms, especially resistant mechanisms to avoid the chemicals or insecticides, how to derive so especially BT maize, where Jesus tell that. So likewise, the emphasis on key learnings, the insights made by the ideas emerged in this conference, and also the interaction discussion, valuable, fruitful discussions made in all sessions here, the participants, eminent the delegates, and also chairmen, co-chairmen. So discussed a lot and enriched a practical understanding. So that is what given here. And also I would like to congratulate all these participants, especially the speakers. They made the presentation in all aspects, including. So that is what the major, and also finally the industry academy. So Dr. Girish Birana Basuraj, yeah. So what even UG and PG students make some academic intelligence, especially in experiment learning and all, linked to policy makers to and address how we can go for the academia with the industry. So that is actually important sessions made that. So this department, as I learned from since 15 years, Department of Entomology, not only contributed in this, they also contributed many. So what one I would like to tell what I observed here, we have released in the university one of the top mega variety. So that is Sayadri Kempumukti. So that is actually released as a high yielding and blast resistance, but in All India Coordinated Research Project under NS1 and NS2, it is also showed high tolerance to BPH. That is not noticed, but these two scientists, Kalleshwar Swami and Sharana Basappa, both actually contributed in our field also lot. I would like to congratulate both team leaders here. So in this 2 days conference, you made successful the conference in all the ways, the way in which the food, accommodation and all they have given very well, and participation, they are so happy. So I finally thanks all the participant delegates. They are moving after completing tomorrow trips, and a happy journey to all of you. And finally thanks to all given me for some address here. Thank you very much.
Thank you very much, sir, for your valuable remarks. Now, as a gesture of respect and appreciation, I request our dear Director of Education, Dr. B. Hemla Nayak, sir, to felicitate our chief guest, Dr. S.S. Udikeri, sir, Vice Chancellor of Gulbarga University, Kalburgi. I request all the dignitaries to please join hands. Now, it is time to hear from our one more guest as well as a delegate for the conference, Dr. A.G. Shivamogga, sir, Dean PGS of University of Agricultural Sciences, Raichur. Please, sir.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. Good evening all. The respected dignitaries on the dais as well as off the dais and student friends. Let me thank organizers for giving me this opportunity to come over here and share my own experiences in respect of the fall armyworm. Either it is more so on the climate change issues, which I spoke here, yesterday as a lead lecture. It was quite interesting, like, of course, it was the opinion posed by all the participants. Fine. So a lot of work has been done even at Raichur also. In fact, I'm the one who did a lot of work on mating reception technology for majority of the pests, not only the majorly the pink bollworm, even the shoot and food borer of brinjal, helicorpa, tuta, everything, almost the 6 insect we did it. So that's about the research part which I have done it. Okay, so I'm not here to give the technical things and other things, like, let me give you the opinion about this particular conference as a participant. Fine. So for any program to be successful, okay, the 4 pillars are very important. So the first pillar is technical part, isn't it? The technical session which they have planned in a systematically the 2 days, the 6 technical sessions are wonderful, like, very wonderful. Both the posters as well as oral presentations, including the lead lectures, are also wonderful, like, that is first part. Then the second part is the food. Wonderful, like, in fact, normally, to be honest, so probably even in Raichur also we cannot give such a quality food, like, honestly speaking, it is not exaggeration. Even I have also, took food at Dharwad also, probably compared to Dharwad and Raichur, probably the quality of the food which they have given is excellent. And the third one is accommodation. Very well planned, neatly accommodated, comfort. So with respect to the accommodations also, it was wonderful, like. Then the transport also, without any single problem, even from Tunga guest out to here also, they made arrangements for the transport also. In the sense, all the 4 pillars are perfectly planned and executed. This is what I can opine. In terms of the marks which I wanted to, just for this one, okay, maybe for 10 marks, I wanted to give 10 out of 10. It is not exaggeration, but let me give 9.5. 9.5. I'll hold 5.5 because if I give 10, probably they will not conduct such events in the future. Probably to do that and to get some more, the 10 plus points, probably I'll give 9.5. Not that it is joke apart. In fact, I can give 10 out of 10. And what I have seen, Sharanu, probably, maybe not only the entomology guys, joining hands by the other discipline group like Naveen, wonderful. This is what the integration approach, that's what it is required, collaborative efforts by the other disciplinary people, other disciplines, that is what required. It is not working as solo, it is working together is the best way. Probably such events you can organize comfortably with the joining hands by the other discipline people. So that's what I have seen, wonderful coordination. At least I have seen the people working like Naveen, even Nagaraj sir, plus Shekharappa, even Virenna, our Swami. Normally Swami also, he has involved very well. I have seen first time Swami involving such a huge this one. And particularly Shivana sir, all entomology. Of course, entomology it is inevitable. For others it is, of course, voluntary, but still they have done it wonderfully, like. So in toto, I can say this conference, wonderfully it is organized, sir. Probably it is a memory. Okay, of course, it is all the efforts and the first step taken by the Sharanu Basappa, right? So the congratulations, Sharanu. You can organize such events in future also. As a seniors, and of course Gurujis, we are with you. We are with you to support you, including my friend the Udikeri also, he endorsed those. Okay, so we'll be with you for any matters, particularly to develop entomology in particular or other sciences in general. Anybody, if they wish to have our support from UAS Raichuru, will be the first. Thank you very much.
Thank you very much, sir, for sharing your enriching experiences of the conference. We now move towards a very special part of today's program. Ladies and gentlemen, please get ready for the most thrilling moment of the evening. The stage is now set for honor excellence dedication as well the remarkable achievements. Yes, it is time for the award session where we celebrate the stars who have truly made a difference. I request Dr. H.K. Veeranna sir to please lead the session and honor our awardees. Please, sir.
Good evening to all the delegates and of the delegates and all my dear students. I think you are all eagerly waiting for the names of the award winners. So let me not take much of your patience. For the information of the dignitaries on the dais, in the 2 days deliberations, as our Dean P. just told, we had 6 technical sessions. In all the 6 technical sessions, I think in the last session, since only one presenter was there, we are not giving the award to that particular session. But totally 5 oral technical sessions were held in 2 days duration, and poster presentation sessions were also to the tune of number 4. 4 poster sessions were there. And first of all, I thank all your active participation in all the 5 oral sessions and 4 poster sessions. Totally there were as many as, apart from keynote speakers presentation in all the technical sessions, 45 delegates, they have presented their research papers in the oral presentation section, and 47 participants, they have displayed their posters in all the 4 technical sessions. This is for the information of the delegates. And I must thank here all the juries of the awards committee in all the 5 technical oral sessions and also 4 poster sessions. And our organizing secretary also, he has taken utmost care to present maximum number of awards to almost half of or 1/3 of the participants could get the awards. In that way he has designed. Out of 92 participants, it's my pleasure to present the names of 25 award winners. So it's a huge number. Congratulations to all the prize winners. So I'll go session by session. In the first technical session on oral presentation was on climate change and ecology of fall armyworm. So here under oral section, the first place goes to a scientist from NBAR Bangalore by name Sagar D, who has presented the research paper on resilience or reproductive decline, a case study on the effects of acute and chronic heat stress in fall armyworm. I request Sagar to come on to the dais, and I request the dignitaries on the dais to present the award to Dr. D. Sagar. I request all the delegates to give him a big applause. And the second place goes to Dr. Ramya R.S. from the Division of Genomic Resources, NBAR again. She has presented a research paper on cracking the code before the cut, optimizing oviposition in Spodoptera pudiforgia for efficient CRISPR-Cas9 genome editing. And the third prize goes to Eureka Savdati from UAS Dharwad. She has presented a research paper on studies on the reproductive behavior of fall armyworm. So under the same theme, climate change and ecology of fall armyworm, under poster section for the same theme, the first place goes to Nishant D. Jatle. Nishant D. Jatle. She has displayed her research topic on thermal influence on the reproductive potential and survival of fall armyworm on maize. Nishant D. Jatle. And the second place goes to Sushant S. Birajadar. Sushant S. Birajadar from College of Agriculture, Latur, Maharashtra. He has displayed his research topic on assessment of Spodoptera fujiforida population, trends in Rabi maize in Marathwada region of Maharashtra. And the third place under the same poster session goes to Kushwarth Prakash Patil He is from Lupin Human Welfare and Research Foundation, Parbani, Maharashtra. Kushwarth Prakash Patil. He has displayed the poster on influence of climate change on the occurrence and severity of fall armyworm from Dhule district in Maharashtra. Under the second technical session for the oral presentation, the theme was on host plant resistance to fall armyworm. The first place goes to Deshika J from TNAU Coimbatore. Deshika J from TNAU Coimbatore. She has presented her research paper on identification of marker trait association for fall armyworm resistance in maize. And the winner of the second prize in the same technical session on oral presentation is Ms. Ramya AR, again from TNAU Coimbatore. She has presented a research paper on electrophysiological behavior and molecular docking analysis reveal adults' attraction to herbivore-induced plant volatile compounds. So in the host plant resistance to fall armyworm on the same topic with respect to posters, the first place goes to Jatin Kakara. Jatin Kakara from iTech Seeds India Private Limited. He has displayed his poster on the topic dissecting the genetic basis of fall armyworm resistance in sorghum through genome-wide association studies. And the second place under this poster session goes to Ms. Sahana BS from University of Agriculture Sciences, Dharwad. She has presented her research topic on deciphering defense mechanism conferring fall armyworm resistance in dual-purpose maize inbreeds. And the third technical session was on biological control of fall armyworm. So under this theme for oral presentation, first place goes to Ratnakala from College of Agriculture, US, Dharwad. She has presented a research paper on biocontrol efficacy of indigenous entomopathogenic nematode and symbiotic bacteria against Spodoptera pugiferida. And the second prize goes to Ms. Bhumika K from KSNUAHS Shivamogga. She has presented a poster on interactions of novel insecticides with entomopathogenic nematode on fall armyworm. And third place with respect to oral presentation in the same theme goes to Pratiksha B. Kedkar from Vasantrao Nayak Marathwad Krishi Vidyapit, Parbani, Maharashtra. She has presented a research paper on assessment of indigenous parasitides and entomopathogens for the sustainable control of Spodoptera pugiferida. Under the same theme, in the posters presentation, the first place goes to a scientist from Sairam B from CSIR, Indian Institute of Chemical Technology, Hyderabad, Telangana. His poster was on the topic potential of Lucanicillium licanae oil-based formulations for the biocontrol of fall armyworm. And the second prize under the poster section goes to Karan Selarka. Karan Selarka from CSK HPKV Palampur, Himachal Pradesh. And his poster was on biocontrol potential of indigenous entomopathogenic nematodes against fall armyworm. The fourth session was on insecticide resistance in fall armyworm. So under this theme, for the oral presentation, first place goes to Ms. Chinmayi S from KSNUAHS Shivamogga. She has presented her topic on variability in susceptibility of fall armyworm population to novel insecticides. And the second place goes to Shambhavi HT from UAS Bangalore. She has presented the research paper on susceptibility status of fall armyworm to the selected insecticides in Karnataka. I think there were no posters in the technical session 4. So now let's move on to technical session 5. So the technical session 5 was on the theme management of fall armyworm. So under this theme for the oral presentation, first place goes to Ms. Arpita KC from UAS Bangalore. She has presented a paper on larval process of Spodoptera fujiforida, a potent oviposition deterrent with potential in sustainable pest control. And the second place goes to, second place under oral presentation goes to Sham Supreet from UAS Raichur. He has presented a research paper on Cremit FAW, a novel mating disruption technology for the management of fall armyworm. And the third place goes to Karianna B from Division of Fluoro Agrochemicals, CSIR, Indian Institute of Chemical Technology. I think he is not here. On behalf of him, if anybody is there, you can receive the award. Okay. So under the posters for the same theme, the first place goes to Anusuya RM. She is from Vanavariyar Institute of Agriculture, Tamil Nadu. She has displayed her poster on anti-insect activities of botanicals against fall armyworm. So second place is again shared by two delegates. Second place is shared by two delegates. I call both the names of the second prize winners. First one under second place is Pawan JK from UAS Bangalore. And the second place also goes to Katla Soumya from PJT SEIU Hyderabad. Both of you can collect the second prize. Please come, please come. And it's a coincidence, third place also goes to two delegates. Third place is shared by two delegates. The first one is SS Patil from RCSM College of Agriculture, Kolhapur, Maharashtra. And the other third prize winner is Mamata T from Indian Institute of Chemical Technology, Hyderabad. So all the postgraduate students and PhD scholars are a sort of inspiration to our UG students also. So our third year BSc Agriculture students also displayed one poster, and they are also deserve to get some appreciation award by our honorable dignitaries on the dais. I call upon the names of Anugraha, Basma, Abhijit, Diya, and Ullas MY. All the students, please come on to the dais and collect your appreciation award from the delegates. These are all third year BSc Agri students, sir. They are inspired by the presentations of our seniors, and now they took part in the conference. So with this uh, honoring all our awards winner is over. I thank all our dignitaries on the dais for having distributed the awards to all the prize winners. Congratulations to all the winners. And those who have not got the award also, best of luck for their future in the forthcoming conferences or symposiums. Thank you very much.
Thank you very much, sir, for conducting the session. Every true event is success through great vision and guidance. I now request our respected Director of Education and also the President of today's function, Dr. B. Hemla Nayak, sir, to deliver the presidential remarks. Thank you, sir. Please.
Good evening, friends. So I think we are almost at the end of the day, especially for international conference we had since yesterday. So for this, we have a distinguished guest and real entomologist, and who is my good friend from Dharwad, Dr. Shashikant Udkeriji. And friends, of course, regarding Shashikant, you must have heard, he is one of the entomologists, renowned entomologist, and who has entered in the traditional university as a vice chancellor. It is a rare occasion, and he has showed the insect from agriculture even to the traditional university we can attack. So, Dr. Shashikantji, and especially my colleagues on the dais, all the distinguished colleagues on the dais, and one, of course, from industry, Dr. S.K. Misraji, and especially, of course, Bamreji is also a very renowned entomologist who has contributed a lot, especially for this conference. And my dear fellow colleagues and beloved distinguished delegates from different parts of the country, and who have attended this very, very memorable first international conference on INCOFA. Okay? So friends, of course, as the Honorable Vice Chancellor from Gulbarga, Dr. Shashikantji, he has given very informative especially on the insects, especially that INCOFA. So how it has created and how it has almost, we can say, awakened all of you. And whenever the organizing secretary was mentioning Dr. Sharanji, so we were really, university was reluctant to have this kind of the international conference, that too on one single insect. The Honorable Vice Chancellor was also doubtful, and when he approached, really I was also thinking, because it's a new pest, so whether we get to the response from the scientists from across the country and in the globe. So by looking this a number, especially almost 140 delegates and young scientists like students have participated in this, really it is the success of his motivation. So really we can say, especially on this insect, so which is not the major pest earlier. When yesterday we were looking for the history of this invasion of this pest, so which has, though it is entered in 17th century, but this has made really a noise, particularly in 2016 onwards. And we are really proud to say, from this college, from this university, Dr. Sharan and his team, they identified this crop and particularly introduced to the entire the country with respect to how it is a really menace for the crop like maize. And friends, of course, after this all, we decided to have this program, and this is really a successful. I'm so happy by just looking to your response and the feedback got from all the delegates, all the scientists, and with the number almost 66 oral presentation and 67 the poster presentation, including the what you call most of the lead talks were also excellent. So this is all only though we are in the entire system of this organizing of this INCOFA. The man behind this is Sharan Deshmukh has taken a lot of pain. And he contacted all these teams, especially as the AG Srinivas was mentioning. Many his friends are there, his supportive are there, including the administration, like Honorable Vice Chancellor and all my colleague friends of all the officers, and particularly the students of the department, entomology. They are very vibrant and they have showed their interest. And with this especially, I don't want to take much time here because this is an opportunity to all of you to think, to address how to address the new pests. And for this, though the research is very, very lesser, in fact, he has prepared very excellent the theme. And in all the theme, more than 95% the response is there. And they have presented excellent papers also. And all the in all the what you call the sessions, so the lead speaker as well as the keynote address is given by the scientists from across the globe through online, as well as many of the scientists like the Prasannaji, then Susil, and even Bamreji, and all the AG Srinivas, almost we can say the scientists from different the field, particularly they planned very systematically to address this pest. And because of attack of this almost 140 scientists who have witnessed today, this pest is totally in a resting period now. So after this 2 year, I think you have seen, there is no much the sound from this fall armyworm. It must be hibernated somewhere, and after some time, as I said, it may certainly come like a locust insect. Once certainly it will come. So you are all planned that, and as we requested you, since we are the agriculture scientists, we should not try for the chemical or killing of any of the insects. So today's pest is tomorrow's friend, absolutely, which may be a balancing friends in the future. So instead of killing that by the chemical molecules or new chemical molecules, so I requested all of you, the young generation, to plan your research in a manageable way by various means. So like the climatic management, yes, absolutely excellent papers have come, and they have proven that this pest can be efficiently managed through manipulation of climatic factors. Of course, that is not in our hand. It's almost the god and goddesses in the, so they can decide how the climate can be manipulated. So if it is a dry spell, of course, this infestation is many more. And if it is again rainy, the nature is taking care. Varuna will taking care. The other way, of course, to address this crop management through the bioagents. So in the presentation, I was just observing in the biofactors like bioagents, there is huge, the research is going on, but availability of the bioagent is a challenging. Yes, absolutely. That way, I think this you can make an excellent business, not only for your farm. So any kind of the insects you can manage to save the nature. So you have to think of especially production or identification of bioagents, so which are predators or those they are available in the nature. So certainly we can address any kind of the dreaded pests. So that way I think there is the issue where the young generation can think you can bring as an entrepreneur. The last sort is, of course, we have to worry about the complete killing of the pest is only to get the yield. So if you target only for the yield, certainly we have to forego for the nature. So we should not spoil the nature. And particularly all the scientists, I have observed all the posters also, it was excellent. Most of them you are targeting with bioagents, with all the different, the IPM, the pest, integrated pest management with all the different kind of approaches. It's an excellent issue. So it shows you people realized, so one should be dependent on this nature loving, these agents to manage this kind of pests. So actually, I'm so happy, and particularly I'm really unfortunate to be with you, especially for all the 2 days, and the Sharan has taken a lot of interest, his team, especially all the Naveen from agronomy, and they are the conveners, and of course Nagarajappa, even yesterday the Shantan Gowda was there, and all his dynamic, the student friends, and particularly my officer friends were all the co-chairs and the convener of the dean of this college. So he has given a lot of support, and this is the strength especially of this INCOFA. So friends, this occasion we created through our entomology department. Hope you all remember lifelong, because this is the first international conference made very success. Let's give a big hand to this that team. Yeah, finally, of course, I congratulate all the prize winners who have presented excellent talk. It doesn't mean that those who have not get the prize, they are not presented the paper. They have excellently presented, but no doubt there should be only in the race 1, 2, 3. So that we must remember. With this spirit, I congratulate especially Bamreji for having brought his all young team. I compliment him, and I thank the Honorable Vice Chancellor of that university, all the officers, the deputed, especially all the scientists here. So also from NBR, ICR, and also the university from Telangana, then TNAU. So I congratulate all of you and thank all your the university for having supported. So let us in the future we can address this crop, and as the suggestion given by our Honorable Vice Chancellor Dr. Udkeri ji, you must remember. So that is the suggestion you have to implement, and it is given the task to our university. So let us have a network project on in this fall armyworm under the leadership of again Sharan. For him we are there to support all kind of the support to him. We shall have a network project immediately submit to the agency, and we can develop. And of course secondly, as he already suggested to have that what he called to open the journal, especially that's ultimately whatever research you conduct, it should be published. Then only people recognize what immediately he noticed that the infestation of the pest, he published a very good paper in the high NOS rated journal, and internationally the CIMET scientist has identified, and he supported immediately to come over the Japan and for his lead talk there, and from then he supported. That means he shows your research should be identified, recognized at national level, and it should be published. So that is another very, very important the suggestion he has given. And the another one, of course, to establish center of excellence, because the fall armyworm is identified in this campus and in the surrounding of Shivamogga district. Absolutely, it should get that advantage as per the Udkeriji's suggestion. Let us have that submit to the concerned agency, and we shall establish the center of excellence for the fall armyworm. Hope you are all fall in that the insect now, isn't it? You are after that insect. So we should be, all this team should not leave from today itself, okay? It should be continued, fall army. Yeah, it's all I said even yesterday also. It is a family for fall armyworm. You should fall in that, And behind that, you should struggle for that, because it will certainly come and disturb you in the future, and food system will spoil because of this, because it is a polyphagous nature, you must be knowing all. So we have to run after this. So with these friends, I have taken much of your time, and if any lapses in the organizing, leave it to us. Of course, Dr. Srinivas said very nicely, the 4 pillars has taken well care. In spite of that, maybe he has given his opinion, hope you are all endorsing, but still, if anything negatives are there, leave it to us, and certainly we will improve in the future. But Shivamogga is known for hospitality. So Shivamogga is known for hospitality, and all my colleagues and Vice Chancellor behind us, he said, don't bother about any money, you just take care of the people, delegates, they have to carry the sweet memories of this campus. So he tried, we tried our level best. So hope you never feel bad, because you can come anytime to this campus. Tomorrow you are visiting our university headquarters. It's in Iruvaki campus. That is also campus is most beautiful that. And whenever you find time, come with the family or couple. So this Shivamogga is an excellent for the tourist place. A world famous, the Jog Falls is there. It is one of the 7 wonders in the world. You can enjoy, come in the month of July or somewhere in August. You will really enjoy. Those who are coming from outside, I'm telling. And you will enjoy really with your the beloved young ones also. So with this, I thank all of you, and I congratulate once again to Dr. Sharan, Dr. Ranman Swami, and University Head Dr. What is that, Hanumantraya, and his team, of course, all the entire team. I don't want to name one by one. There are a very dynamic and vibrant entomologists. They showed they can do anything in this campus, especially, and they have really ignited to start any kind of the international conference. This is nowhere uncomparable, or we can say our university is very, very happy with this, and we are proudly say, not because of exaggeration, all your feedback, we are very happy. So tomorrow, let's enjoy, go and enjoy with the nature, and visit to this campus. So with this, I thank the organizer once again, and I say goodbye for your future life. Thank you.
Thank you very much, sir, for your inspiring presidential remarks. Every successful event is built on hands working with dedication, and also on countless minds contributing with passion. It is now time to express our gratitude to all who made this possible. I now invite Dr. Naveen N.E., Scientist of Agronomy, as well as the Co-Organizing Secretary of INCOFA 2025, to deliver the vote of thanks. Please, sir.
Thank you, Chaitanya. So kindly bear another 5 to 10 minutes because the many have put their hard struggle and the nights to get success of this conference. It's a time to thank each and everyone who stood with us for the successful completion of this international conference on sustainable management of this fall armyworm. First of all, I would like to thank Dr. B.M. Prasanna sir, Simit Regional Director of Asia, for accepting our invitation and inaugurated this conference and delivering the keynote speech. As a organizing committee, we thank our B.M. Prasanna sir. And secondly, I would like to thank Dr. S.N. Sushil, the Director NBR Bangalore, for his presence and delivering the keynote address during the inauguration, sir. Thank you very much. And next, I would like to thank Dr. S.S. Udikeri sir. It is a privilege that I also worked with Udikeri sir in the cotton technology mission and cotton. Really, it was a wonderful. We got a very good team at the Cotton Research Station. So, sir, really thank you for accepting our invitation to grace this occasion as a valedictory address. Then we thank our organizing committee. Would like to thank our Honorable Vice Chancellor, Dr. R.C. Jagdisha, for serving as the driving force really behind with this event. His support was instrumental, really. It should be in a befitting manner. So, sir, thank you very much for your support. So, and we extend our sincere gratitude to our Director of Education, Dr. Hemla Naik sir, and the in-charge Vice Chancellor KSNUAHS Shivamogga for his continuous support. Since from last 2 days he was with us. So from morning 9 to evening was continuous support throughout the conference. Sir, thank you very much. And we extend our sincere thanks to all the key officers. So I don't want to name them individually, all the officers of the university who attended the inaugural and valedictory function. And I especially thank the campus head, Dean, the agriculture, so who was stood with us for almost 2 days, who has given all the support for this grand successful. So thank you one and all. And we express our sincere gratitude to the chairperson and the co-chairpersons of the various committees for their constructive suggestion and guidance for the better improvement of this scientific forum. And I thank all the committee chairmen and as well as chairperson and also for the rapporteurs. We express our gratitude to the lead speakers and the keynote speakers who have given and enlightened the present and as well as the current and the past research who has worked on in the different, almost more than 10 keynote speakers and the lead speakers from the different countries, almost more than 10 countries they have delivered this talk. So once again, I thank them for their wonderful deliberations. And we thank our giant collaborators, basically the ICR, CIMIT, CRISAT, US, the Harvard, and Entomological Society of India for joining hands to get success of this international conference. And we extend our sincere gratitude to our esteemed sponsors with their generous support. Really, without their generous support, this international conference would not have been much. It will be in the good, now we are in the best. So because of their generous support, so Syngenta, Multiplex, Bayer, PI Industries, Crop Care, PC, BASF, Cortiva, UPL, Godrej, Woolworth, GU, Karnataka Soaps, and among them, so most of them, they have already vacated this place. So on behalf of them, I will call Raghunath Pheromone Chemicals to receive the token of love from the organizing committee. So please Raghunath, come over here and take on the Pheromone Chemicals from Hyderabad. And we thank the professor and head department of all the departments, the scheme heads, ADRs, KVK heads, teachers, non-teaching staff and supporting staffs of College of Agriculture, Shivamogga and the other campuses also. We thank the press and media persons who were all the deliberations covered during the conference and gave the wider publicity. You could have been seen the today's all the leading newspapers, it was been covered. More than 6-7 newspapers it has been covered. And we thank all the particularly the participant delegates who come across all over India, okay, to take part in this conference and delivered all your presentation, both in oral and as well as the post presentation. We wholeheartedly thank you for your participation. And special thanks, as Srinivas was telling, really it is a very happy moment to thank. First one is logistic support provided by the College of Agriculture, Shivamogga, and particularly I want to mention few names, Manju, Chandru, and as well as Raju. So there are many, Ashok, there are who have worked 24/7 because some of the times the delegates have reached by 2 o'clock, 4 o'clock. They took us the delegates and they have been safely reached their origins. And particularly as our many of the delegates said, a special thanks to our person who prepared and delivered the delicious food, Mr. Raju. I request the Raju to come on the dais and receive the token of love from our organizing committee who served the excellent food to all of us. Please give him a very big request, entire. So some of the participants were telling, sir, आप शादी का खाना खिला रहे हो क्या कॉन्फ्रेंस का खाना खिला रहे हो? Thank you, Raju. And I also thank the audiovisual aid person, particularly electrician Anumantu. Please come on the dais and receive the token of love from the organizing committee, because since all the 2 days he took the charge of this electrician activities. Please, both of you can. Ibru bani. Iskoli, please. Innonu ankodi. Soap inni ankodi. Inankodi, inankodi. Sure. Yes, sir. And next man is our photographer. So, Mr. Suresh, please come on the dais and receive the token of love. He was so happy to take the picture of everyone. I think since from yesterday, he has taken more than 2,000 photos with all smiling. Thank you, Suresh. Yeah, and for the entire 2 days deliberation, the live streaming was done by the Kannada medium news channel. So, Mr. Kotresh, please receive a token of love from the organizing committee. Thank you, Kotresh. Thank you, one and all. Now, on behalf of the organizing committee, we thank, yes, close. On behalf of the organizing committee, we thank particularly the head of the Department of Entomology for his continuous guidance and support, wherein instrumental to the conference successful, and the entire department also. And we thank the student cultural committee team who entertained us for almost 45 minutes from our students and all the participants from Arunachal Pradesh, Maharashtra. They also entertained us. Thank you, one and all. Really, I request all the Entoverso, please stand up. So this is a success if the Entoverso was not there. All the Entoverso team, PSD, team of Entoverso, please. Entoverse, please come on the dais. Okay, this was really successful because their extraordinary dedication for nearly, sir, almost a month. So for 24/7, they worked like anything. So usually we win, sir, particularly in insect, we tell some of their nocturnals, but these insects are 24/7. Really. So really we thank from the organizing committee to all this young and energetic Entoverse. Let us give them a very big clap. Without them, really, it could have been not completed. Right idea. ಯಾರು ಸಣ್ಣರಿದ್ದರೆ ನನ್ನ ಮುಂದೆ ಬನ್ನಿ. Thank you one and all. Nice. And made it success because of all these students. Yes. And finally, we owe a special thanks to our organizing secretary, the captain, champion, Dr. Sharanabasappa Deshmukh, whose vision and the leadership convinced this conference. He devoted nearly almost 3 months for his restlessness, meticulous effort to get this conference to be most successful. So thank you. Felicitation. Yes, sir. Please. I request. Yes, sir. I also thank the young master of ceremony who took all 2 days with her smiling. So took all the, so let us, he is also a student of Dr. Sharanabasappa, completed recently PhD in entomology, Chaitanya B. Patil. So I request uh, yes, and finally I thank one and all, so who directly or indirectly supported for this grand success of the event. Thank you one and all.
Thank you very much, sir, for a meaningful expression of gratitude. I just want to mention that it was my guide who has, Sharan Basappa sir, who had confidence in me that I could host this event. I thank Sharan Basappa sir for giving me this opportunity. Thank you, everyone. And we would it's an honor to also present a memento to Dr. Naveen sir, the co-organizing secretary of the INCOFA. And it's also a moment to present a token of love to our Dean sir, Dr. D. Tippesha. It's okay. It's okay. Once. And head of the department, Dr. B.C. Hanman Swami sir. And as well, the senior most professor of entomology, Dr. B.K. Shivanna sir. I also request the other co-organizing secretaries, Dr. Girish sir, and Dr. Rajasekarappa sir to please come on the dais to receive a token of love. Rajashekar. Thank you, everyone. I thank all the dignitaries for presenting the memento. So before concluding this valedictory ceremony, I request Dr. Nagarajappa Adivappa sir to please come on the dais to receive a token of love. Please, sir. And as well for Dr. H.K. Virana sir, Controller of Examinations of KSNUAHS Shivamogga. Yes, or that is the next name. Thank you, everyone. So before concluding this valedictory ceremony, I request everyone to kindly rise for the national anthem to honor our nation.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
1 second. I request Dr. Vinay Kumar sir to please receive a memento.
Please receive a memento.
And also Dr. Shantan Gowda sir. So with this, we formally conclude the valedictory function of INCOFA 2025.